Welcome everyone to the Regional Science Consortium's 16th Annual Research Symposium. I'm Jeanette Schnars, the Executive Director of the Regional Science Consortium, which is located in the research wing of the Tom Ridge Environmental Center at Presque Isle State Park. The RSC is a great organization composed of colleges and universities, state and federal agencies, school districts, and other nonprofits supporting our mission of scientific research and education in this region. During a time when scientific research is more important than ever, we are excited to host the 16th Annual Symposium and our first 100% virtual conference. Real science brought to you virtually. Over the next 3 days, we have a great number of presentations which will be live streamed, including 55 oral presentations and 24 poster presentations, given by researchers, faculty, and undergraduate and graduate students. In addition, we will also have 2 evening presentations for a general audience, highlighting some of the most valuable environmental resources in this region. As you are viewing now, presentations will be live streamed on the RSC YouTube channel at www.reachscitv.com. However, also tune into our poster session later today, starting at 4:30, on a platform called Discord. You can get more information on our website, how to tune in. You will also find the proceedings for the symposium, which lists all the presentations and their presentation times on our website under the Symposium button. Also, tonight at 6:30, our guest speakers will be David Frew and Jerry Skripczak, talking about their most recent publication, Accidental Paradise, A Natural, Political, and Social History of Presque Isle. Tomorrow evening at 6:30, we welcome guest speakers Brian Gula and John Laskos from PADCNR, and they are the environmental education specialists there. Finally, I encourage all of you to tune in Friday at 1 o'clock for the closing of the symposium and the presentation of student awards. I'd like to thank the RSC staff, including Jen, Amber, Sean, and Sarah, for all their hard work in planning this event. I would also like to thank the RSC board members that assisted in the planning process for the last several months. I'd like to thank our sponsors that realized the value of the symposium and continue to support the RSC over the last several years. I'd like to thank WQLN Public Media for sharing this event with their viewers, and finally ETD Photography that has made this event possible and has done so through multiple levels of media support and live streaming here at Radius Cowork at the Renaissance Center in downtown Erie. So now we get to sit back and enjoy the next 3 days of presentations and learning about the scientific research that has been conducted over the last year, despite all of the roadblocks that we might have experienced. We will get started with our first session today. If you have any questions, I encourage you to use the chat box, and please do so during the presentation, and then at the conclusion, I will be relaying those questions to our speaker. So I'd like to go ahead and introduce our first speaker. And for those of you that are serving as RSC judges, you will not have to judge this presentation. Lynn Beatty is a faculty member in the biology department at Penn State Erie, the Barron College, and she will be presenting today on an unsolved mystery snails. Lynn?
All right, can everyone hear me? I can't hear anything through the Zoom, so I'm not really sure where we're at. Okay, thank you. Let me go ahead and share my screen with you guys. All right. All right, so hopefully you guys can see that on there. I have the live stream up over here and I can see my face, but not my presentation. But maybe it's just delayed, so. Okay, I'm gonna go ahead and get started. Looks like it's up. So I'm Lynn Beatty. I'm an assistant professor at Penn State Behrend, and I'm going to talk to you guys about some of the research that my lab and the Simpson lab and the Newt Tile lab are doing on some invasive snail species that we have here in Western PA. So let me just start by saying that we should all be familiar with the idea that invasive species are detrimental to the ecosystems that they invade. And in particular, aquatic ecosystems and invasive species in those aquatic ecosystems are considered to be one of the greatest under threat by invasive species. So the first step, of course, in figuring out what invasive species are there and how to prevent and remediate their presence is to actually figure out who's there and how abundant they are. And one invasive species or group of invasive species that tends to kind of sink under the radar are snails. Right? And the reason being they're not the easiest to ID. Not a lot of people like me go out and look out for them. A lot of people just kind of pass them over, but they can really have detrimental impacts to the ecosystems that they invade. Not only do they compete with native snail species, which play important roles in aquatic ecosystems, but they can also transmit parasites because nearly all trematode parasites have to have a snail in their life cycle at some point, which means we could be either augmenting native host parasite dynamics, or we could be introducing native parasites that came over with the snails. So that's a big issue. So let me introduce you to our natives, or our invasive snails, I should say. The first is the Chinese mystery snail. The Chinese mystery snail is native to Southeast Asia, and it is quite large. It's a large snail. It ranges from about 35 to 65 millimeters when they're adults. And they were initially brought over for human consumption. So they were brought over in the early 1900s thinking like, yum, this would be delicious. And they're called mystery snails because they're actually ovoviviparous. So they give birth to eggs that the female immediately breaks open with her foot, and then you have the young that crawls away. So you start out with one snail and then it's the mystery, you have 30 more, seemingly out of nowhere. Our second invasive species that we're working on is the Japanese mystery snail, which is also native to Southeast Asia. It looks very similar to the Chinese mystery snail, but it, it looks very similar, but it can get quite a bit larger. It can get up to 70 millimeters long, and it's a bit pointier compared to the Chinese mystery snail. Although there are reports of a second morph of the Chinese mystery snail that looks like the Japanese mystery snail, but is much smaller. They're also ovoviviparous, hence the mystery snail. We do have a third species that we're looking at, and this is the faucet snail, also known as the mud bithynia. These snails are a bit smaller than the mystery snails, and I do have one here just to kind of show detail, and then I have one kind of indicating its scale relative to the mystery snails, so they're a bit smaller. They lay eggs. They're primarily filter feeders, and these ones are known to transmit parasites that can be lethal to migratory waterfowl, and that study was done in the United States. So in the US, these guys do have an evidence, or have been shown, I should say, to cause detriment to native populations. So like I said, for invasive species, we need to figure out who's there, where are they, how many are there, and that's kind of the outline for this talk today. So I'm going to first tell you where they are, where we found them so far, how many of them do we think there are, how does their size vary between sites, because size is really important for a number of different aspects of their life history. How many eggs they have, for example, or how many offspring they have is dependent on size, plus how big they are influences who can eat them and what they can eat. And then I'll tell you about what we're planning to do next, what we'd like to do next, what's kind of our big dream for the future. So first, let's start out with where are they? And I should preface this by saying we've only been doing this for 2 years, and one of them was 2020. So you have to acknowledge the data we have so far is the data we have, but of course we're going to keep collecting this in the future. So in 2019, we started looking for these guys. We focused primarily on Presque Isle State Park. So I have Presque Isle State Park shown here. The X and Y are just Latin long. And in 2019, we sampled 95 locations over 3 months. So the different colored dots on this figure show you which month the sampling was conducted. Blue, May, green is June, et cetera. And you can see we repeat sampled some months just to kind of double check how their presence changed over time. And this was done with Dr. Simpson, Dr. Newtile, and Austin, Matt, and Cassidy were our undergraduates that really helped out with this sampling. So at Presque Isle in 2019, we detected 322 Chinese mystery snails. Here again is our map of Presque Isle with our locations that they were found, as well as the color indicating what month they were found. So you can see that most of them were found later in the summer, primarily in June and July. We did find one shell down here at the Feather Observation Platform in May, but we didn't find live individuals. And when they were found quite dense populations, especially in Thompson Bay and near the boat launch in Graveyard Pond. And we primarily just picked them up by hand and found them crawling either on the substrate or on submerged, coarse woody debris or vegetation. We also found a number of faucet snails on Presque Isle in 2019. These were primarily in the earlier months. So in May, we found a lot more than we did in June and July. And most of these, or at least the highest apparent densities, were in the lagoon area. So we didn't sample entirely in the lagoons, but we sampled kind of the area adjacent to Graveyard Pond, as well as the bridge that connects the lagoon to Marina Bay. And they were, as I said, quite dense. This picture I have here shows the side of a boat launch in the lagoons, like where you can launch canoes off of there, kind of those metal structures. And these are all faucet snails, just present in a small area, just on one boat launch. And it was all the boat launches were similar. So they were present in quite large abundances, but we didn't find any Japanese mystery snails on Presque Isle, which is interesting considering they've been detected in the Ohio area of Lake Erie. So in 2020, we were able to broaden our search quite a bit. It was just the dynamic trio, me, Dr. Simpson, and Dr. Newtile, obviously because of COVID, but we still managed to look at 360 different sample locations. So we were able to sample Presque Isle again, where we again found faucet snails and Chinese mystery snails. We also looked at Lake Pleasant, where we found just Chinese mystery snails. We sampled Lake Canadota, where we found just Chinese mystery snails. We sampled a pond in Sagertown, where we had been told that there were mystery snails present, and these were Japanese mystery snails, which I've indicated by blue here. We also sampled once at Pymatuning, where we found both Chinese and Japanese mystery snails, but the Chinese mystery snails tended to be on the western shore. We didn't find very many, we only found 3. And then the Japanese mystery snails tended to be on the eastern shore of Pymatuning. But again, that was just one sampling occasion. We haven't been out there very much. Also in 2020 is when we started to get smart and bring a quadrat with us. So that way we could provide density estimates so we could figure out how many there are. And that's what I'm going to do next, is tell you about our different density estimates for our mystery snails. So here I have, again, our map where we found all of our mystery snails in 2020. The color code indicates what species there are, with the reddish being the faucet snail, the green being the mystery snail, and the blue being the Japanese mystery snail. Those colors also correspond to these data points on this figure on the left. And on this figure, I have our 5 main sampling locations on the x-axis and our snail density in meters squared. So snails per meter squared on the y-axis here. And I should say that we primarily sampled in the shallow area of the shore. So we didn't really sample deeper than 1.5 meters or so. We didn't go over our waders. We did kayak around a few areas just to see if they were present, but these density estimates primarily come from really shallow parts of the shore. So first for Lake Pleasant, you can see that, Lake Canandaigua, you can see that there was moderate density, about 1 snail per meter squared. Most of these snails were on the western shore of Lake Canandaigua, which was interesting. There were very few on the eastern shore. And the reason that's interesting is at Lake Pleasant, you can see density was quite high, on average 2 snails per meter squared, and this was on the eastern shore. Those also correspond to where the respective boat launches were in either water body, so that could be indicative of a mode of transport for each of those species. At Presque Isle, I have both the faucet snail and the Chinese mystery snail densities here in their relative locations. You can see density is still quite high. I forgot to mention too, these numbers up top represent the number of quadrats that those values are based on. So it's the number of samples. For Pymatuning, the density of Japanese mystery snails was much higher than the density of the Chinese mystery snails. Like I said, we only found 3 Chinese mystery snails, so not very many. But we did find quite a few Japanese mystery snails, particularly juveniles. And in Sagertown, density was also quite high. So we have fairly good density for each of these invasive species, which I guess is not good after all, at a number of locations in Western PA. So in terms of size, which again is important for a number of life history values and in terms of the impact on the environment, let's first look at the mystery snail size because they do operate on quite different scales in terms of our y-axis here. So again, I have our blue being our Japanese mystery snail, our green being our Chinese mystery snail, our 5 main locations on the x-axis, and the size of the snail on the y. So you can see at Lake Canandaigua, where they were a bit less dense, they were a bit bigger. Most of these snails too had kind of a pointier morph of the Chinese mystery snail. At Lake Pleasant, most of these snails were quite small or quite short, and they were quite dense. So it could be indicative that density indicates growth rate, or not indicates, influences growth rate. Presque Isle as well, we have quite short Chinese mystery snails, while our Chinese mystery snails at Pymatuning were quite large. Again, there were only a few of them. And we do have a small average size for our Japanese mystery snails, but this is due to the fact that we found so many juveniles. And it's that large number of juveniles that's bringing down this average size because they are quite large. Here, for example, is the shell of one adult that we found while we were sampling Presque Isle. That's about 70 millimeters long. For those, I have really large hands for people that don't know my hands. So this shell is quite large compared to my hand. And then of course, Sagertown, we did find a lot of adults and some juveniles, 12 individuals in total at Sagertown. So we have quite a range in size, but they were quite big. For our faucet snail size, again, we only found these in Presque Isle. So I've broken down our locations based on where we found most of the faucet snails. So here I have that map of Presque Isle, just so you can correspond these locations to where they actually are. And these numbers represent the number of snails that were measured to get that mean size. So at the bridge, we did find a large density of snails. I think all 47 of these were in 1 quadrat. So they were quite dense. And they were of moderate size, not near the max of 15. Feather observation, they were quite small. Graveyard pond, there were quite a few of them, and they were of moderate size, mostly close to the boat launch and on the vegetation. Lily pond, we didn't find very many, hence the large variation in size. In Thompson Bay, they were quite big. So these snails did appear older, their shells were more worn. These are few individuals, so we need to collect some more individuals from Thompson Bay to get a better measure of size. All right, so now what's next? Where are we going? And we have a lot of things planned. It all depends on how things go, of course. One of the things we'd like to look at and document a little bit more finely is this variation in shape. As I mentioned, there are 2 morphs of Chinese mystery snail, but I think it would be good to document and actually quantify using geometric morphometrics that variation in shape, so that way we can maybe even categorize these shells a little bit better, because just looking at the spread, you can see why it's difficult to distinguish these species from each other. We also, along those same lines, want to look at genetic patterns of species between all of our different sites, because there are some indications that maybe we have hybrids. I don't know if they hybridize. They are very closely related. But just as an example, this here is the foot of a Japanese mystery snail. You can see it's quite yellow. This is the foot of a juvenile Chinese mystery snail, which is dark gray. And this is the foot of the pointy Japanese-like morph of the Chinese mystery snail, which is both yellow and gray, which could suggest that maybe that's a hybrid, but we need to do some genetics before we actually get into that. We also are interested in looking at the kind of microcosm that goes on the shell of these mystery snails. So not only are they invading habitats, but they're also creating habitats on their backs. They're really slow moving, lots of things grow on their shells. So who knows what's going on there? What are they fostering? We also want to look more at the parasites that these snails have. Did they bring any with them? Are they transmitting any additional, are they serving as an additional venue for other parasites that are native to the area that might be then affecting native snails, native wildlife? And we do want to look at what they're eating to better know if they're competing with native snail species, and how that might affect their growth. There are studies that show that they do, I mean, these guys eat indiscriminately. They filter feed, they eat whatever they can put their mouth on, and whatever they eat tends to influence their growth rate. Especially if they eat a lot of diatoms, they tend to grow bigger, which could be influencing these morphs that we see as well. So with that, I want to thank Penn State Behrend for funding me and my students to go out and look for snails, different regulatory agencies for letting me search and collect snails on state land, the Western PA Conservancy for allowing us to sample and collect snails at Lake Pleasant, as well as different folks, the Behrend Biology Department for being supportive with all of our stinky snail stuff, Behrend Maintenance for dealing with all of our stinky snail stuff, and then of course the undergraduates that helped collect a lot of this data. So if you're interested in hearing more about mystery snails or about just research that Behrend undergrads, at least in the biology department, have done, there's a lot of opportunities for you later today. Immediately after this, my students, Joey and Brianna, are going to be talking about their research with different ways that we can assemble amphibian coverboard arrays, so that way we can come up with best practices. Right after them is Cassidy, who's going to be talking about some of our population models that we've constructed for these mystery snails, try to figure out how we can control them. Later today, this afternoon, Sage is going to be talking about the mystery snails and all of the heavy metals they consume, what they do with those heavy metals. And then tomorrow, we have two students from Dr. Newtile's lab talking about their toxicity work, so a behavioral assay that they've developed to better assess toxicity, as well as the toxicity of urea, which is a common road salt substitute. So with that, I'm done, and I will take anyone's questions.
Hi, Liz. This is Lynn. This is Jeanette. Can you hear me?
I can hear you, yes.
We did have one question from Dr. Mike Campbell at Mercyhurst University. First off, impressive data set with an exclamation point, so very exciting.
Thanks.
He wants to know, is there any observational evidence that other common snail species in these habitats coexist or not with these invasive species?
So just in our sampling, we have been keeping track kind of loosely, not officially, of the native species present in the same area. And there are some that you kind of always find. You always find physids in pretty good abundance. Really, the only species that's close in size to the mystery snails is the pointed Campyloma that is in Presque Isle, but we seem to find those closer to the lagoons. I know that there are studies that have documented negative effects of, I think, Chinese mystery snail on some Helosoma species, which are kind of the ram's horn snails. But it's kind of hard to tell since most of these guys have been here since the 60s. Everyone we ask to actually think they're native. So we kind of have an issue with shifting baseline syndrome here, where people don't know that they're invasive. So we don't really have a point of comparison to go back to necessarily. But we could look at direct interactions between species in the future, definitely.
We have another comment. Great presentation. Do these invasive species have any native predators that you know of?
No. I assume that probably your usual snail eaters will eat them. We do see some shells that have evidence of like bird damage, like beak pecks in them. I would imagine things like raccoons might eat them as well. At least the faucet snails, waterfowl will eat because they're small, right? But the big guys, I think it has to be something bigger. I know turtles also eat them, and maybe crayfish, the smaller ones anyways. But once they hit a certain size, the crayfish actually can't do any damage. I would love to know more about who's actually eating them though. So if anyone has ideas on how to get a better idea of that, let me know.
Okay, Lynn, we have another question from a fourth grader at Iroquois Elementary School. And they want to know, how deep in the water have you gone to collect these snails?
So I am about 5'6", maybe 5'7" on a good day. And I've gone up to my rib cage. I've gone into my waders to get them. So about up to here, which is pretty deep. It was a mistake. I shouldn't have gone that deep. We do have grabbers now though, like claws that you use to pick up trash and things. We use those to pick up snails. It helps.
Well, great, Lynn. Thank you so much for your presentation today.
Nope.
We're looking forward to the other presentations you mentioned by your students. And thank you again.
Bye, guys.
All right, so we will be moving on to our next presentation. And for our judges, this presentation is by Joey Forish, Forish, and Brianna Payton. And they are both undergraduate students at Penn State Erie, the Barron College. And they will be presenting the effect of covered board array design on the probability of amphibian detection. And so Joey and Brianna, we're excited to have you here today virtually, and we will pass it off to you. If we could just pause for 1 second, we're going to switch things over.
All right, good morning, everyone. My name is Joey Forish,
And my name is
and we worked under Dr. Lynn Beatty at the Penn State Barron Biology Department, and our research project focuses on the influence of coverboard arrangement and abundance on amphibian detection within the Wintergreen Gorge area.
So the objective of the research was to determine the number of boards, spatial arrangements of these boards, influence the probability of detecting amphibian species. The species that we're most interested in are those that around Penn State's campus, such as redback salamanders, Eastern American toads, and red-spotted newts. And by spatial arrangement, we're referring to the distance between the boards, and the count of boards refers to the number of boards within an array. This research is important because currently the influence of coverboard arrangement and design is unknown on amphibian detection. And by filling in this gap within the research, we can highlight the functionality and reliability of coverboards as a mechanism of estimating populations and monitoring individuals. By knowing how the amphibians or other species prefer to orient themselves within an array or between a certain board, it will be easier to find them compared to looking through their natural habitat. And also the coverboards serve a good purpose. They offer a microhabitat for the species within their natural environment. These microhabitats have more stable conditions and offer a little bit of protection as the species would reside underneath the board.
So if you are curious, coverboards are thin pieces of wood, metal, PVC plastic, or other natural materials that are, as Brianna said, used to create a microhabitat for amphibians and other organisms that may find these as a temporary home. Best practices have come out of studies that use native wood that is untreated, as it serves a similar home to that of what the organism or amphibian would find normally. Some problems have come out of studies that are using metal boards as they provide premature heating coming out of the winter months, which could be detrimental to the species. And treated wood has the danger of toxin addition, both to the amphibian species through their permeable skin or the environment through leaching into the soil. Coverboards are inexpensive for deployment and maintenance, as you normally just have to acquire the wood, cut it up, set them out in the array that you would like, and then wait for your results to come to you. Scientists have implemented population dynamic studies using coverboard arrays for over 50 years, as they pose little to no harm to the organism and the environment. And the observation process poses little disturbance, again, both to the environment and the organisms. So for our specific study, our setup and data collection, we set up our arrays in 3 separate locations. Those are the Devil's Backbone, Happy Hollow, and Golf Course. And each location contains 100 boards split into 3 arrays, as noted in the middle figure here, totaling 300 boards. Our A array contains 50 boards that are 5 meters apart on each side. Our B array contains 25 boards that are 5 meters apart on each side, and our C array contains 25 boards that are 2.5 meters apart on each side. Through our data collection season, we observed and recorded biotic and abiotic conditions. Some of our abiotic conditions that we recorded are weather, temperature, both underneath and above the boards, moisture and pH of the soil, barometric pressure, elevation of the locations, and canopy cover from the surrounding trees. And some of the biotic conditions were weight, sex of the individual, species, snout vent length, tibia fibia length, and tail length. We utilized aseptic technique to make sure we were not spreading any viruses or anything throughout the same ecosystem or transporting it to another ecosystem. And our boards were checked weekly to ensure a routine check of all amphibians present. So for our first season, we detected 10 different species, with our redback salamander being the most common with 65 detections, and our northern dusky salamander and our Eastern American toad only having 1. And then as you can see, all of the other ones followed suit. We didn't really find much of the other ones, but we're hoping that through further investigation, we will.
So in order to analyze and assess how the variables we manipulated in this experiment influence the probability of detecting the 10 species previously mentioned, we had to run a single species occupancy model. This type of model estimates the probability of a species occupying a certain site and then also the probability of detecting that species within the site. These models were ran for each of the 10 species. And before running these models, we had to consider many combinations of variables and factors. Some of the combinations and variables we included were those related to our experiment, such as those we manipulated ourselves, which were board distance and board density, or the count within an array. We also had to consider location, as we had 3 different locations for our array design. And then we needed to account for any factors that weren't considered in this experiment, as this created our null model, which stated that no factors within this experiment influence the probability of detection or occupancy. And that was what we wanted to dispute. So all in all, we ended up creating 16 different combinations, which we ran for each species. And the validity of those combinations was tested through Akaiki's information criterion, also known as AIC, and this estimates the quality of statistical models. And those combinations with a delta AIC less than 2 were the ones that were most supported. Unfortunately, only 4 of the 10 species which we found throughout our season had the most supported models that involved the variables we actually manipulated. And those included the bullfrog, the redback salamander, the green frog, and the four-toed salamander. And we'll discuss those models right now. So for the bullfrog, of all the combinations that were the most supported, location influenced the probability of occupancy the most, as you can see in this figure. The highest probability of occupancy was found at the golf course location, followed by Happy Hollow, and then Devil's Backbone. And the probability of occupancy for Devil's Backbone was so low as we didn't find any individuals there at all. In regards to the variables that influence the probability of detection, the combinations that were most supported include the number of boards in the day of year, which is represented by this figure. As you can see, the highest probability of detection of bullfrogs beneath a board was in the arrays that had 50 boards compared to 25. And you can also see that the probability of detection was a lot higher in the arrays with more boards earlier in the year. And we began our sampling around the end of August and beginning of September. So that would have been when the highest detection was. The second most supported combination for the bullfrog was distance between the boards and day of year, which influenced the probability of detection. As you can see in this figure, the probability of detection was highest when the arrays had the distance between the boards of 5 meters compared to 2.5 meters, and also when it was earlier in the year.
So moving forward, our next best supported species was a redback salamander. And similar to Brianna, how she mentioned, the greatest occupancy model that we came up with was the variable of location. And as seen on this figure, the greatest probability of detection was at Happy Hollow, then followed by Devil's Backbone and the golf course, respectively. Moving forward, as you can see in this figure, the distance between the boards had a highly variable effect on the probability of detection, as the greatest probability of detection was found when boards were further apart or 5 meters apart from each other rather than that of 2.5 meters apart. And as you can see, in comparison to the bullfrog, these individuals were found later in the sampling season. Similarly, when looking at the number of boards and how they affect the probability of detection, the higher number of boards had the greatest probability of detection, and then arrays with lower counts of boards had lower probabilities of detection. And finally, in this final figure, you can see that our ARA had the greatest probability of detection, which was characterized by 50 boards 5 meters apart on each side, then by our B array, which was 25 boards with 5 meters on each side, and then finally our C array.
For the third species that had the most supported combinations, this would have been the green frog. Similarly to the last two species mentioned, the variable that had the greatest influence on the probability of occupancy was location. And this figure also is similar to that of the bullfrog, in that the highest probability of a green frog occupying a board was at the golf course, followed by Happy Hollow and then Devil's Backbone. And then also with this species, the probability of occupancy at Devil's Backbone was so low as there were no individuals detected there either. And then for the green frog, there was only one combination that was supported by the AIC that involved the variables we manipulate in our experiment, which were the number of boards and the day of the year. Here you can see that the highest probability of detection was in the arrays that had 50 boards compared to 25. And you can see that there was a great difference between the probability of detections. You can see that the probability of detection was greater, more so in the beginning of our sampling season. However, it doesn't change that much throughout the rest.
And finally, our fourth species that had supported trends was the four-toed salamander. Similar to the 3 species identified before, the greatest probability of occupancy, the variable that we used was location, and the highest probability of occupancy was at Devil's Backbone, followed by the golf course and Happy Hollow, as there were no occupancy. There was no occupancy recorded at either of the latter 2 locations. As you can see, when talking about detection for the four-toed salamander, there was a greater detection probability underneath arrays that had 50 boards, which was our ARA, rather than that of 25 boards in our B and C. And similar to the redback salamander, the probability of detection increased as we got later into our sampling season.
So overall, for the 4 species that had the most reliable and supported results, we can convey that the highest probability of occupancy kind of varied between the sites, but the highest probability of detection overall was between 0.1 and 0.4. Also from these figures, it seems as though salamanders have a higher detection later in the year compared to the frogs, which had a higher detection earlier in the year. And the probability of detection was mainly influenced by our variables board count and board distance. From the models that we've shown so far, you can see from the dashed lines and the error bars, these represent our 95% confidence intervals. And a lot of these had a wide range and were very far from the solid graphs. And this indicates that there were many values included on our confidence interval, which can convey that the probabilities we found of detection and occupancy may not be as close to the true value. Some other reasons for the variation in our data is because we had small sampling sizes. This was our first year for data collection, so there weren't many occurrences with a lot of the species, except for the redback salamander. This also relates to why many of the 6 other species of the 10 didn't have reliable models. And also with these species, the null model was the most supported for them, and that would convey that none of the variables we had measured or manipulated influenced their probability of detection. So it didn't really relate to our experiment.
So moving forward with our project, we would like to continue data collection over multiple seasons so that we can increase our population size and that way strengthen our trends and see what variables are providing the most information on occupancy and detection of these individuals. We would also like to further interpret other abiotic factor influence, specifically other abiotic factors that didn't happen to get into our 16 variable combinations. On the basis of species dispersal, we would like to find out whether or not inter or intra-specific competition between species has anything to do with the dispersal, as well as their territoriality and competition over available resources. And finally, we would like to monitor individual species and individuals to learn more about them, specifically through tagging or mark recapture, so that we can monitor their movement throughout the ecosystem, specifically their philopatry and board preference. This came to us as we noticed that our frog species in the golf course were constantly being found under the same board. And we would like to investigate whether or not that was a fluke for the season, or if that's actually how they go about their life as staying underneath one board.
We would just like to thank you for listening, and also, I know not Dr. Beatty isn't here, but she was a great help with this experiment, and we'd like to thank her as well. And that's it, sorry.
Thank you, Joey and Brianna.
Mhm.
We do have a couple questions for you. Did you observe any indication that a disturbance such as trail effects, occupancy of cover boards such as trail effects impact the occupancy of cover boards by the amphibians?
I think the most site that was influenced maybe by trail and footpaths was at Devil's Backbone, at Golf Course and our Happy Hollow, which was down further in the gorge. Those weren't really close to any trails, but at Devil's Backbone in our A array, some of the boards had been turned over and were broken just because they were near trails. And it turned out, and I don't have the numbers, but I remember that we had never found any species beneath those boards. So that could have correlated with high traffic and disturbance from humans as to why they wouldn't have resided there.
Okay, we have another question. How far were these boards placed from the edge of a forest? And can you remind me how big each cover board was?
The boards we don't know the exact distance from the forest. The golf course one was pretty much placed within the forest itself. Same thing with the Happy Hollow one. The golf course or the Devil's Backbone array was within the forest, but it was pretty much a sparse area as it was near the edge of the cliff. And I think with the boards, Joey, how big were the boards again?
The boards were 1 foot by 1 foot.
Mhm.
Okay, we have one more follow-up. Was the golf course location nearest to the water? And you gave a really great presentation.
Oh, thank you. The golf course location wasn't near any water. There was, it's at the golf course near Penn State Fair, and it's not used anymore. There was a pond sort of on the outskirts of our array design, but no water was near it directly. I think the only site where we had water nearby was the Happy Hollow down lower in the gorge, as it was kind of along a stream. And when it was really saturated, there was a pond sort of nearby the boards. But other than that, there was really no water nearby.
All right, are we ready to go? All right, Cassidy, we are ready to go, so we are going to turn it over to you. Thank you.
Hi, my name is Cassidy Lanowitz, and I worked with Dr. Lynn Beatty at Penn State Behrend, and I'm going to tell you about how I use integral projection models to evaluate data availability and sensitivity of two invasive snail species. So just a little background, invasive species are any species that significantly change or disturb the ecosystem it takes over. These species are also called introduced, alien, or exotic species and can arrive to new habitats through migration or with the assistance of humans. These species are also considered invasive when they have a natural advantage over indigenous or native species. Their breeding populations of these invasive species can especially prosper when there are no natural predators in their new habitat. These species cause ecological disruption by reducing the biodiversity of an area, and not to mention the invasive predators will reduce prey populations over time and eventually lead the prey species to extinction if that prey species cannot respond appropriately. In addition, invasive species can eat the food that the native species would eat and inhabit the places that they would live, leaving them without food or shelter. Two species that are classified as invasive in the US are the Chinese mystery snail and the Japanese mystery snail. These two snail species are very similar in their morphological appearance, but they differ in life history. The Chinese mystery snails are native to Thailand, Burma, South Vietnam, China, Japan, Korea, and the Philippines, while the Japanese mystery snails are only native in Japan, Taiwan, and Korea. They can both be found living in freshwater lakes, river ponds, rice paddies, roadside ditches, irrigation ditches, and slower regions of streams where they give birth to live crawling young. These Chinese mystery snails can grow to be around 25 to 38 millimeters, but can reach up to 64 millimeters. And the Japanese mystery snails grow to be approximately 30 to 65 millimeters. They are both described as having smooth, light, olive green to brown shells, and they tend to get darker with age. Their shells are slightly different, however, with the Chinese mystery snails having 6 to 7 whorls, and the umbiculus is small and round with the spire at an angle of 65 to 80 degrees, while the Japanese mystery snails has 7 to 8 whorls, and their umbiculus is narrow with the spire at 50 to 55 degrees. While they both have very similar characteristics, as I mentioned before, their life histories are very different. For example, the female Japanese mystery snail tends to live for 7 years, and the Chinese mystery snail female tends to live for 4 years. These different life histories allow for the Chinese and Japanese mystery snails to impact the ecosystems in different ways, especially in locations that they are deemed invasive, such as the Great Lakes. According to the USGS Aquatic Invasive Species Database and our previous observations, the Chinese mystery snails have been found in the waters around Presque Isle State Park, and the Japanese mystery snails are only reported on the Ohio region of Lake Erie. However, some Japanese mystery snails bear a close resemblance to the Chinese mystery snails and may only be distinguishable by genetic means. Therefore, we likely have an imperfect knowledge of the distributions of Japanese and Chinese mystery snails in Western Pennsylvania, and this causes an incomplete understanding of their impact on the ecosystem. So the objective of this study was to construct and compare population models for Japanese and Chinese mystery snails, and then determine the most sensitive life stages to target control efforts for each species. So to start out with my methods, the first thing I did was I wanted to determine the appropriate population model for these species. To do so, I conducted a literature review to collect different life history parameters on both species. These demographic parameters included age, growth rate, intrinsic rate of increase, density, fecundity, mortality rates, and sex ratio. After the literature review, we decided that we would create an integral projection model, otherwise known as IPM, and this model determines how an individual's state, such as its size or age, influences its vital rates, such as survival, growth, and reproduction. Here I'm going to explain a little about how an IPM works. So we started off with 10,000 individuals of randomly assigned sizes, and then in May through June of each year, the snails would breed. but snails that were at a certain threshold, which is 35 milliliters, could only breed. So once they hit 35 milliliters, they were able to reproduce. And then the amount of offspring produced is dependent on the female size. So the bigger the female, the more offspring she will have. And then the size distribution of these offsprings comes from the functions defined from the parameters that we gathered from the primary literature. And then individuals that are breeding do not grow because they are investing in reproduction and not growth. And then all individuals will continue to grow each month. However, once we hit the winter months, no individuals are growing. And then this model ran for 50 years, and we did it in time-wise steps in months, so they ran for 600 months. And this model tracks snail population size, the size of snails in the population, and the number per size range. So these graphs show the influence that some of the important parameters can have, and these are for the Chinese mystery snails. These values were pulled from the literature review. The first graph at the top shows the lifespan of the Chinese mystery snail, which, as mentioned before, is about 5 to 6 years, and it will continuously grow until about the end of its life, where it will start to plateau at about 5 years. It can also be seen that on the right side, the fecundity and size is extremely important to the larger. As the larger the female, the snail, the more maximum offspring she can produce. And then on the bottom left, it can also be seen that survival is also very important. As mentioned before, when writing the code, survival is dependent on the size. The larger the snail, the more probable the individual, the snail, will survive, especially during the winter months. And then these graphs also show important parameters for the Japanese mystery snails that were pulled from literature reviews. The trends among these graphs are the same as the Chinese mystery snail. In the growth versus size graph, however, the only difference is that they tend to live for 7 to 8 years. So their growth plateau is at about the same time as the Chinese mystery snail, but they live a year or two longer. And then fecundity and survival are both still very size dependent. So this first graph is the baseline that was ran from the parameters pulled originally from the literature review. So on the x-axis is the months ran, so the 50 years or 600 months, and then the y-axis was the population size of the snails. Each peak represents reproduction events. And then this graph shows the parameter changes conducted by the sensitivity analysis. So a sensitivity analysis was run for both snail species. The parameter values were changed in either the +5 or -5% direction, and then the model would be rerun. This helped determine which demographic parameters have the greatest impact on population size and mean size of the snails. The graph here shows that the growth parameters shown by K and length asymptote greatly influence the population size, while other survival parameters such as survival slope, winter adult survival, and winter survival juvenile slope also heavily impacted the population size. It can be seen that the winter adult survival is especially important and can dramatically affect the population size. This graph here is the baseline ran for the mean size of the Chinese mystery snails. And then this graph here shows, again, the parameter change results. It can be seen that the growth parameters and survival parameters are, again, both extremely important as they were for the population size. However, if you see that ZS and ZS slope also heavily affect the mean size, and these are the relationship between female size and number of offspring produced. And this makes sense, as we saw earlier, that the size of the female snail affects its fecundity. So just to summarize the results from the sensitivity analysis for the Chinese mystery snail, growth and survival parameters were heavily affected the population size and the mean size. The reproduction parameters dealing with female size and number of offspring produced influenced the mean size of the Chinese mystery snail, but not the population size. And then here is the baseline for the Japanese mystery snail population size. It is the same idea with the months on the X where we ran it for 50 years or 600 months. And then the snail population is on the Y with the peaks being reproduction events. And this shows the parameter changes results again. Survival and growth parameters were seen to affect the population size similarly as they did the Chinese mystery snail. However, it seems as though the growth parameters, which are K and length asymptote, again, had a much greater effect when the parameter values for both were changed in the +5 direction. This is the baseline graph for the Japanese mystery snail mean size. The means are much smaller than the Chinese mystery snails just because there were more juveniles in this population, so their sizes were smaller. And then this is the results for the parameter changes. The growth and survival parameters did affect the mean size of the snails as previously seen for the other results. However, the reproduction parameters did not have nearly as large of an effect for the Japanese mystery snail mean size, except for the reproduction threshold, which is labeled ZS threshold. So this is the size of the snail that it must reach to be able to reproduce. And it seems to have an effect on the mean size when the parameter was changed in the -5 direction. So just to compare the two results, both survival and growth parameters were heavily affected on the population size and the mean size. However, for the Japanese mystery snails, there was a greater effect in the +5 direction. Reproduction parameters were important for the Chinese mystery snails, but only reproduction threshold was important for the Japanese mystery snails, and both of those were for mean size. So moving forward, the first objective was to compare and contrast the population models, which was done through the IPM model. The second objective was to determine the most important parameter through the sensitivity analysis. The growth and survival parameters were both very important, with potentially the adult winter survival being the most important. This is essential in figuring out how to control these invasive snail species populations to be able to preserve the native species of the areas they have already invaded. To date, most control efforts focus on the hand removal of adult mystery snails, which, given the abundance of these adult mystery snails collected by the Penn State Barron faculty and myself during the summer of 2019, we removed 322 individuals in 9 days by hand. So this is probably not the most effective approach. And since these mystery snails are reclusive invasive species, there's not much known about how they have and will continue to affect the wildlife in their introduced habitat. And this was very evident throughout the literature review. I found that there was not much information on the Chinese mystery snail life history parameters, and there was even less about the Japanese mystery snail parameters. I was able to assume for any missing life history parameters needed for the model that the Japanese mystery snail parameters were the same as the Chinese mystery snail parameters found. We need more information on their life histories in order to get a better sensitivity analysis result. And so we can figure out the best way to control these invasive species and preserve the native species. Additionally, these findings can be distributed to parks profoundly affected by the mystery snails, such as those in the Great Lake regions. I just wanted to thank Penn State Barron School of Science for funding this project and allowing it to be completed. And if there's any questions, you can ask now or reach out by email to me or Dr. Lynn Beatty. Thank you.
Hi, Cassidy. Great presentation. We do have a question for you. Have you found any comparative data on growth rates and sizes for mystery snail populations that occur in southeastern Pennsylvania?
I have not. I did not find any myself personally through the literature review.
Okay, we have one more question coming through. If you can just sit tight there. Okay, were you surprised that survival probability was equal throughout the size and thus the lifespan of the individual of the CMS?
I was surprised that survival was so important, especially during the winter months. It seemed as though if they could survive winter, then they were going to survive the rest of their lives, as they would keep making it through the winter. But I was surprised to see that once they basically hit that 35 millimeters size threshold, that they were probably going to survive. So I thought that was surprising.
Great, thank you very much, Cassidy. Okay, we will move on to our next presentation. Our next presenter is Isabella Petronka, and for those of you that are judging, she is an undergraduate student at Allegheny College. Her last name is spelled Petronka. She is a student of Kelly Pierce at Allegheny College and an undergraduate. She will be presenting today on evaluating the detection of predators at river otter latrines in northwestern Pennsylvania. And we are ready to go. Isabella, I'm going to turn it over to you now.
Hi, my name is Bella Petronka. I am currently a junior at Allegheny College, and I am studying environmental science and biology as a double major. The research that was conducted in this presentation was started through an internship that I completed last semester through Allegheny, where I was required to go and check trail cameras and catalog the images to look at the presence of river otters. And through the analyzation of these images, I came up with the research question to look at predator detection at river otter latrines in northwestern Pennsylvania. River otters are semi-aquatic mammals that are found throughout North America. They are carnivores in the weasel family and typically hunt at night. They feed on fish, amphibians, crayfish, and turtles. And there's a seasonality to their diet, as in the spring and summer, they typically feed on crayfish. However, during the fall and winter, the crayfish will burrow in the mud, so they switched to slow-moving fish in the fall and winter. River otters breed in late winter and early spring and will give birth to 1 to 3 pups. The adults live alone or in small social groups, but they typically live in the small social groups. River otters in the wild will live 8 to 9 years and can grow to 3 to 4 feet in length. However, their tail makes up almost 1/3 of this total length. They use their tail to propel them through the water. They are 11 to 30 pounds, and the males are larger than the females. And river otters spend almost equal amounts of time on land and water. River otters are found in much of Canada and the United States, and they thrive in almost any aquatic habitat. They can be found in ponds, marshes, lakes, rivers, and estuaries. They have dens and latrines along the shorelines of these aquatic habitats, and they have underwater entrances to their dens so that they have easy access from the water. During the 19th and 20th centuries, river otters were hunted and trapped for their fur. Because of this unregulated hunting and trapping, they were extirpated from many portions of their range. Habitat loss and water pollution also contributed to the decrease in population of river otters, and these factors continue to be threats to river otter populations. However, reintroduction and conservation efforts helped with the recovery of the species. Specifically in Pennsylvania, the Pennsylvania River Otter Reintroduction Project occurred between the years of 1982 and 2004. During this time, 153 otters were released in these reintroduction efforts. Because of this, river otters are now expanding back into their historic ranges. The camera traps that were set up throughout this study were used to evaluate the presence of river otters in northwestern Pennsylvania, since they are now expanding back into this range. River otters frequent their latrines along shorelines of the aquatic habitats. At these latrines, they deposit feces, urine, and glandular secretions. These are areas of high activity and areas where river otters will scent mark. They typically scent mark in these areas during breeding season. Because of this, the areas have a very strong and persistent odor that occurs throughout the season. And as you can see here, there is an image of 3 river otters visiting a latrine and a typical feces deposition that is left. Camera traps were used in this study and are also used as survey tools in many ecology and conservation research studies. Camera traps can be used to detect elusive wildlife that are difficult for humans to observe. River otters and predators are among these types of wildlife, as they are typically active at night and avoid human contact and interaction. Otter predation is not common, as otters are typically hard to catch. However, predators like otters are expanding back into their historic ranges due to hunting and trapping regulations and reintroduction efforts. And they are an important issue along with the river otters. As you can see in these images, there is a fisher and a red fox. Fishers were also reintroduced into Pennsylvania and the surrounding areas like river otters were. Through looking at the images that I collected from the camera traps, I hypothesized that latrines could serve as olfactory cues that attract predators and other carnivores due to their strong smell that is generated from the otter's diet, especially from the fish in their diet. There's also evidence through literature and through research that supports this notion that there is a high detectability of predators at latrines. So my objective for this research was to determine if latrines attract predator species. My data was collected at the Erie National Wildlife Refuge. It started with surveys being completed to determine if river otters were present at some of their pools within the refuge. Whenever the river otter latrines were then located along shorelines, and trail cameras were set up at 2 ponds within the Sugar Lake Division. For data collection, 2 camera traps were set up at these pools, and they were regularly checked over a total of 57 trap days. They were checked during the months of January to March, when scent marking is repeatedly at its peak. I checked the trail cameras every other week, and they were checked during the day when otter activity was at its lowest, to limit disturbance in the time spent at each site. The camera traps were then used to determine the latency of detectability of predators after river otter activity was detected on the cameras. The images that were collected were then cataloged by species, location, and time. We completed a mathematical analysis to determine the latency of detection, and we defined the latency of detection as the sum of time between an otter detection and a predator species detection at the site. So therefore, the latency of detection tells us the elapsed time between an otter being detected at a latrine and then a predator being detected at the latrine. The predator species that were included in this study were the red fox, the coyote, and the fisher. We used these predators for the study because these were the ones that were detected and found at these particular sites. By analyzing the latency of detection, we were able to make assumptions to determine if predators were attracted to the odor of river otter latrines. We then were able to further hypothesize whether otter latrines can be used as attractants and then become target sites for camera traps when you are researching predator assemblages. For the results, when finding the average latency of detection of a predator species after otter detection, we found to be approximately 25.5 hours. However, there were some outliers to this data that were causing the number to be so high, so we redid the calculations and got rid of the data that was outside of 24 hours. So then whenever the detections were within 24 hours of each other, the average length between otter and predator detection was approximately 4.5 hours. We found that predator species typically visited otter latrines within 24 hours of otter detection. The latency in detection of predator species is likely due to activity patterns in the species. So the 14.5 hours may seem like a larger amount of time, but it is likely because the river otters were most active during around midnight. They were also detected at other times, but they were most active and most detected around midnight, and predators were most active around dusk and dawn. So as you can see in the sequence of pictures below, there is otters being detected at the latrine a little bit before midnight on March 6th. And then you can see a fisher leaving the site the next morning around dawn, and once again visiting the site again the following evening on March 7th. And here's another example where you can see river otter activity at the latrine right before midnight on January 16th, and then a coyote visiting the site the next morning right around dawn, around 6 a.m. And once again, another example of many that were detected. You can see a social group of river otters being seen at this site shortly after midnight, around 2 a.m. on the 30th. And the next evening, you can see a red fox visited the site and is now leaving. And in this image of the coyote, which you saw before, the coyote has its nose to the ground. So this provides evidence that the predators are smelling the river otter latrines. This image, along with there are many other images that showed that predators to species were detected with their nose to the ground. So this detects that, or this suggests that predators are attracted to the scent of river otter latrines. And you can see in this image where the coyote is sniffing, the snow is compacted down, and that's due to the river otter activity at the latrine. So you can tell that that is specifically where the predator is scenting. So predators are often elusive and difficult to detect. So since they are attracted to river otter latrines, these latrines could be targeted as attractants for predator studies. Typically, when predator studies are done with camera traps, bait is used to attract the target species to these camera traps. And this is not very natural, and it can be disruptive to the ecosystem. However, if you use otter latrines to attract predator species, this is an extremely natural method, and it can be an effective tool in protected areas where baiting is not allowed, such as national parks. So the fact that river otter latrines can be used as attractants for predator species is significant when studying predator assemblages. So I would like to thank the Pennsylvania Game Commission for providing the trail cameras used in this study in the Erie National Wildlife Refuge for their assistance in this project. And I would also like to thank my professor, Kelly Pierce, for her knowledge on river otters and her support throughout my internship and throughout the research that has been conducted because of it. So I can take any questions now, if you have any.
Hi, Bella, this is Jeanette. So we're gonna just pause for a second here and see if any questions come through. There is a delay between the two platforms that we're using.
Sounds good. Thank you.
Amber, do you have plans?
Okay, we do have a question for you. For the future of this study, do you plan to conduct it in other locations? And is this something that you would conduct year-round or just during certain seasons?
So personally, I will not be conducting this in other areas, but it's definitely something that can be done. And it can be done in other seasons, but the scent marking is highest during the winter months due to breeding season. So that is when you'll see the most activity at the river otter latrines and the most scent marking being done, which would attract the predators.
We have one more. Do river otters in Pennsylvania interact at all with beavers?
That's a good question. I'm not entirely sure. I know at the site that I had one of the trail cameras, there were beavers present, and they were right in the same area as the river otters, but I am not sure exactly how they interact.
Great, thank you so much, Bella. We appreciate you participating today, and thank you again for your presentation.
Thank you very much.
Okay, let's do like a 5-minute. So we're just a little bit ahead of schedule. What we're going to do is take a maybe just like a 3-minute break, and then we will come back and we have a series of speed talks that will start right at 11:30. Thank you. Okay, so we are back. Our next presentation is by Bailey Crable. And for those of you judging, this is a speed talk, so you do not have to worry about judging this talk. And Bailey is going to talk to us about the use of Erie Marsh dominant areas in Erie County, Pennsylvania by waterfowl during breeding and migrating seasons. So Bailey, I'm going to hand it off to you now.
All right, sorry about that. So my name is Bailey, and I have been doing research on the use of Erie marsh dominated areas in the Erie County of Pennsylvania and how waterfowl are using them during breeding and migrating seasons. So just a little bit of background information. The habitat that I am observing are marshes, which are a type of wetland habitat that are mainly dominated by herbaceous plants as opposed to woody plants. I'm also looking specifically at waterfowl and how they use those areas. And I'm mainly doing a comparison between the use of different species during breeding season as well as during migrating season. So breeding season can vary amongst different species, but for the most part, it happens during the warmer months. So breeding season here is mainly around mid-May to mid-June and July, which is the end of the spring migration. So the winter migration can also vary amongst different species, but also takes place here around November through December, which is kind of where I'm at right now in my research. So my main reason for doing this research is I took an ornithology course in my junior year of college, and I became really interested in birds, and I have taken up birding as a hobby, and the eBird hotspots that I visit the most are marsh areas. I'm also really interested in how climate change could have a potential effect on waterfowl migration because of how the environmental cues birds pick up on when it comes time for migration. Climate and temperature are part of that, so that could definitely have an effect on when they migrate. So my research is taking place in 3 different locations as of right now, 3 marsh areas around Erie County. I'm going to Lake Pleasant, I'm also going to the state game lands, and I'm going to the West Branch French Creek Conservation Area. So here is my first location. It is Lake Pleasant. It comes right off of Lake Pleasant Road, and I believe the species count for this area is around 118 species. It includes songbirds and waterfowl, but like I said, I'm mainly interested in the waterfowl. This is my second location, the game lands. I believe the species count here is around 164. And I'm mainly going to a area of the game lands off of Barton Road. There's a little spillway, and that's not where I've been going for my research. Then I've been going to the West Branch of the French Creek Conservation Area, which comes off of Route 8. And again, they have a little marsh area, so I walk the trail and I check out the marsh area to see what waterfowl are using it during that time. So my methodology is I have been doing at least a 10 to 15 minute birding session every time I go to each of these sites. I have a set of binoculars as well as a spotting scope that I set up for seeing waterfowl at farther locations. I use a Merlin bird ID app to help me identify the birds based off of their look and also their sounds that they make. And I have been recording each species that I see in a checklist and then updating that to the Cornell eBird database for collection. So that's how I keep track of all the birds that I've spotted at what time and at what site. So here is some of the data that I've collected so far. I have right here is the hotspot observation, which is everybody's observations, not just my own, that include for all months of all different species that have been seen at that location. So as you can see, I've been seeing a lot of the same species as everybody else has in that area. I have not quite started my November and December birding checklist, but using this data, this is the kind of birds that I would expect to see, such as mallards and Canada geese, which have been seen throughout the breeding season so far. And then once again, here is my data for the state game lands, where Canada goose have been seen throughout the entire year, which is then consistent pretty much with what I've been seeing when I've been going. And I'm expecting to see in November, maybe a few tundra swans, some more mallards, of course. And then going into West Branch French Creek Conservation Area, this is a newer hotspot, so the data is newer. But I'm expecting to see pretty much the same thing going into November and December, which is more geese and some mallards. Going back, I'm expecting a lot of differences during the migration that can happen during March. So I expect to see an increase in diversity probably around March when there's another migration that takes place. So based off of everything that I've started so far, I can see that there is starting to get a small increase in diversity, given that now in October, I started seeing a few new species, not a lot, but just a few, such as the ruddy duck, that the diversity of the marshes is becoming greater, and also the abundance of birds is also becoming greater. So there's more Canada geese that I'm seeing now than as opposed to in July. So right now it's starting to look like during the migrating season, the species diversity is going to be greater then. So for the future, I am planning on continuing to monitor all 3 of the sites that I've been monitoring, and I'm going to monitor them throughout the migrating period to see whether or not there are more species that will be observed. I'm hoping to see maybe a couple of species that I have on the slide here, maybe a redhead or a bufflehead or something like that, but only time will tell. So the only thing I can really do now is continue to keep going. And that's all I have right now.
Hi, Bailey, this is Jeanette. I have one question for you. Have you observed any large predatory birds at your study sites, and do they interact with these waterfowl?
So I have definitely seen some predatory birds at these sites. It's not unusual to see a bald eagle at Lake Pleasant or at the game lands. I have yet to see any real predatory birds at the French Creek Conservation Area, but it wouldn't surprise me if they were there. Have I seen them interact with these birds? I personally have not seen them interact, but it wouldn't surprise me because they are always there when I see them, kind of hanging out around the marsh area, watching over everything. So it wouldn't surprise me if they were watching over the waterfowl, because it's also not a surprise that some of these birds of prey will eat some of the waterfowl. So I've definitely seen them. There's definitely been hawks at Lake Pleasant. They're not so much waterfowl eaters though. So maybe the eagle is probably the predator of interest in this case.
Bailey, I have another question for you. First off, very cool. And this viewer has never used this app to ID birds. Are you able to quantify species using this app, or is it more of a presence absence type monitoring?
So this app is mainly specifically for IDing, and then you can use the eBird website to quantify all your information. But what's great about this app is that you can download different packs, so that way they know which birds will be in your area at the time. So I use this app and it changes daily to accommodate for migrating seasons. So that way I know which bird I'm more likely to see. It also tells me which birds are rare that are in my area right now. And it categorizes everything. So it has different categories of waterfowl. It has birds of prey, seagulls, everything that you might see listed. And you can look up different pictures. And it also has different sounds that you can use for comparison.
thanks Bailey. That was a great presentation. And the one nice thing about Speed Talks is it really creates awareness to our viewers of the different projects that are in progress or being conducted into the future. So thank you very much. Okay, unmute myself and talk to the camera, or?
You can go ahead, just keep going.
Okay.
You're live now.
Okay, our next presenter is Isabella Petita, and she is a student at Allegheny College. This will be another speed talk, so our judges do not have to worry about filling out the forms. And she'll be talking to us about wild bee species diversity supported in a conventional soybean field located in Crawford County, Pennsylvania. Isabella? Isabella, you can go ahead with your presentation now.
Okay, thank you. Hi, everyone. I'm Isabella Petita, and I'm a senior at Allegheny College majoring in environmental science. And today I'm going to be telling you about my senior comprehensive project titled Wild Bee Species Diversity, supported in a conventional soybean field located in Crawford County, Pennsylvania. And this project is under the advice of Dr. Beth Choate in the Environmental Science and Sustainability Department. And my project was actually started in 2017 by a former Allegheny College student, and it was never completed. So my project is to take all the bees that were identified from this soybean field, or collected, and to identify them to figure out the diversity in this field. My research is based on wild bees, and I would like you to take a moment to think of how many native bees that you can think of that are native to Pennsylvania. If you thought of a honeybee, these are actually not considered a native bee species. Species such as Apis mellifera, the European honeybee, and Osmia coniferans are introduced species, meaning that they were introduced into our region and they're not native to Pennsylvania. If you thought of maybe a sweat bee or a bumblebee, you're thinking of a native species. And there are 437 bee species that are in Pennsylvania, and 23 of these are not native to North America. Wild bees are typically solitary, which means that they nest alone and they're less aggressive. And they can nest in the ground, like the soil, or in woody material, which is why you might start to see bee homes popping up in stores that have hollow plant material and kind of like a birdhouse shape. And there are 6 different families of bees in Pennsylvania, and there are 7 in the world, but the 7th is only found in Australia. And you can see that in the figure. This is out of a study that was published this year out of Penn State. And it's a checklist of all the bees in Pennsylvania. So there are 6 families, and the most common bee family is the Apidae family. And this has common bumblebees, squash bees, mining bees, and the honeybee is also in this family. And the second most common is the Halictidae family. And this includes bees like sweat bees, and they're a shiny, bright metallic green. So if you ever see a shiny green bee near you in Pennsylvania, it's a Halictidae bee. And these are bees that I'm really expecting to see a lot of in my research. So it's a family that I'm focusing on. And the term wild bees encompasses wild bees that are native to our region and that are unmanaged. In 2020, 83 million acres of soybeans were planted in the United States. So this crop is a huge part of American agriculture. And in 2019, 252,571,000 was the production value in Pennsylvania alone for soybeans. And soybeans are not reliant on pollinators to fruit. They're actually wind-pollinated crops. However, research has found that soybean pollen was found on pollinators and that they can have a modest dependence on pollinators.
And the same study also found that there was a diverse community of wild bees in the soybean field, which is something that one might not expect because there's only one crop or one plant in this field. So this was kind of motivation for my comprehensive project to determine if we could find a diverse community or populations of these bees. And this project is relevant because of the urgent demand to understand wild bee populations and their behavior in human-dominated landscapes that have disrupted their natural habitat. More than half of the bee species populations that are native to North America are currently declining due to anthropogenic effects on their habitat. And these effects can be pesticide use, habitat destruction, or urbanization. So understanding more about diversity in human-managed landscapes can allow for a better understanding of the human effects on bee populations in their response to agricultural intensification. And my project aims to determine if diverse species are present and abundant in areas of agricultural intensification. The question that I'm trying to answer in my project is, what is the diversity and abundance of wild bees in a conventional no-till soybean field? And in this study, the term conventional is referring to the way the land is managed. So it's referring to that there is one crop over many acres of land, in this case soybeans, and that pesticides and herbicides are used in the growing process. And I'm hypothesizing that the greatest mean species richness, which is the number of species, and diversity, which is the number of species accounted for with abundance, will be found at exterior field sites, and few generalist species will be found in all the samples. So this picture on the left is an aerial photo of the farm where the bees were collected. In areas where you can almost see like a road, that's where I'm expecting to find more diversity because there might be more floral resources in these areas, rather than the middle of the field, which just has soybeans. And the term generalist is referring to the way that the bee forages. So a generalist bee would be a bee that can forage on different types of plants, as opposed to a specialist bee, like a squash bee that pollinates squash crops, that just focuses on one plant that is co-evolved with. And for my methods, I'll be identifying these bees that were collected in the summer of 2017 and using taxonomic keys. I have a book called Bees in Your Backyard that I use, and an online key that's called Discover Life that's widely used, and it's really nice. And I'll be measuring abundance, richness, and diversity of about 1,000 bees that were collected. And this picture on the right is a picture of my remote lab that I've made at home. I have my microscope, my keys, and in the bottom right, you can see a few of the bees that I'm working on, but I have a lot more than that. And the methods for my project really start with taking the bees that were collected, some that were frozen, and defrosting them and pinning them. And the term pinning is just taking a bee and pinning it on a pin, and that preserves the bee so that I can look at it under the microscope and keep it in a collecting box. And once I was done with that, I took all the bees that were pinned and organized and grouped them based on family, genus, and species. But they weren't grouped as species immediately because that's what I'm working on right now. I'm working on identification, so I'm taking all the bee samples and identifying them to genus and species. And I was able to identify them all to genus, and I'm currently working on species. And as I identify them, I record them in a Google Sheet with other information like where was this bee collected, how was it collected, and on what date. And once I finish all my identification, I can analyze everything that was collected to determine the richness and diversity of all these bees. When I'm identifying the bees, I mainly use the online key because it's really nice to use for more difficult species, a dissecting microscope, and a lot of patience. And I look at different characteristics of the bees, like their size, color, wing shape, the location of their scopa, which are pollen collecting hairs, their antennae, and many more things. And their antennae are really helpful because I can determine if it's male or female. And this is important because males and females can look very different in bees, so much so that you might think it's a different species when it's really just a male or a female. So this bee on the right, this is a Lasioglossum, and this is a common bee in Pennsylvania and common in my study. So you can see on the wing, this vein right here is shaped like a J, And immediately that tells me that this is in the Halictidae family. And this color of the bee is like a dull green. So it's probably a Lasioglossum. Based on the size and the shape of it, I can determine that. And I'm anticipating to see a high number of Flugella species in the Halictidae family, just like the Lasioglossum that I just showed you, and to find more diversity at the field sites at the exterior of the soybean fields. But I'm also hoping to have a documentation of bee species as a resource for population studies that will happen in the future. So once everything's identified, researchers can look back and see the species that were present in 2017 in Crawford County. And also, a high abundance of these ground nesting bees may support that no-till agriculture is more beneficial for these pollinators. My preliminary results show that abundance was highest over the 4-week blooming period of the soybeans, near full bloom, and at the end of the beginning bloom in late July. And I was able to find that there's 15 different genera in my study, and these are lists of all the genera that I found. And Lasioglossum and Halictus were definitely the most abundant, and it seems like there is a decent diversity being found in this field, which is what I wasn't expecting to find. So that's all I have. Thank you for listening.
Great presentation, Isabella. We do have a couple questions for you. Do any of the 437 species of native bees are used by humans for honey?
Yeah, so the honeybee is primarily the species that will be used for honey production.
Okay, and one additional question. Love your remote lab. Which Pennsylvania bee species do you hypothesize is most negatively impacted by human activity?
That's a difficult question because they're really all wildly different in their behaviors and their pollinating behaviors too. But I would say just generally ground nesting bees, which would be the Halictidae family like I shared, but also Megachilidae and different families, a lot of them are ground nesting. So these are really going to be the most that are detrimentally affected by human activity because of urbanization and like creating impervious surfaces and disrupting their native soil habitat. It makes it harder for them to find suitable habitat, which is why maybe bee homes are starting to be sold in stores because their habitat isn't suitable. So I'd say just generally ground nesting bees.
Okay, one more question for you. Have you considered comparing your bee species richness in the soybean field to a control site presenting non-ag, like an open field, or more mixed agricultural uses?
Yeah, I think that would be great for my study. There really isn't a lot of bee research in Crawford County. There's really only 1 or 2 other studies documenting bee species that are there. So that's where it becomes difficult and why this research is so important to document them. So if I did have a control that I could compare it to, I would, but as it does get kind of hard with the lack of research, but there was a study that was published in, I think, 2018 that does record species that were identified that I can compare to, and just generally other research that has been done in agricultural areas.
Great, thank you so much, Isabella. It was a great presentation. Our next presenter is Julie Webster, and she is also giving a speed talk today. And she'll be talking to us about the predictors of success in MCGA community gardens. Julie, I'm going to hand it over to you.
Okay, my name is Julie Webster, and I am a second-year student at Lake Erie College of Osteopathic Medicine. And this was a project that I was working on when I was living in Indiana on community gardens in that city. So community gardens are generally established in direct response to communities diagnosed as food deserts. Food deserts are considered any area that doesn't have access to fresh produce like tomatoes and vegetables. And these often occur in areas that are low income. And together, lack of access with low income usually results in nutrition-related health problems like diabetes and poor dental health. Some businesses have noticed these food deserts and that they are lacking in food, so they've started to establish some stores there like Dollar General and Family Dollar. But that just furthers the problem because a lot of times that's pre-packaged food and they don't offer a lot of fresh produce. In doing a literature review of community gardens, we found that there were a number of articles that pinpointed some main factors that play into a garden's success or failure. And I have some of them listed here. The garden needs to have policies and support systems in place, ready to tackle some problems when they present. All good gardens also have a form of exclusion, actually, where you can tell exactly who can garden there, who can't garden. A lot of times it deals with having to live in the neighborhood to actually participate in the community garden. And then you need to have established systems for biosecurity. Theft is actually a big problem in community gardens. It seems pretty innocent when you might have done it yourself, actually, if you're walking through a neighborhood and you see a bush or something that has a tomato on it or some other fruit like an apple, and then you just take the apple and eat it. And some gardens will consider that as theft because you didn't participate in any of the growing of that, and that's somebody else's produce. You also need to be sensitive about what plants are going into the gardens. This one article talked about a southern garden that a resident wanted to grow tobacco, which that was a point of contention for other gardeners. Another plant that is kind of sensitive is marijuana currently. And then these last two articles talked about 9 barriers to urban gardens. And they recommend that community gardens that are trying to plot their first garden look at these barriers and establish some sort of system to tackling those, because those barriers are going to present at some point for that community garden. So Marion, Indiana was the town that we were focusing on, the specific food desert. It has a childhood food insecurity rate for the county of 23.5%. And that means that 3,270 children don't have stable food sources, and 84% of those children are below the poverty level. So like I said earlier about the food desert, when you've got lack of access paired with poverty level, a lot of times that results in nutrition-related health problems. And so that's why we were focusing on this one. The Marion Community Gardens Association was established in 2010 to counteract that problem. So that's what the association we were specifically looking at. And our goal was to see what needed to be done to make these gardens more successful. The Bare Roots was already there. They had an association and people willing to work to make these gardens happen. But some gardens were succeeding and some were failing. So our project was trying to figure out what the major trend was, what the major difference was between the successful gardens and the failed gardens. This was a collaborative project between the Marion Community Gardens Association, the Foundation of Grant County, Marion Public Library, the John Wesley Honors College, and the IWU Division of Design. So some general benefits associated with community gardens. The most obvious one is that they combat issues associated with food deserts. They provide fresh produce and vegetables to communities that don't have access to that. And it's a safe space for those people too. They know what went into the vegetable or the fruit, and they know how it was harvested because they did it themselves. Gardening is also exercise and a social act. A lot of gardens will have communal plots. And the gardeners who are participating in that garden are expected to participate in those plots. And that's where larger vegetables can be grown, like zucchini or eggplant, or you can grow pumpkins and watermelon there. And a lot of gardens are established in association with libraries or churches, and those businesses and churches and organizations often will offer classes on cooking and agriculture, so gardening becomes a hobby as well. And another factor that has been seen with community gardens is that it actually lowers the crime rate in those communities. Some issues associated with the general community garden is that it requires a lot of effort and planning to happen. There has to be someone in charge who knows who to contact if you have problems with theft or with getting resources for maybe gardeners who don't have much money. It's difficult to promote longevity in areas with variable populations. A big problem we saw here actually in the Marion, Indiana, was rental neighborhoods, because there was a garden that was planted there and the population wasn't that stable year long, so not many people felt that they could participate in a community garden. It's also difficult to persuade people to participate because we live in a culture of immediate gratification. And gardening is a long-term process. You plant the seed and you don't see much happen with it for weeks. And so it's a long-term goal that you have to convince people that it is worthwhile to wait. And as I mentioned earlier, theft is a problem. And modern farmers markets are morphing more into craft fairs than places that you would go for produce. This is a problem for community gardens, not just because it's another source for people to get food and vegetables, but people who participate in community gardens used to be able to go to these farmers markets and sell their produce, so then they would have an extra source of income. But you don't go to farmers markets much anymore for cucumbers or apples. A lot of times it's for hand wood carvings or sweaters or other baked goods, really. So this plot shows the general trend of the gardens in Marion. There was one established in 2010 in partnership with the Marion Public Library. And then the next year, 7 more were plotted. And that stayed stable for a little bit, but by 2019, there were only 2 gardens that remained. So general rules for the MCGA gardens is each community member will get a 5x10 plot for free, and then extra plots after that point are $20. And that gives a sense of ownership to those people who want to invest more. They put more into the project or into the garden, and also applies the exclusion principle where if you want more land, you have to pay a little more for it. And they're going to be a little more protective about what goes on in the garden after that point. Members are also expected to work the community plots, and it's managed overall by Megan McCracken, and she appoints resident gardeners who are in charge of finding funding by applying for grants and supplying water and managing pests for those residents who are gardening. Here's some locations for the gardens. These were all the ones that were ever planted. And you can see by looking at this that they're spaced enough apart that the gardens aren't really competing for participants. They're in different neighborhoods, but yet some still failed and some succeeded. So in meeting with resident gardeners and with Megan McCracken and Kayla Johnson, who was another person involved with MCGA. These are some trends we found among the successful gardens. There's always an established system of organization and planning where someone is in charge and it's easy to contact that person. The communal plots were important parts in the successful gardens, as well as plentiful resources and grants. So the gardens that had more funding did better. And promotions and advertising were another important factor, as well as decorative greens. The picture I have here with the bicycles on the side of the building, that was one of the most successful gardens here. It was run or hosted by the Cyclery, and they did a lot of advertising through the Cyclery on their sign that you can see there and inside the building. They also included a lot of decorative greens like flowers and bushes that weren't necessarily produce, but it made the garden feel special, and it is a part that was set apart and the gardeners could enjoy. The other garden I have pictured here on the left was an unsuccessful garden. This picture was taken a little bit before it kind of fell apart, but you can really tell a difference between this Cyclery garden and that garden just by looking at it. I mean, if I told you which one would you want to participate in, I know what my answer would be. The main factor we found that was really successful for those successful gardens, and it was really lacking in the failed ones, was advertising, actually. The gardens were already established. They had systems in place to regulate the gardens and control how they got resources, but sometimes people just didn't know they were there. an example where advertising really would have been helpful was in that rental neighborhood. Some residents there didn't know that gardens existed, but other ones did, but they didn't think they could participate because they couldn't commit to a year of gardening. They were not knowledgeable about the communal plots. They didn't know that those were a thing. And that garden was set up so that the rental residents could participate in only the communal plots and not the individual if they didn't want to plot themselves. So if they would have known about that, they might have participated a little more. It's also important to target neighborhoods in the vicinity of the gardens and local churches and community centers who are focused on community outreach. These are some contacts that we came across who were interested in promoting the success of gardens through advertising. The two people in the middle there are actually professors of graphic design, and they said they would be interested in helping with the advertising procedures. So future direction is to partner with the Marion Public Library, who helped establish that first plot, to set up a website for the community gardens and to provide pamphlets and some weekly events or some sort of event that's a little fun for the residents to participate in. And that would be all done under the guidance of those previously listed contacts. And I just wanted to bring to your attention a garden that's here in Erie, PA, actually. This garden is I think it was the Mercy House Garden, and it's run by, I believe it was a church, but they're in a Nepali neighborhood that has a lot of Nepali families, and they have 28 beds, and 11 Nepali families participate in those beds and grow foods from their native lands. And so it's really important that you bring to attention the gardens that already exist here, because many people are interested in participating, but they just don't know they're around. Like, I wasn't aware of this garden here in Erie. And so that is all I have today. Here's my references. And if you have any questions, I'll take those too.
Okay, Julie, thank you so much. We do have a question for you.
Okay.
Do you think community gardens can reduce the use of fossil fuels for food transportation or for food transport, or is food production by community gardens too minor to make a difference in this problem?
That's interesting because in bigger cities, it is a little harder to establish gardens there. But I believe it was in New York. I can't remember specifically, but there was a community that established a garden in their neighborhood, and that's all they used. They only used that neighborhood for all of their food, or that garden for all of their food. So that was a whole community that relied only on that garden. They were also able to have chickens there and some other things that provided meat for them. So I think it could cut down on transportation costs and fossil fuels, but again, it's the thing of getting people to participate is really hard to do. But if enough people were invested in it, I think that could definitely be something that would happen.
Okay, we have one more question for you. Were any of these community gardens associated with local food pantries?
Yes, there were 2 that were run by churches, and those churches had food pantries. So a lot of times it was the communal plots where they would take foods that were grown there and then add them to the food pantry. And so that was another way to get fresh produce out to people who didn't have access to it, which I think is a really good idea for getting other businesses involved. They can play a big part in that as well.
And one last question for you. You may have mentioned this, but are the community gardens strategically placed to reduce food deserts in these neighborhoods?
Yes, they were. So Marion, Indiana as a whole they're pretty much the whole area is considered a food desert. So you could really place a garden anywhere and it would be helpful. But they tried to space them out so the most people were affected.
Okay, great, Julie. Thank you so much for your presentation.
Thank you.
It's no secret, education is the key to success. Serving more than 2,200 families, WQLN brings learning to life by combining PBS Kids programs with reading and hands-on activities.
People say it takes a village to help raise students. It also takes a village to educate them.
You're part of a community, and we're all here together, and we all care about the children and the future of the community.
To support the program, visit WQLN.org.
ETD Photography is an Erie-based media and creative services company by Eric Dye, creating media to expand your reach and further your goals. With a diverse project history, ETD Photography brings unique and comprehensive creative media to Erie individuals, businesses, and organizations. Services include photography, videography, web design, logos, podcast production, YouTube channel production, live streaming, and more. Check out our website at etdphotography.com.
Hello, my name is Dr. Jeanette Schnars, and I'm the Executive Director of the Regional Science Consortium. And I'm here at Presque Isle State Park today exploring the priority wetlands that we've been working on with our collaborators to restore over the last decade. Now, wetlands are a critical ecosystem that help protect Lake Erie and other bodies of water that they're connected to from runoff, and that would include sediment in the runoff, nutrients, and pollutants. And then wetlands also serve as critical habitats for all different kinds of species, including plants and insects, fish, birds, and other mammals that live here. Today we're going to investigate the relationship between humans and wetlands and how this has changed over time. So wetlands, once thought of as swamps or just pools of stagnant water that were unsightly, were often destroyed as a result of construction and development. However, today their importance is realized, and a healthy wetland exhibits high plant diversity and animal diversity. However, due to invasive species and some other human interactions, some wetlands are not considered healthy, and that's where we come in as researchers to help restore these areas.
My name is Holly Best. I'm one of the assistant park managers here at Presque Isle State Park. I work with the resource management program. A large part of that program, we try to manage invasive species that are growing on the park. Some of the main targets are Phragmites and narrowleaf cattail. Phragmites are specifically important to manage because not only are they very prolific when they come into an area and take it over, they spread through rhizome, they also spread through seeds. They also are allelopathic, so they actually put poison or toxin down into the soil and prevent anything else from growing. So that makes a monoculture of that plant, which for species diversity is very detrimental. At this park, we're very fortunate that we are able to use herbicide to help control these species. We have done everything from have interns with backpack sprayers, doing a very focused attempt, all the way up to helicopters actually doing kind of broadcast spraying over some of those very large monoculture areas. We're fortunate that we're able to use that as a control technique because if you don't have that ability, it's very hard to control these species and try to return these areas back to their native environments.
My name is Jen Salem. I'm the horticulture specialist for the Regional Science Consortium, and I'm also the founder of Go Native Erie. Go Native Erie is a program that operates under the Regional Science Consortium, and it helps educate the public about the benefits of native plant species and using natives in their gardens. Native plants are actually, they're defined in two ways. They are plants that were discovered here growing in North America at the time of European settlement. But a better definition of that is that native plants are a plant that has been in an area for a long time and it has evolved with something else to form an important relationship. We are in the Regional Science Consortium greenhouse at the Tom Ridge Environmental Center. So we use this for propagation. So we get things growing and started here. So this is the beginning of the process. So every year I'm looking at what seeds can I grow, what did well. Every year we're trying to increase our diversity and the amount of plants that we grow, but also pay attention to things that are going to do well. It's not just about quantity, it's also about quality. So now we're down at the Regional Science Consortium's cold frame greenhouse on Presque Isle State Park. And this is used as a distribution center and holding area for all the plants that are produced in the Regional Science Consortium propagation greenhouse. So when plants grow up to be a certain size and they're ready to be planted in a wetland, they are brought down here and held at this site and cared for. Then when we have groups of volunteers and employees that can come and help, we decide what plants are going into each of the restoration locations, and we'll just load these all up on a truck and have a planting day.
All right, so I hope you enjoyed the tour of our wetlands and all the work that we're doing with our collaborators to help restore these areas. So if you'd like to learn more about this project or other projects, please visit the Regional Science Consortium's website at www.regsciconsort.com.
It's no secret, education is the key to success. It's no secret, education is the key to success. Serving more than 2,200 families, WQLN brings learning to life by combining PBS Kids programs with reading and hands-on activities.
PBS Learning Triangle, I feel like it's a simple step for parents to grasp. So it's not something over their head or too complex.
Watching my students for the 4 weeks develop a little bit more of their personalities, have the opportunity to read and have the opportunity to be excited.
People say it takes a village to help raise students. It also takes a village to educate them. So WQLN coming in to partner with our kids helps those kids get those strategies and the parents the strategies that they can help their kids at home.
You're part of a community and we're all here together and we all care about the children and the future of the community.
To become an EITC partner and support this program, visit WQLN.org.
ETD Photography is an Erie-based media and creative services company by Eric Dye, creating media to expand your reach and further your goals. With a diverse project history, ETD Photography brings unique and comprehensive creative media to Erie individuals, businesses, and organizations. Services include photography, videography, web design, logos, podcast production, YouTube channel production, live streaming, and more. Check out our website at etdphotography.com.
Hi, I'm Dr. Jeanette Schnars. I'm the Executive Director of the Regional Science Consortium, and I'm standing here on the shores of Lake Erie, which is known as one of the best sport fisheries in all of the Great Lakes. And two of the most popular species include yellow perch and walleye. The fishery historically has always been great here, but it has had some pressures in the past. including over-harvesting of certain species and other things like invasive species. These invasives have really paid a toll on some of our best sport fisheries.
Hi, my name is Greg Andreso, and I'm a biologist from Gannon University here in Erie. And for about the last 20 years, my students and I have focused on the fish population in Presque Isle Bay behind me. A lot of our work has focused on the round goby. The round goby is a non-native species from the Black Sea, Caspian Sea part of Eurasia, and it was first introduced into Lake Erie in the early 90s by ballast water in freighters. So these freighters brought these fish across the Atlantic and offloaded them unintentionally into the Great Lakes. So a lot of the work that we've done is focused on round gobies and their interactions with two species of mussels, which are actually non-native as well, what we commonly call zebra mussels. And there's actually another species that's closely related called quagga mussels. And our work has really looked at predation by round gobies on these Dreissenid mussels, the zebra mussels and quagga mussels. Round gobies eat things like smallmouth bass eggs, so they've had an influence on the ecosystem. They're here to stay. I don't think we're ever going to get rid of them, and part of my job and the job of my students has been to try to better understand their roles in that ecosystem.
Hi, I'm Dr. Casey Bracha-Wilson from Allegheny College. I am studying the invasion of round gobies from the Lake Erie watershed into the French Creek watershed, which is a stream that ultimately flows to the Mississippi River. So this invasion is a really important one that we've been studying and monitoring since they've first been introduced, probably through some type of fishing activity, whether it was through a boat or through bait bucket transfer or something like that. We have a lot of projects going on, including what the round gobies are eating, how they're impacting native fish through behavior and aggressive interactions, also how they can change communities through eating macroinvertebrates and other fishes and their eggs and those types of things. French Creek is an extremely important stream in Pennsylvania. It houses more diversity than any other stream in the state, including many threatened, endangered, or species of concern. So round gobies have recently been known to eat native mussels in the juvenile phases when they're very small. And so a lot of the mussels in French Creek are threatened or endangered species. And so the impacts to mussels from round gobies, we don't know exactly how catastrophic that will be, but it could be really bad for the French Creek watershed.
The Pennsylvania Fish and Boat Commission regulates fishing here in Erie, Pennsylvania, and on the waters of Pennsylvania for all of Lake Erie. Lake Erie has been subject to a lot of invasive species throughout the years, all of which have unknown effects when they first get here, and it's only through their actual arrival that we're able to manage them. 2018, the Pennsylvania Fish and Boat Commission joined with our cooperators in Ohio, Ontario, and New York and started tagging walleye here off the shores of Pennsylvania, right here off of Walnut Creek Marina, looking at spawning site fidelity as well as fish movement throughout the summer months and even into the winter. To date, 41 walleye have been tagged here at Walnut Creek and released back into Lake Erie, and we've watched those fish travel for almost 2 years now. We have 21,000 detections of those fish in the last 2 years. And with that, we were able to see if these fish do exhibit spawning site fidelity and return to Pennsylvania waters to spawn. The Pennsylvania Fish and Boat Commission operates the Lake Erie Boat Angler Survey annually from May 1st to October 31st. We run a bus route style creel survey in which we sample anglers and boaters from Walnut Creek, Lampe Marina, East Avenue, Boat Launch, and Northeast Marina. In this process, we count all the boats that launch, and we count all the boats that land at those marinas during the times that we're there. And while we're there, we try to interview all the anglers that land. We try to find out some economical information from them, where they have traveled from by getting their zip code, how many folks were here, how many folks fished, how many rods they used, how long they fished, what they were targeting, and what they caught. And from this information, we can start to come up with a figure of how many fish are successful, or how many fishermen are successful, and how many fish are harvested from Lake Erie annually by our recreational anglers. The Pennsylvania Fish and Boat Commission is very interested in sustainability. So with all this information and all this work we do throughout the summer.
Hi everybody and welcome back. My name is Sean Dalton. I am the Lab and Field Coordinator at the Regional Science Consortium. Without further ado, we're just going to get right into our next presentation. Our next presenter is Sudi Wang, who's an undergraduate student from SUNY Fredonia for you judges. The last name is Wang, W-A-N-G. Her presentation is, how does the non-native invasive periwinkle outcompete native species? So without further ado, Sudi, I'll turn it over to you.
Okay, hey, how was everybody? So I'm presenting how does a non-native invasive periwinkle become minor or compete native species. So what is invasive species and why become invasive? Well, invasive species, they are invasive because human activities. When we travel, we tend to carrying the seeds from one place to another, like they were stuck into your shoes or your clothes. And then when they germinating in the root, in the soil, and they were tend to take over the native plants, root space, and lose native biodiversity. It gives little or none food value for wildlife. Compete resource with native species, such as the invasive species like Oriental bittersweet or garlic mustard. They grew very fast, and then when they shade out, they can when they grow and they shade out the native species by compete with the sunlight and moisture, soil, water moisture. So the higher reproduce rate give competitive advantage over native plants. Those are some more examples for invasive species that common in New York State. Those are two species insects are the hemlock, woody adderjet, and amber ash border. They can grow on the ash tree or hemlock. They will take over the canopy cover. There's more than 16 billion ash tree actually killed by that insect. So for my research, I study for the periwinkle Vinca minor. So periwinkle is native to the Southern Europe and Southern Western Asia. It's very easy to identify by the long-leaved purple flower bloomed in the spring and summer. They have lower seeds production. They're produced asexually. They distribute in a temporary area used as herb medicine. The leaf contain the chemical called vincamine. Normally it can treat leukemia and high blood pressure. So the purpose for this research is to understand the impact of periwinkle on our native forest, understory species, and the mechanism it used to outcompete those native species. So to begin with, I had to understand the allopathy. The allopathy is plants' interactions. One organism produces biochemicals that influence the germinations, growth, survival, and reproduction for the species that shared in the same communities. So my prediction is periwinkle actually can produce some chemicals that give no benefit or suppress the native plant growth in the same communities. And also for the mycorrhizal. The mycorrhizal mutualism benefit both the plant and the fungus. The fungus receive sugar from photosynthesis and the plant receive phosphorus and other nutrients. Mycorrhizal fungi can be killed by invasive species which emit fungicide. Percent mycorrhizal mycorrhizal inoculum potential is a capacity for the soil to colonize a plant roots with mycorrhizae. As the picture showed here, with plant with mycorrhizae actually have more stuff going on. And we actually locate the mycorrhizae plot in the college lodge. We collect 4 meter square plot in dense periwinkle and adjacent patches, non-periwinkle plot. So in the field, we examine richness, diversity, cover, canopy cover, slope, aspect. For the lab and the greenhouse, we determine the seed bank, soil moisture, mycorrhizae, and allopathy. Here are some field pictures we took over the summer and the spring in the college lodge. So for the seed bank, in the greenhouse, we used 1 liter soil to collect from periwinkle and non-periwinkle plots at each side. We grew for 4 months. And here's a picture that seed bank in July and August. There are some interesting species we identified in the seed bank, such as pine and violet. So in the lab, we did the mycorrhizal test by placing the collected soil in 2 pots of each plot. The corn seeds were planted in each pot. Water the pot every other day in the greenhouse, and after 30 days, harvest the corn and the corn roots. We wash the corn roots use the stem and the stem with the tri-paint blue. So I cannot pronounce that. The chitin stem. So place the roots in a titrate pan plate over a 1 centimeter grade of line, and 15 places where roots roasted in a line that are scored for the percent of mycorrhizae fungi. So this is like the grade that we use. Under the microscope, we can see some hyphae, which that's the mycorrhizae that use to communicate with another plant. Those are pictures I took on the microscope, and it's very easy to identify the hyphae and plant roots, stems, and those small little vesicles. They're tiny. So for soil moisture, we select 10 grams of soils from each plot. Preheat the soil sample overnight at 100 Celsius to remove the water. For the allopathy test, this actually really fun. I enjoyed this a lot. We grind vinca leaves from the field and mix with desalinated water. Moisture the seeds with 5 milliliters of the extract. We record the proportion of the seeds germinate and the length of the roots after 6 days. They grew like really fast. I was kind of surprised. So my results for the seed bank shows there's no significant difference between seed bank in the richness or number of plants in no vinca and vinca soils. There was a significant correlation between the species distribution for the 2 seed bank. So no vinca seed bank have total 20 species with 3 non-native versus vinca seed bank with 23 species, there's 4 non-natives. So this results actually shows there's no significant use for the seed bank. They're kind of the same growth after all. So for the soil moisture, there is a trend of no vinca plots to hold more moisture than vinca plots. This actually supports my prediction, which vinca may suppress the native plants by compete with more moisture. For the allopacy, the corn seedlings are significantly lower when grown in water than in the vinca extract. Radish seeding did not germinate in vinca extract, which is surprisingly means that vinca actually produce some chemicals they didn't support plant growth at all. For the mycorrhizae, the percent of mycorrhizal colonization for corn roots was significantly greater in soil from vinca plots than in soil from vinca plots, no vinca plots and vinca plots. So the vinca may suppress the mycorrhizae fungi. They probably like have less plant growth, more native plant growth in that community than the non-vinca. The richness for the summer, spring richness and the cover value do not include in vinca minor in this data. The richness is not significantly different in between non-vinca and vinca plot in the spring and summer. Number cover is significantly great in no vinca plots than in the vinca plots in both spring and summer. The vegetation in no vinca and vinca plots is not correlated in the spring and summer. So in the spring, actually, vinca plots tend to have more species to grow around, which also could be like the vinca suppress native plant growth. The percent of canopy cover is significantly greater in vinca plots than in no vinca plots in the spring, but no significant difference in the summer. So the conclusion for this research shows that periwinkle has impact on our native forest understory species. The mechanisms such as biochemicals, mycorrhizae fungi, and soil moisture examined in this research provides the evidence that invasive species significantly influence how to compete with native species. So vinca, invasive species actually is really bad for native. So acknowledgement, special thanks for Dr. John Titus. He helped me so much for my research. And the references. Is there any questions for my research? And that's it. Thank you for everyone who support me. If there are any questions, should I wait for questions?
Thank you, Sudi. We're just going to give it a couple moments to see if any questions come in from the chat.
All right, thank you.
It doesn't look like any are coming in, so thank you for your presentation. We're going to take a short recess until about 12:50 until our next talk.
Okay, so should I just leave?
All right, everybody, welcome back. Thank you for hanging in there with us. We're going to go ahead and get started with our next presentation. We have Sage Grenz, who's an undergraduate student from Penn State Behrend. For you judges, that last name spelling is Grenz. So Sage, I'm going to go ahead and just turn it over to you.
All right, thank you. So hello, everybody. I'm Sage. Thanks for tuning in today. I'm here to talk to you a little bit about a project my research advisor, Dr. Sam Newtile, and I have been working on a little bit last year, but a bulk of this year. So to get some background information in, what is an invasive species? Roughly speaking, an invasive species is any species from another environment that's introduced into a new one. And when we think of them, we typically think of them as degrading the ecosystem's health. And most of the time they do this through outcompeting native species for resources, but it's also been seen that they can do so through the introduction of toxicants. So the toxicants of interest for us were heavy metal toxicants, and we chose these because they're known to be toxic to nearly all organisms, and they're normally introduced into environments from us. And the interesting thing about these toxicants is that they're not always metabolized and excreted, but rather they can accumulate within the tissues, and then what also can happen is they can move across the trophic level, so up the food chain, and become more and more concentrated. And that concept is termed bioconcentration. So we've talked about invasive species and we've talked about heavy metals. Let's talk about what happens when you combine the two. So it's known that ecosystems are typically facing multiple stressors at the same time. And these human-introduced heavy metal toxicants can interact with invasive species and become concentrated in them to affect the native members of that ecosystem. So gaining a further understanding of how these two factors intertwine is really critical for protecting the wild areas. So our particular species and area of interest for this study was Presque Isle State Park, which is a recreationally and commercially valuable asset in Erie, PA, and is currently under threat. It's under threat from human-introduced heavy metal toxicants from wash-off from roadways and industrial practices, and it's also under threat due to a growing population of the Chinese mystery snail. So a little bit of background on the Chinese mystery snails. They were first reported in Thompson Bay back in 2006, but as we've seen, they're quickly spreading to different areas of the park. The females give birth to live young, and they grow to be about 64 millimeters in size, which is quite a bit bigger than the native species that we've seen around Presque Isle. And this is going to be important because them having a larger size allows them to hold on to more metals, which can be potentially more harmful to the ecosystem than if a smaller snail was accumulating these metals. So our goal in this study was to determine the role of the Chinese mystery snails in the bioaccumulation of heavy metals within Presque Isle State Park. And the way I thought of this was almost as a model organism, if you will, because if we can understand how it's going on in our backyard, we can also begin to understand how it may be occurring in different ecosystems across the country or even across the world. So this experiment or project began with collecting the snails from Presque Isle. Unfortunately, I wasn't able to be there this summer due to the pandemic. I was actually working remotely on a different project. But Dr. Lynn Beatty, Dr. Adam Simpson, and my research advisor, Dr. Sam Newtile, did a great job collecting the snails from the different points listed below. And they also went ahead and collected water and sediment samples as well. And one thing that I thought was interesting was the water and sediment actually had to be held in plastic to prevent heavy metals being leached from the glass. That's something that tends to happen if you have impurities in your glass, and especially when you're working with concentrated nitric acid, which I'll explain in just a second. So once we got to the sample processing phase, which was in the fall, I was actually there and I was on my own because, as you might be able to imagine, dissecting the snails, it's not a good smell. No one else wanted to be in the room. So the way we did it was, well, I guess I should say I did it, was I dissected the snails and separated their shells and tissues. And I wanted to make a note that whenever I was dissecting the shells, I made sure not to include the operculum, which is like a little trap door that these snails have that allows them to close themselves in within their shell. Just because the operculum is actually made of keratin, whereas the shell is made up of calcium carbonate. So the concentrations in there could vary a little bit, and it wouldn't be quite representative of what's actually in the shell if you were to use the operculum. So anyhow, we took 0.5 grams of tissue or shell and weighed them into the microwave digester tubes, and we added our concentrated nitric acid. And then once they were digested, we diluted them up to 5% nitric acid in preparation for the ICP. And just as a quick note, the water and sediment samples followed a similar protocol. They had a different computerized program in the microwave digester, but the overall process was pretty similar. So quantification. We use the ICP mass spec, and I put a little heart on it here because this has really been the backbone of my project. If we didn't have this instrument at my campus, I wouldn't really be able to quantify the heavy metals and all their concentrations. So it's been really fun to learn this instrument and get to learn the intricacies of it. So we made calibration standards so that way we could quantify our target ions. And then we also made sure to purchase and use internal standards in order to control for the internal instrument variation. So when it came to data analysis, we couldn't just pull the numbers off of the ICP and use those directly. They had to be quote unquote fixed. And I put the formulas there. I won't get too far into them. If you have specific questions, we can answer them at the end. But I also wanted to explain this little meme here. So actually, whenever I was first doing this last year, I learned I was really bad on Excel. I was really slow and I wasn't good with the shortcuts and formulas. So I decided to put this little picture on there where I envision Dr. Newtile is the girl asking how's the data coming along, and I'm sitting there exhausted, working all night, trying to do it the slow way because I didn't even know there was a fast way. But through all of this, I've actually gotten much, much better with Excel. Okay, so before we get to the results, I just wanted to mention our target metal ions. So today we're showing data on nickel, cadmium, lead, copper, and zinc. And these ions can have varying effects. So heavy metals, like I said earlier, are toxic to nearly all organisms, and they can have varying effects depending on the species. But some general things I found that they cause in invertebrates is decreased growth, reduced fecundity, and reduced food consumption. So overall, it's gonna affect their survival rates. So we'll take a look at the environmental concentrations first, and then we'll take a look at the snail concentrations. So just to orient you to see like what you're looking at here, on our y-axis, we have our concentrations of metals, in this case nickel, in nanograms per gram, or in the water's case, the nanograms per milliliter, and then location on the x-axis, and those are indicated by the colors. So I won't take the time to explain each one of these graphs. I'll just go through some of the general trends that we've been finding as I flip through these ones. So one thing we've been finding is that our sediment concentrations are much, much higher than the water concentrations, which is both a good and a bad thing. It's good because we're seeing that the water isn't contaminated, but at the same time, it's raising some questions on where these snails are getting the metals from, which we'll see in a second. We've also seen there seems to be general trends, but there's also some variation within it. So for example, here we can see there's a high lead concentration in feather point. In feather point sediment. And we were thinking that this is probably due to proximity to the road. So for example, in the zinc, in Lily Pond, we're seeing higher concentrations of certain metals, particularly zinc in this case, and that's also due to the proximity to the road. So now we'll get into our snail concentrations a little bit, and we're also seeing trends here that the shell concentrations tend to be higher than the tissue concentrations. And this is likely due to route of exposure. I'll get into that in a little bit. But here, like I said, seeing the same general trends where the shell concentrations tend to be lower than the tissue concentrations, but we're seeing a decent bit of variety in here as well. So we can see on our shell concentration graph to the left here that Lily Pond is showing higher concentrations. Like I mentioned a second ago, that's probably due to the proximity to the road. And like I said, we are seeing some decent variation because here at Feather Point, we're seeing really high lead concentrations, which was surprising. And one thing I'd like to do in the future is kind of try to gain a better understanding of the area because like I said, I wasn't there this summer, and speculate as to why that may be happening, why lead in particular is high in Feather Point. And here we have copper. So again, Lily Pond has a higher concentration, probably due to the proximity to the road. And then here we have zinc. So if you look over to the tissue concentration graph on the right-hand side and look at that feather point bar, we see that it's really high up in the tissue concentrations for the zinc. And I actually recently learned that there was a pesticide program in Feather Point, and we were speculating whether or not that maybe the pesticide had zinc in it, and that's why we're seeing higher concentrations in that. But we're not exactly sure, so that's something we'd like to look further into in the future. So what does this mean? Well, basically what the data is showing us right now is that these Chinese mystery snails are capable of holding these metals in their shells and tissue. Although it appears as though they're not gaining it from the water, but more so the sediment, which may be part of the reason that we're not finding many dead Chinese mystery snails. They're actually still alive, because one thing that could be happening is that when they're ingesting it, like through their digestive tract, they're more able to sequester it in their shell. And once it gets to their shell, it's not really toxic to them anymore. It's not involved in their metabolic machinery or anything like that. So they can kind of just tuck it away. Their shell's kind of a storage container. So one thing I also wanted to mention was for my summer project, I was looking at toxicity data for various invertebrates with a focus on snails. And all of that data focused on LC50 data from water exposure. And one thing that I thought was interesting was there was no sediment exposure LC50 values. So that's something I'd like to look further into, or maybe even try to test myself to see if we can translate these high sediment concentrations and determine their possible effects on the Chinese mystery snails. So why should you care? Well, like I mentioned earlier, Presque Isle State Park is a recreationally and commercially valuable asset to Erie, Pennsylvania. It's also home to a richly diverse ecosystem. So if these Chinese mystery snails come in and they're sequestering these metals, even though it's in their shells or in their tissues and it's not directly affecting them, if they do get eaten by higher up predators or they eventually die and decay in the waters, they can release these high concentrations into the waters and start affecting native species. So what's next? Our next steps include sample processing and more data analysis, and that's going to continue through the rest of this semester and potentially into next semester. And included in that are going to be more replicates of the Chinese mystery snails, so we can get a better idea of what's going on with them and try to paint a clearer picture. And then also we wanted to compare them to the native species concentrations that we may or may not see. So if we can gain an understanding of the relative proportions that Chinese mystery snails can contain and native species can contain, we can start to paint the picture of how these invasive species are going to affect native species in a way that isn't directly outcompeting them. So I'd like to give out some much deserved thank yous. So especially to Dr. Newtile, he's really been a huge help with this project, and he's really helped me along every step of the way. Dr. Adam Simpson, Dr. Lynn Beatty went out this summer, the three of them did, and collected all those snails when we weren't able to. And then actually the summer prior to, some of my classmates, Austin, Cassidy, and Matt, along with Dr. Newtile, Simpson, and Beatty, went out and collected some snails, and that allowed me to conduct some preliminary testing to see if our methods would be valid or not. So big thank you to those guys. And if you guys have any questions, you can ask them now. Or if you aren't able to ask your question at the moment, feel free to reach out to either me or Dr. Newtile, and we'll answer your questions as soon as we can. Thanks.
Thank you so much, Sage. We do have a question for you. Did you find that when the snails were exposed to any high concentrations of the metal toxicants, that there were any morphological changes? And if so, were any of those passed on to the offspring?
Right. So we haven't seen whether or not they're passed off to the offspring. That's something I would like to look further into. One thing I did notice, though, however, was the graveyard pond snails in particular had very thin, brittle shells. So my professor and I, Dr. Newtile, we're going to try to look into whether or not the heavy metals being sequestered into the shells affects their brittleness.
Thank you. We have another question. Many of the sites you studied are also historic native freshwater mussel habitats. Do you think that the mussels can accumulate heavy metals in a similar way? And if so, do you think it could lead to their decline?
That's a good question. I would hypothesize yes. I'm not sure to whether or not it'd be a similar magnitude or a similar mechanism as the Chinese mystery snails, but I would imagine that they would be capable of accumulating these metals into their soft tissues and their shells. The trend seems to be that they sequester them in their shells, is kind of, like I mentioned earlier, kind of like a garbage can, if you will, because then it won't affect their physiology.
Thank you. We've got one more question.
Okay.
This is from Mr. McKelvey's fourth grade. A fourth grader wants to know if another animal, like a bird or fish, eats a snail whom has digested metal. Will the other animals get sick too?
Right. So that's actually kind of one of the big things, or one of the big problems that we're investigating, is that it is possible that if an other species eats these snails, that those metals could be released into the bloodstream of the predator and end up causing harm to the predator, yes.
All right, and we have one more. What are the main sources of heavy metals from roadsides? Exhaust, road materials, or are they other sources?
I would hypothesize mostly road materials, not necessarily from the exhaust quite as much, but from the different materials used to lay down the road and everything. And then those would be washed off by like heavy rain and things like that.
All right, thank you so much, Sage. We're going to go ahead and take another 5-minute break until 1:10 before our next talk. So we'll see you all then.
Welcome back, everybody. We're going to go ahead and get started with our next talk. We have Megan Adams and Nicholas Thompson, who are both undergraduate students from the University of Pittsburgh at Bradford. For you judges, this is a speed talk, so you don't have to worry about judging it. So Megan and Nicholas, I'm going to go ahead and turn it on over to you.
Thank you. Hello, my name is Megan Adams, and my partner here's name is Nicholas Thompson. We're both senior biology students at the University of Pittsburgh, Bradford, and we're here today to talk to you about Syringia reticulata, better known as Japanese tree lilacs. So the purpose of this presentation today is to briefly touch base on what this species is. We also aim to weigh in on if this landscape plant is benefiting our environment or potentially posing a threat, and discuss how this might present in future botanical research study. So what is a Japanese tree lilac? This is a deciduous tree-form lilac, similar to the common lilac, but different in species. They resemble each other in leaf shape, arrangement, and showy flower clusters, but differ in that the Japanese lilac has more of a tree-like form and can grow to 20 to 30 feet tall. Common lilacs reside around 10 feet tall. They also bloom at different times. The Japanese lilac blooms in late June and early July, and the common lilac blooms usually in May around the McKean region. So where is it? Syringia reticulata is native to Northeast Asia, originating specifically in Japan, China, Korea, and Russia. Subsequently, its presence has been reported across many countries. This flowering plant is grown and sold as an ornamental landscape plant in Europe and North America. Due to demand, wide distribution, and large population size, it's important to ask if this plant is benefiting our environment or potentially posing a threat. Here in North America, we're seeing naturalized populations popping up in the United States, such as New York, New Hampshire, Massachusetts, Vermont, Michigan, Minnesota. It's even reported to be in Ontario, Canada. The USDA considers a naturalized plant to be a non-native plant that does not need human help to reproduce and maintain itself over time in an area where it is not native. Once a population has been considered naturalized, it can then be put into the subcategory considering it an invasive population. So with all things considered, why is this such a popular landscape plant? This is a popular landscape plant because it's easy to grow, it has a moderate growth rate, and it grows to be a small to mid-sized tree. It's aesthetically attractive, producing showy, fragrant white flower bundles that attract hummingbirds and butterflies. It's also low maintenance and tolerates harsh conditions such as extreme cold, droughts, urban soil, and it's somewhat disease resistant. So that brings us to why we're here today. Around campus, our professors, Dr. Mulcahy, Dr. Piechnik, and Dr. Frantescu took notice of a plant growing on and around campus, and they took observations of where it was growing and how. They noticed that they were clustering around riparian habitats, the origin of this plant likely spreading from a landscape source. They took special interest in this plant because it looks to be establishing itself, and it may be threatening to local ecology. This is also concerning because it's deliberately being planted by the city of Bradford. So Dr. Mulcahy, Dr. Piechnik, and Dr. Frantescu wanted to answer the question, what does the presence and seed distribution of Syringia reticulata mean to our local environment? Just to make sure we're all on the same page here, a riparian habitat is an area that occurs along watercourses and water bodies. Typical examples of this include floodplains and stream banks. These habitats often serve as forest buffers. These riparian habitats serve as a transition from land to water, and they play a crucial role in maintaining the health of our waterways. And this is not the first time this riparian clustering pattern has occurred with Syringa reticulata. This occurrence has also been reported in Northwest Vermont and Western New York floodplains. So, what these studies suggest is that long-distance dispersal by water may be a factor in the widespread dispersal and spread of Japanese lilac trees. And the possible dangers of this species in Pennsylvania, it deals with the riparian buffers playing a crucial role in maintaining the health of our waterways. They filter stormwater, sediment, nutrient runoff, and they improve water quality. And it's also a source of food and structural habitat for aquatic organisms. So since we already talked about the potential dangers in Pennsylvania, I'm going to go ahead and hand this over to my co-speaker, Nicholas, so he can discuss the benefits.
Other than the plant being aesthetically pleasing, we're not really sure if there are any benefits to the presence of Syringa reticulata in Pennsylvania. There may be a role that it plays in the Pennsylvania buffer system, but to our knowledge, that link is yet to be discovered. We're interested in this plant, Syringa reticulata. Also, it may be a dead-end host for the ash borer. The ash borer is attacking ash trees here on our campus and throughout the state. And both of these plants are both members of the Oleaceae family, and we're not sure if Syringa reticulata is a host to the ash borer or a dead-end host. So we're interested in also understanding this relationship further as well. Thank you, Megan.
You're welcome. Okay, so unfortunately, what we are certain of is that neighboring states with similar climate and landscapes are seeing naturalization. Once a population has been considered naturalized, it can then be put in the subcategory considered as an invasive population. So this leads us to the question, does this fit the criteria as an invasive species? The USDA's National Invasive Species Information Center, enacted under the Clinton administration in 1999, defines this criteria used to declare a plant invasive as a non-native plant that is under consideration that will likely cause or is likely to cause economic or environmental harm and harm to human health. So by federal criteria, this species would meet the first criteria as a non-native plant under consideration, that due to its patterns on that due to its patterns on naturalization in the States. However, due to uncertainty, the second criterion couldn't be met. And in Canada, it has caused reason for concern due to it being invasive in its natural areas and has been placed on their watch list. But the US federal and state agencies have yet to declare it a plant of concern. To explain why there's a division in federal and state efforts in managing invasive species, it's important to recognize that this is too big of a job for a single agency, so they coordinate together. So, in conclusion, it makes sense that this is a popular landscape plant due to its growth patterns, attractiveness, low maintenance needs, and tolerability. But with all things considered, it may not be healthy to our environment. due to its reputation in terms of naturalization and suggested long-distance dispersal by water. Although we're not certain it is establishing itself at Pitt Bradford, it is suggested that it may become invasive due to its patterns in its natural habitat. This also suggests that there is room for us to be concerned that this plant being deliberately planted in Bradford City. So, we suggest that future research examine the role that Syringa reticulata has on nutrient cycling and buffer zones in order to determine if it's impacting the ecology of the riparian zones or altering the cleanliness of our Pennsylvania water passages. As well as we also suggest future research expand upon the examination of distribution patterns such that it include dry forest land and how that is impacted as a consequence of these riparian buffer distributions. And does anybody have any questions for me?
Thank you so much. We don't have any questions for you, so we're going to go ahead and get ready to move on to our next talk.
To Dr. Mulcahy, Dr. Piechnik, and Dr. Fantescu for allowing us to present on their research. And I hope you guys will stay tuned and listen to the ideas expanded upon by a few of our fellow students. Thank you for your time. I hope you enjoy the rest of your day.
Am I talking? All right. So we're going to go ahead and get started with our next talk. We have Samantha Kircher and Arian Holness, who are both students from University of Pittsburgh at Bradford as well. And so without further ado, I'm going to pass it over to you guys.
Hello.
Hello.
Hi, everybody. My name is Samantha Kircher, and my fellow partner and I, Arian Holness, expanded upon the previous research by asking, how is the tree lilac spreading? So a little bit about us. We are students at the University of Pittsburgh at Bradford, and we currently are enrolled in our field botany class with Dr. Mulcahy. We are both undergrad students. I am a double major in criminal justice and forensics, and Arian is a major in biology. And then Dr. Francescu, Dr. Piechnik, and Dr. Mulcahy were the ones that asked us to kind of help out with this, kind of brainstorm and come up with some ideas with this research. And then they also did most of the research over the summer.
Our main focus is in knowing, are the seeds of the tree lilac spreading by floating down the Tonunguan Creek? In the later slide, I will briefly go over how this local waterway may be impacting the spread of the seeds. As you can see in this centered picture, that the tree lilac is obviously fitting in and it's not planted. It seems to have blended in with the other shrubs around it. And those are other pictures of the seeds, and those are other blossoms of the flower of the tree lilac. Other questions that may support these questions are, how do other invasive species spread? So in general, we like to ask this question, what are some ways that this invasive species spread? According to the National Park Service, humans are probably the biggest cause for the spread of the invasive species in general. Things that can spread are by way of travel or clothing, just latching onto clothing in general. Other large causes do include animals, in which that things may latch onto their fur or are disposed onto their waste. Elements of the weather may also easily play a role due to strong winds or heavy rains. Another important question we like to ask, this summer, as mentioned before, Dr. Mulcahy and Dr. Piechnik were trained and proceeded to geolog the tree lilacs presence in and around our campus community right from their cell phones. They used IMAP invasives. A cause for some gaps in this map were due to unsafe conditions and inaccessible private property. The map tree lilacs are likely to be clusters of trees found together. To the far left here in the corner, Willowdale Cemetery, there may have been tree lilacs planted there and hypothetically spread down into the lake, the Marilla Brook Creek Lake. The hypothesis can work because our school, University of Pittsburgh Bradford, is down in a valley in between those dark indentations, which may be hills. So the hypothesis is that the elevation is coming from a higher elevations up by the cemetery down into the valley where the university is, where our instructors have found and mapped several clusters of the tree lilac tree.
So a few other questions that we have proposed are, do the seeds spread by floating downstream? As Ariane kind of established, this is one of the biggest questions that we have. And then something to go off of that is, what determines whether a seed will germinate in a particular location? So based off of that, it would be the environment in which the seed is in for germination. The next question is the plant invading riparian habitats? So as reflected off of the previous presentation, that has been apparent of a very big question that we are looking for in this study. Another question is, how detrimental is this plant to the riparian plant communities around Pitt Bradford? That is also another question that we have posed as well, just because of the abundance of clusters that have been found near our campus. And then the last question is, how could future students attempt to answer these questions experimentally? The reason why we asked that question is because Ariane and I are both graduating. So we would like to see if there is a way that we can assist future students with how to go about this experiment and see if we can aid in it before we graduate. Some possible studies that we thought could result in solutions to those questions would be comparing the seeds based on the size, shape, and density. And then once those comparisons are found, figure out which seeds are likely to float on the water. Once we have found the density of the lilac seeds, and we would like to make a homemade water system that is like a creek to see how the seeds do in the water conditions in terms of floating down the creek. The next one is create a seed capture system and then yearly record the tree population on campus to see the impact based off our hypothesis.
So one tool that was made available for future Pitt Bradford students was with the help of Dr. Francescu. He's purchased and provided a stream table kit that can help stimulate the conditions of the Tunungwat Creek. We believe that many factors contribute to how the seeds are moving down the stream and what other conditions are needed for the seed to actually germinate there. These factors can include water elevation levels, rainfall, high winds, and hot or cold weather.
So we are going to go ahead and wrap this up. We would like to shout out a big thank you to Dr. Mulcahy, Dr. Piechnik, and Dr. Francescu for asking us if we would like to participate in this. We would also like to thank the University of Pittsburgh at Bradford for their resources and everything to go through this experiment. We would like to thank the Symposium for giving us the opportunity to present our information. And we would also like to thank our field botany class for the interest in this topic and for the assistance with it. Thank you all.
Thank you.
Thank you so much, you two. We're going to go ahead and get started with our final talk in this session. We have Amanda LaVose, who is a student as well from the University of Pittsburgh at Bradford. I'm going to go ahead and turn it right over to you, Amanda. Amaya, sorry.
It's okay. Hi, everybody. Can you all hear me? Let me share my screen. Okay. Hello, so my name is Amaya Lovos, and I'll also be discussing some aspects for the Japanese lilac tree. Listed below are questions I came across looking into this research. I am a senior, and hopefully this could be something I could continue to look into during the spring semester. I wanted to talk about the range a little bit, and this, I had some questions in what areas are the Japanese lilac tree mainly grown? Another thing that'd be good to look into is, how did they become popular in certain areas in the U.S.? In regards to the plant, do they have any healing properties? How many shops sell them, or how much do they charge? And in regards to the ecosystem in the community, how are the Japanese lilac tree affecting the environment and the other plants around them? So one thing I was thinking in regards to this research is that could an online database be useful for my research? And I came across this database, GBIF, which is the Global Biodiversity Information Facility, which contains free and open access to biodiversity data. It consists of species of all biodiversity, occurrences, data sets. It consists of publishers, resources, and background information. This is specifically a website that you can go into, and I also included the link here, which I think would be something very useful for this research. And here is why. In this particular website, what I did was put in the Japanese lilac tree, using their scientific name, of course, Syringa reticulata. And I compared how the trees were spreading from the 1950 through the year 2020. As you can see here, they show you, some areas where the occurrences are for this lilac tree in the US, in some particular areas in Europe, and some areas around East Asia. And, it's adjustable with the time frame. You can also change the month, look into different data sets and countries within this plant. So I feel like that was pretty interesting. And they even offer you, so you can look at an entire data with the occurrences throughout the year, and they even provide you with the different months and what months are more common than others. So I feel as though that would be interesting as well. Here are a couple of businesses in the Pennsylvania area that advertises and sells the Japanese lilac tree. I found a website online from York, PA, and another one that is located in Allentown, Pennsylvania. So these are just two examples of some businesses that I found. And up here, I have uploaded a website that also advertises the Japanese lilac plant. And in this particular website, depending on the plant, you can incorporate your zip code. So I put in Pitt Bradford's zip code, and it says here that for the Japanese lilac tree, the growing zones are from 3 to 7. And here it says that we are growing zone 5A. And as you can see here, when they're advertising the lilac tree, they're describing how, you know, it blooms when other flowering plants are not in bloom. It's a massive, it creates a massive flower display. You know, it's beautiful. The fragrance are amazing. Perfect for the winter weather. So they use like these really good targeting marketing words to get people to purchase this, not understanding that it could possibly be invasive. On this last slide, me and Dr. Mulcahy was kind of discussing some things that I was interested in. I am a bio major, so I get interested in looking into like home remedies, medicinal properties. If you know, certain plants contain certain components that are important. So in this particular research, it says that, you know, there's been a new discovery with 2 phenolic glycosides from the Syringa reticulata. The phenolic compounds are known to be used for reducing the risk of cancer by nutritional properties. And also low concentrations of phenol is known to be found in disinfectant products for household items, as well as for dental care products such as mouthwash. And a medicinal property that perhaps it may help with, it was known that this plant has been used as a traditional Chinese medicine for the treatment of bronchial asthma and as an expectorant. So I figured this was just something interesting, a possibility of looking into as well. Of course, there's still research out there that still needs to be discovered. But I want to end this by saying thank you to Pitt Bradford. I want to give a thank you to Dr. Mulcahy, Dr. Pitchnick, and Dr. Francescu. And I also want to give a thank you to the Regional Science Consortium. Thank you.
Thank you so much, Amaya. We do have one question for you. Is there a large population of these Japanese lilac trees in the Bradford region?
Within the Bradford region, when I was looking up that area, it really depends on the certain areas within the database. It also shows like other states, like for example, Mississippi, you know, it's a little bit more common there than the Pennsylvania area, but Pennsylvania do heavily advertise these plants as well. And, you know, you'll find a lot of websites out there that would, you know, also advertise these plants too.
All right, thank you so much. And with that, that brings us to the end of this session. So we're gonna go ahead and take a short break until about 1:50, and at which point we'll be back.
It's no secret, education is the key to success. Serving more than 2,200 families, WQLN brings learning to life by combining PBS Kids programs with reading and hands-on activities.
PBS Learning Triangle, I feel like it's a simple step for parents to grasp. So it's not something over their head or too complex.
Watching my students for the 4 weeks develop a little bit more of their personalities, have the opportunity to read and have the opportunity to be excited.
People say it takes a village to help raise students. It also takes a village to educate them. So WQLN coming in to partner with our kids helps those kids get those strategies and the parents the strategies that they can help their kids at home.
You're part of a community, and we're all here together, and we all care about the children and the future of the community.
To become an EITC partner and support this program, visit WQLN.org.
ETD Photography is an Erie-based media and creative services company by Eric Dye, creating media to expand your reach and further your goals. With a diverse project history, ETD Photography brings unique and comprehensive creative media to Erie individuals, businesses, and organizations. Services include photography, videography, web design, logos, podcast production, YouTube channel production, live streaming, and more. Check out our website at etdphotography.com.
Hello, my name is Dr. Jeanette Schnars, and I'm the Executive Director of the Regional Science Consortium. And I'm here at Presque Isle State Park today exploring the priority wetlands that we've been working on with our collaborators to restore over the last decade. Now, wetlands are a critical ecosystem that help protect Lake Erie and other bodies of water that they're connected to from runoff, and that would include sediment in the runoff, nutrients, and pollutants. And then wetlands also serve as critical habitats for all different kinds of species, including plants and insects, fish, birds, and other mammals that live here. Today we're going to investigate the relationship between humans and wetlands and how this has changed over time. So wetlands, once thought of as swamps or just pools of stagnant water that were unsightly, were often destroyed as a result of construction and development. However, today their importance is realized, and a healthy wetland exhibits high plant diversity and animal diversity. However, due to invasive species and some other human interactions, some wetlands are not considered healthy, and that's where we come in as researchers to help restore these areas.
My name is Holly Best. I'm one of the assistant park managers here at Presque Isle State Park. I work with the resource management program. A large part of that program, we try to manage invasive species that are growing on the park. Some of the main targets are Phragmites and narrowleaf cattail. Phragmites are specifically important to manage because not only are they very prolific when they come into an area and take it over, they spread through rhizome, they also spread through seeds. They also are allelopathic, so they actually put poison or toxin down into the soil and prevent anything else from growing. So that makes a monoculture of that plant, which for species diversity is very detrimental. At this park, we're very fortunate that we are able to use herbicide to help control these species. We have done everything from have interns with backpack sprayers, doing a very focused attempt, all the way up to helicopters actually doing kind of broadcast spraying over some of those very large monoculture areas. We're fortunate that we're able to use that as a control technique because if you don't have that ability, it's very hard to control these species and try to return these areas back to their native environments.
My name is Jen Salem. I'm the horticulture specialist for the Regional Science Consortium, and I'm also the founder of Go Native Erie. Go Native Erie is a program that operates under the Regional Science Consortium, and it helps educate the public about the benefits of native plant species and using natives in their gardens. Native plants are actually, they're defined in two ways. They are plants that were discovered here growing in North America at the time of European settlement. But a better definition of that is that native plants are a plant that has been in an area for a long time and it has evolved with something else to form an important relationship.
We are in the Regional Science Consortium greenhouse at the Tom Ridge Environmental Center. So we use this for propagation. So we get things growing and started here.
So this is the beginning of the process. So every year I'm looking at what seeds can I grow, what did well. Every year we're trying to increase our diversity and the amount of plants that we grow, but also pay attention to things that are going to do well. It's not just about quantity, it's also about quality. So now we're down at the Regional Science Consortium's cold frame greenhouse on Presque Isle State Park. And this is used as a distribution center and holding area for all the plants that are produced in the Regional Science Consortium propagation greenhouse. So when plants grow up to be a certain size and they're ready to be planted in a wetland, they are brought down here and held at this site and cared for. Then when we have groups of volunteers and employees that can come and help, we decide what plants are going into each of the restoration locations, and we'll just load these all up on a truck and have a planting day.
All right, so I hope you enjoyed the tour of our wetlands and all the work that we're doing with our collaborators to help restore these areas. So if you'd like to learn more about this project or other projects, please visit the Regional Science Consortium's website at www.regsciconsort.com. Hello, my name is Dr. Jeanette Schnars. I'm the Executive Director here at the Regional Science Consortium. So the RSC is a collaborative. We are made up of colleges and universities, state and federal agencies, K-12 school districts, as well as other nonprofits with similar missions. And some of the research we do focuses on water quality, especially here at our swimming beaches, harmful algal blooms, different types of fishery, including yellow perch and walleye, invasive species, wetlands, native plants, and even shipwrecks and exploration of other submerged cultural resources in Lake Erie. If you get a chance today, make sure you ask at the front desk for a tour to learn more about the different projects that we have going on back there. So we use these research projects as fuel for some of our education and experiences that we offer to students in our school district membership. So these students get to experience things with invasive species, with our native plants, with fisheries, and we even have other experiences that they participate in, including a college prep workshop series. To find out more about our research and education, and also to find out if your organization, school district, college or university is a member of the RSC, just check out our website at regsciconsort.com. Hi, I'm Dr. Jeanette Schnars. I'm the Executive Director of the Regional Science Consortium, and I'm standing here on the shores of Lake Erie, which is known as one of the best sport fisheries in all of the Great Lakes. And two of the most popular species include yellow perch and walleye. The fishery historically has always been great here, but it has had some pressures in the past, including over-harvesting of certain species and other things like invasive species. These invasives have really paid a toll on some of our best sport fisheries.
Hi, my name is Greg Andreso, and I'm a biologist from Gannon University here in Erie. And for about the last 20 years, my students and I have focused on the fish population in Presque Isle Bay behind me. A lot of our work has focused on the round goby. The round goby is a non-native species from the Black Sea, Caspian Sea part of Eurasia. And it was first introduced into Lake Erie in the early 90s by ballast water in freighters. So these freighters brought these fish across the Atlantic and offloaded them unintentionally into the Great Lakes. So a lot of the work that we've done is focused on round gobies and their interactions with two species of mussels, which are actually non-native as well, what we commonly call zebra mussels. And there's actually another species that's closely related called quagga mussels. And our work has really looked at predation by round gobies on these Dreissenid mussels, the zebra mussels and quagga mussels. Round gobies eat things like smallmouth bass eggs, so they've had an influence on the ecosystem. They're here to stay. I don't think we're ever going to get rid of them. And part of my job and the job of my students has been to try to better understand their roles in that ecosystem.
Hi, I'm Dr. Casey Bracha-Wilson from Allegheny College. I am studying the invasion of round gobies from the Lake Erie watershed into the French Creek watershed, which is a stream that ultimately flows to the Mississippi River. So this invasion is a really important one that we've been studying and monitoring since they've first been introduced, probably through some type of fishing activity, whether it was through a boat or through bait bucket transfer or something like that. We have a lot of projects going on, including what the round gobies are eating, how they're impacting native fish through behavior and aggressive interactions, also how they can change communities through eating macroinvertebrates and other fishes and their eggs and those types of things. French Creek is an extremely important stream in Pennsylvania. It houses more diversity than any other stream in the state, including many threatened, endangered, or species of concern. So round gobies have recently been known to eat native mussels in the juvenile phases when they're very small. And so a lot of the mussels in French Creek are threatened or endangered species. And so the impacts to mussels from round gobies, we don't know exactly how catastrophic that will be, but it could be really bad for the French Creek watershed.
The Pennsylvania Fish and Boat Commission regulates fishing here in Erie, Pennsylvania, and on the waters of Pennsylvania for all of Lake Erie. Lake Erie has been subject to a lot of invasive species throughout the years, all of which have unknown effects when they first get here, and it's only through their actual arrival that we're able to manage them. 2018, the Pennsylvania Fish and Boat Commission joined with our cooperators in Ohio, Ontario, and New York and started tagging walleye here off the shores of Pennsylvania, right here off of Walnut Creek Marina, looking at spawning site fidelity as well as fish movement throughout the summer months and even into the winter. To date, 41 walleye have been tagged here at Walnut Creek and released back into Lake Erie, and we've watched those fish travel for almost 2 years now. We have 21,000 detections of those fish in the last 2 years. And with that, we were able to see if these fish do exhibit spawning site fidelity and return to Pennsylvania waters to spawn. The Pennsylvania Fish and Boat Commission operates the Lake Erie Boat Angler Survey annually from May 1st to October 31st. We run a bus route style creel survey in which we sample anglers and boaters from Walnut Creek, Lampe Marina, East Avenue, Boat Launch, and Northeast Marina. In this process, we count all the boats that launch, and we count all the boats that land at those marinas during the times that we're there. And while we're there, we try to interview all the anglers that land. We try to find out some economical information from them, where they have traveled from by getting their zip code, how many folks were here, how many folks fished, how many rods they used, how long they fished, what they were targeting, and what they caught. And from this information, we can start to come up with a figure of how many fish are successful, or how many fishermen are successful, and how many fish are harvested from Lake Erie annually by our recreational anglers. The Pennsylvania Fish and Boat Commission is very interested in sustainability. So with all this information and all this work we do throughout the summer with the other jurisdictions that manage Lake Erie, we're able to make sure that the fishery is sustainable and that the anglers have the best opportunity of success every time they go out in the water.
So as you can see, our evolving relationship with the fisheries and the people that live here is so important, both historically as well as currently. And researchers will continue to work on the different issues that arise and challenge our sport fishery, and managers will continue to manage and sustain these populations for the future.
It's no secret, Education is the key to success. Serving more than 2,200 families, WQLN brings learning to life by combining PBS Kids programs with reading and hands-on activities.
People say it takes a village to help raise students. It also takes a village to educate them.
You're part of a community, and we're all here together, and we all care about the children and the future of the community.
To support the program, visit WQLN.org.
ETD Photography is an Erie-based media and creative services company by Eric Dye, creating media to expand your reach and further your goals. With a diverse project history, ETD Photography brings unique and comprehensive creative media to Erie individuals, businesses, and organizations. Services include photography, videography, web design, logos, podcast production, YouTube channel production, live streaming, and more. Check out our website at etdphotography.com. The Regional Science Consortium is a collaborative of colleges, universities, state and federal agencies, K-12 school districts, and nonprofits. The mission of the RSC is to promote and enhance our knowledge and understanding of the Lake Erie and Ohio River Basin ecosystems through research and education. The RSC also provides educational lessons and experiences for students, including forensic science escape rooms, hackathons, and a college prep workshop series. Each year, the Regional Science Consortium hosts an annual research symposium to highlight local research and projects. The Regional Science Consortium's 16th Annual Research Symposium, November 4th through 6th, will be live on YouTube at regscitv.com. Presenters will highlight local research on water quality, harmful algal blooms, wetland restoration, invasive species, fisheries, and more. David Frew and Jerry Skripczak will present Wednesday evening, November 4th, and PADCNR Environmental Education Specialist will present November 5th. All are welcome. Details online at regsciconsort.com. Supported in part by WQLN.
This piece right here is called a sonde, and it's the exact same thing that's under the buoy, and it measures temperature, pH, conductivity, dissolved oxygen, and so we measure this handheld unit against the buoy, and that helps us determine if we need to calibrate it or not, make sure it's measuring correctly. Temperature is 22.250 degrees Celsius. This unit right here is our weather station. Measures things like wind, air temperature, even takes data on rain. In the center of the buoy right here, we have our beacon. That light is a white light that comes on at an interval of every 2 seconds to make sure that mariners in the evening or at night are aware of the buoy and can see it. Our Garmin GPS unit helps us keep track of the buoy. If it were to ever break free, we would be able to find it. We use these solar panels here to recharge the marine batteries inside the core of the buoy. So they've taken energy from the sun every day and recharge those every night. Our antenna right here helps us with cellular reception, so we transmit our data every 20 minutes through a cell phone connection or a cellular modem back to our computer that processes the data. And then on the opposite end of the buoy, we have a video camera, and that video camera allows us to get 20 or 10-second clips of the conditions out here, the lake conditions, and update that to our website so people can see what those conditions are. Inside, in between all of these solar panels, is the wave meter. And that wave meter is measuring these waves, which are pretty decent today. And those waves are being monitored over a 10-minute interval, and then it will give you the maximum wave height as well as the average wave height during that time period. Now, under the buoy is what's called a water quality sonde, similar to what we were just using and dropped in the water. And that's going to measure things like temperature of the water, pH, dissolved oxygen, turbidity, and blue-green, the presence of blue-green algae. So the Regional Science Consortium has 2 weather stations on land and 4 buoys that they put out each year. This is our nearshore buoy, 2.5 miles off of the lighthouse at Presque Isle State Park. We also have another buoy similar to this, a little bit smaller, that we deploy off Walnut Creek to the west of here. And then we have 2 smaller beach buoys that we deploy off Beach 2 and Beach 6. Those beach buoys are just measuring parameters about the water, like temperature and the chemistry of the water, and that helps us with predicting if there's blue-green algae, harmful algal blooms, bacteria in the water, so we use that for a lot of research. The buoys are deployed in May, so as early as we can get out here, and then we usually retrieve them around October at the end of the boating season. Okay, so now we're back to my office, and this is where the information from our weather stations and our buoy come back into the computer and then get processed and eventually to the website. So this is the software that runs the data that comes in from the buoy, and it does some calculations to figure out things like the maximum wave and average wave, as well as your maximum wind, your maximum rain, and then averaging those data points as well. So right here you can see we're looking at the nearshore buoy data. We have a lot of the information you might see on the website, as well as some information that we need here just to make sure things are operating correctly. And we can scroll through any of our weather stations or buoys, and we can also make sure they're all updating when they should. So this will scroll new data about every 20 minutes, and then this data will be processed, and I'll just click on our website. This is PALakeErieBuoy.com, and then if you come into our website, you can see our weather stations as well as our different buoys. And this is the nearshore buoy that we were just visiting. We click on that and it will go ahead and load the data, and we can see how big the waves are, how fast the wind is blowing, and what kind of temperatures we have out there. So right now, looking at it, we have about a maximum of 2.8 foot waves, about an average of 1.6. And when we were just out there earlier today, we probably saw good 2 foot waves with a little bit of white capping. All of the data looks good and current, and that's how we process the data. So another project that we work on at the Regional Science Consortium is looking at barotrauma and yellow perch. And yellow perch is a really important fishery to this area. And it's one that reproduces naturally, so we want to make sure we take care of it. And one of the issues is that late in the summer, yellow perch can be as deep as 50, 60, even more feet of water. And when anglers pull them up, a lot of times their swim bladder will expand, which pushes on their stomach, and their stomach protrudes through their mouth. And there's other symptoms that they experience as well, such as bulging eyes, hemorrhaging around the fins. And so what we're trying to do is see if they are capable of recovering from this barotrauma, the stress, And one of the ways that we do this through some of our research is by putting them in these cages. And with these cages, it's almost like an elevator. We can sink them right back down to the bottom pretty quickly. Now on these cages, we outfit them with a GoPro camera across the top, and we can monitor what they're doing for the first hour that they're submerged. And we have found that within about 10 to 15 minutes, these perch will right themselves. So when we put them down, they're kind of belly up and bloated. So in about 10 or 15 minutes, they'll right themselves or turn themselves right side up again. Their stomach goes back into their bodies, and they're able to swim around like a normal fish. So then the next question is there any latent or delayed mortality? Did they have any injuries inside their bodies? And we found after 3 days, they are still healthy fish and able to overcome this stress. So we'll go ahead and we'll deploy this cage. We'll just hold the top so it expands out. So when we're out here, we'll collect perch by angling. We'll take that perch, we'll put it in the cage, we'll lace the cage up, and then we'll go ahead and put it down to the bottom. So when we pull these cages back up after 3 days, the perch are still healthy fish and they were capable of overcoming that stress. However, that's the story we found in the beginning of the season when the bottom temperatures were still pretty cool. As summer goes on, the bottom temperatures in the water get warmer, and as temperature increases, dissolved oxygen decreases. And when this happens, that's just another stress that's put on these fish. So we find that when we pull these cages up, we have about 100% mortality. And so between the barotrauma as well as low dissolved oxygen, high temperatures, these fish just can't overcome all of those stresses. So early in the season, real early in the spring, we might find yellow perch as shallow as 17 feet of water. And as spring comes on and moves on into summer, we find that these fish migrate from the shallow waters out into the deeper waters. And that's how we can see that, you know, the seasons are changing and that these fish move with the change in temperature, other fish as well, and they move out into the deeper waters by the time we get to summer.
Hello, welcome back to our afternoon session of the Regional Science Consortium Symposium. My name is Jen Salem, and I'm the horticulture specialist for the RSC. I'll be your host for this afternoon's proceedings. So our first speaker is Sarah Magian, and she is presenting on RSC Online Education Initiative, Addressing COVID-19.
All right, am I good to go? Okay. Okay. Hi, I'm Sarah Mackian. I'm the Education and Outreach Coordinator for the Regional Science Consortium, and I'm going to talk to you guys today about our new RSC online education initiative. This is something that we developed in a response to COVID-19. So we wanted to address the challenges that COVID-19 puts forward, and those include things like changes in school schedules. So we know that a lot of schools haven't been able to stick with their normal schedule. They either have certain grades at home or they're swapping them out on different days of the week. There's definitely no in-person interactions that we can have right now, but we still want to maintain our hands-on experience that we're known for providing for our students. Also, a lot of schools have a variety of responses to this, so we want to make sure that our lessons can work for those who are in their classroom and even for those students who are doing their work from home. We also know that this will go on for an uncertain time frame, so we want to make sure our lessons are available no matter what. And we want to make sure that they're available whether or not internet access is available to the students. So the solutions that we have to these challenges are to create both hands-on and descriptive lessons. And we are only doing virtual interactions, and we have two different styles of these. So the first of these is our pre-recorded virtual interactions. And these are our videos, which you can see an example of in the upper right of my slide, and our lessons. The lessons are pre-recorded PowerPoints where we voice over the lesson and can give it to the students, but in a pre-recorded fashion. We are still doing live interactions, which we're excited about in our Skype with a Scientist sessions. I will give more details on that in a moment. All of our components are ready from the start, so you have the full lesson the instant you log on, and that allows this to incorporate easily into a teacher's schedule, so they can look at the different components, decide what they can handle that day, if they can get it all done, if they need to split it up, it'll work very easily for that. We also developed a variety of activities that work either in the classroom, or if the student needs to perform these from home as individuals, the activity can work in both ways. This is also a long-term education initiative, which means that even once we can go back to working with our students in person, we will continue to offer these lessons. That'll be really beneficial to some of our longer distance members and maybe even allow us to add more long distance members. We also are providing offline versions of most of these materials. So the way that the materials are accessed is online on our website. However, all of those materials can be downloaded and distributed and do not need the internet to be completed. So that will be helpful for students who have trouble getting internet access. So we are using a variety of different topics for these lessons, which is really exciting. Right now we have 6 different topics available. There are bacteria in aquatic environments, buoys and weather, harmful algal blooms, microplastics, pollinator pathway, and stormwater runoffs. You can see those cover a wide variety of different things within the sciences. And we also plan on adding 7 new topics, which are aquaponics, erosion, geology, herpetology, herbarium, macroinvertebrates, and seed germination. We have many, many more ideas, but these are all the ones that are currently finished and in the works. So each of our lessons has 7 main components. Some of our lessons have extras, a little bit more than 7 of these, and that can depend on if we wanted to do more than one activity or vary it for different grade levels. But overall, they all have these 7 basic components. So that's an introduction, a teacher guide, a narrated presentation, an educational video, activity instructions, student worksheet, and a Skype with the Scientist session and discussion points. So I'm going to go in depth into all of those, starting with our introduction. So when you first look at a lesson, this is what you're going to go to. This is our introduction document, and you can see on the right, this is for our stormwater runoff. That's what I'm going to use for our examples in this PowerPoint. And this has our subject for the topic, the grade levels we feel it is appropriate for, and the number of participants that this activity will work for, all right on the front cover. And then once you go inside the document, you find a supply list, a timetable, the goal of the lesson, additional resources for the lesson, such as other related RSC online lessons or citizen science programs, as well as the PA academic standards that this lesson meets. So let me show you that. This is still for our stormwater runoff lesson. These are the 2 other pages. So you can see, I don't know if you guys can see my cursor or not, but over here on the left, our supply list, which shows all of the materials that are included within the lesson that are available online on our website. as well as materials that we will provide to the schools and the students, which would include things like gloves, materials that the students will provide themselves, things that are very common household items like a trash bag, a pencil, if they happen to have a clipboard to help them work on their worksheet. We also have over here our timetable. This will be super useful for our teachers whenever they're planning their lessons. They will know exactly how long the PowerPoint is, the video, how long the activities are going to take, those sort of things. We have our goal of the lesson down here, and then our additional resources are listed. So for this one, we have 5 related RSC online lessons, some of which have already been released, some of which have not been released yet, but they will pair well with this lesson once it has been completed. We also have a citizen science program listed here for students who want to keep doing this work outside of the lesson, and then our academic standards. All right, so our next components we're going to talk are presentation for us, so you get to see her actually talking to the students and describing these different concepts to them and teaching them all in a pre-recorded fashion. So most of the presentations are between 10 and 20 minutes. One of them goes a little over. That was the stormwater one that I filmed. And the presentation is what is going to provide the majority of the information to the students. That's where they're going to start. That's where they're going to get their basis in the topic. And then that will be followed up by the educational video. And each of our videos covers a slightly different information. So some of them will show field work that is related to the topic. Some of them will be a demonstration of the activity that goes with that topic, and others will provide additional information, maybe from special guests or things like that, that won't be included in the PowerPoint. So I have included here the video from our stormwater runoff, and you guys should be able to hear the audio. I'm just going to show you like the first 20 seconds. This one is a demonstration of our activity. We have this little introduction that is taken throughout all of our different lessons. And then
hello everyone, my name is Sarah Maggian. I'm the Education and Outreach Coordinator with the Regional Science Consortium.
So you can see here, this is just going to show me going through the activity with the students, which will be really helpful, especially if the students are trying to do this from home. This is a great way to show them an example so that they can go out and do it themselves. And it helps them feel more connected to us as we educate them. All right, let's see here.
And I'm going to show you guys.
Okay, now our next components are the activity instructions, as well as the student worksheet and the teacher guide. So our activity instructions are fairly simple. It is walking the students through how to complete the activity. It does include background information, which is a general summary of the most pertinent information to complete that activity. So it's the information they gain from their presentation in a summarized quick form to help remind the students before they complete the activity. It also has an objective and then the instructions. We have the student worksheets. So you can see on the right, this is the student worksheet for the stormwater runoff lesson. And the activity we're doing for that one is a storm drain survey. So students get to go out, examine a storm drain, clean it off, find different information about it. And then a really favorite one of mine is our teacher guide, which includes an answer key to the student worksheet, as well as lesson tips and tricks. So I'm going to show you that on the next slide. Here is our teacher guide for stormwater runoff. So you can see over here is our answer key on the left to the storm drain survey activity. And then on the right, I have shown our PowerPoint and activity tips and tricks. These are going to be really helpful for teachers before they start doing this to just take a quick look, see if there's anywhere like we have here. We recommend pausing the presentation to allow students to answer the questions that we have in it. Or down here, we don't want students going out to do their survey if it's raining, just little things like that. That we've included in here as little tips and tricks to help our teachers. All right, now the final lesson component is the Skype with a Scientist session. This is going to be really fun. This is our opportunity for our students to engage with us directly, even though it's not in person, it's still that one-on-one interaction that we get to have. So our sessions will either be with a member of the RSC staff who is an expert on that topic, or with another expert in the field. So if you wanted to talk to a specific researcher who does work on Lake Erie, we could connect you to them, say if it's a professor at Gannon or something like that. These are going to be about 15 minutes, but they can be longer or shorter depending on the number of questions students have or technical difficulties. And we do include frequently asked discussion points. So it's just a list of questions to help get students thinking about what they can ask us and things like that. All right, so now I'm going to take you through how you would access all these materials on our website. So of course you would go to www.reachsciconsort.com. And then up here on the very top tab, you're going to scroll over to our education button and click on that to reach our education page. So this is our education page, and then you would scroll down. So this little blurb down here that we've input with our little green herbarium book next to it, this takes you to our Education Initiative online lessons. So what you see here is a brief description of the lessons. And then if you are a member, you would click on this link where you will have a, you'll be able to input your password to access the site and access all of our lessons. If you're not a member and you still want to be able to test out our lessons, we do have our stormwater runoff lesson available. Even if you're not a member, you don't need a password to access this one, and then you can use that lesson. So after you click on that, you'll enter your password here, and then you'll be able to access our main site. So you can see all 6 of the lessons are listed on our site right now, and the site might change how it looks in the future, but this is what it looks like right now. And you can see we have bacteria and aquatic environments, buoys and weather, harmful algal blooms, stormwater runoff, pollinator pathway, and microplastics all on there at this time. So I'm going to go to stormwater runoff to show you guys that one. And here is the main lesson site. So this is where you access all the materials that are needed for the lesson. They're all listed right down here in the order that we generally recommend you'll look at them in. And then there's also a button right here that just downloads all of them for you, so you don't have to click on each of them individually to download it. All right, we are really excited about this initiative because it provides a lot of benefits both to us and our members. So it allows us to maintain our quality educational opportunities that are expected by our members. We really miss seeing all your students and being able to interact with them. So we're really excited to be able to do it in this fashion, provide them with our same activities and be able to talk to them through Skype with a scientist. It's also going to allow us to provide more long distance membership opportunities and be able to increase our flexibility with our education events. So if we can hold 5 different Skype with a scientist sessions in one day, we can reach 5 different members without having to drive all over the state. It actually allows us to talk with you guys a lot more. We can also feature local research, such as our members at, say, different colleges. We can talk to those professors, bring their research into it, talk to students about it. And we can also feature our local information. So things that are seasonal. Most students aren't in school during the summer, and that's when all of our field research happens. So having this format of education lesson allows us to show them what we do all summer long in a recorded format so that they can experience it when they get back to their school in the fall. It also lets us bring in a lot of different guest speakers, which I've already talked about a little bit. It is very exciting. We're going to have other fully virtual events this year, and the first of those is our Forensic Science Escape Room. That will be starting on January 25th. It will be fully virtual, and we're going to do a module-based multi-day event, which means that each step of our mystery will be located in a module, and once you complete all the steps of that module, you'll get a password to unlock the next one. This will include lots of different interactive videos and many hands-on forensic lab activities. We can drop off all the supplies at the school, and then everything can be completed online at the teacher's convenience and at the student's convenience. We'll also be having our hackathon event starting on March 1st and our college and career fair on April 12th. Both of those will also be fully virtual. All right, I wanted to thank the entire RSC team for their help with this. They have been really involved. Everyone is helping create lessons. Everyone will be part of the Skype with the Scientist sessions. So thank you so much to all of them. And of course, thank you to our members, to our teachers and our researchers. Teachers, we admire everything you are doing during these challenging times, and we're so excited to get to work with you. And our researchers, thank you for helping us out and allowing us to use your expertise with our students. And of course, thank you to Eric Dye at ECD Photography. He has been essential in helping us plan our website as well as all these different videos that we are making. All right, and then these are my last couple slides here. This is all part of the template that we have for our PowerPoint lessons that we're using in the initiative. Does anybody have any questions?
Hi, Sarah. We do have a question. There was a question as to whether you will still be offering the virtual programming in next year or in the years to come, if in fact we can get together in large groups again, or will you be just not offering virtual after we're able to meet in person again?
I think that we will definitely continue to offer our virtual option. We're really excited about this. It actually lets us reach more people than if we were to just have our in-person. So I think that even when we can go back to visiting schools in person and having large group gatherings, we will still offer this virtual side of education. So we'll be able to have both.
Okay, Sarah, we have another question that has come in. It says, how can non-members get more involved? And how can researchers participate if they want to share what they're working on with students?
Researchers, totally contact me or Jeanette. My email is sarah@regsciconsort.com. Jeanette is jeanette@regsciconsort.com. We would love to hear anything that you want to help us get involved in, anything you want to help do with the students. We can definitely organize that with you. If you are not a member and you want to access this, if you can go to our website, you can go all the way to the education page where I showed you. Right underneath our members line, you see the non-members. I have information there. You can reach the stormwater runoff lesson. And then you can also go to that Become a Member webpage and get more information on that and how you can maybe become a member of the RSC and get access to the rest of the lessons.
Okay, thank you, Sarah. We're going to break for about a minute or so as we set up for the next presenter. Hi there, welcome back. With us right now, we have Kristen Currier from the Erie County Conservation District, and she is going to be presenting how to connect with the community without large group activities. All right, Kristen, whenever you're ready.
Great, thank you very much. As Jen said, my name is Kristen Currier. I'm with the Erie County Conservation District. I'm their environmental educator. And I'd like to give a couple disclosures before we really get into the discussion. One, if you haven't met me, I tend to be a really outgoing, lively person that likes to really engage with people. And one of the things I've had a lot of trouble with since March is not being able to feed off of that group atmosphere. So going into this forum of working with people but not actually being with the people has led me to a second part. It's really hard to read the room when you're not with people and seeing face to face. So anybody out there who's a teacher or has done teaching before, one of the main aspects of working with a group of people is being able to read the room and to look at body position and facial expressions and really gauge where people are at. So with this format, it really changes the game a little bit, trying to figure out how to get your concept across to a group of people when you can't read the room the way you're used to. So thank you for working with me on this, and let's continue looking into how we can do outdoor and environmental education without actually being with them in person. And this is something that I have been working with since March 13th. So let's take a look here at how we can do that. There we go. So as I had mentioned, I'm an environmental educator and a park manager. I do a lot of informal programming, things like scavenger hunts, identification walks, geocaching. I don't necessarily need to be there in person for that sort of connection with folks. I also do the formal programming though. So I do work with scouts and schools and other groups that schedule programs face-to-face with me. In addition to the conservation district portion, I also maintain Headwaters Park throughout the year. And a lot of that means actual maintenance where I make sure that it's safe for the public and that it's something that they can enjoy for all ages. However, it also means that I need to expand and keep it fluid enough to interest people, but at the same time, static enough that it is safe and we're still respecting the preservation and conservation of the natural resources. It's a balance that we're constantly working through. In addition to being an educator and a park manager, I also write grants because somehow all of those activities have to be funded. So those three things are the main things that I do as an educator and a park manager. How does that play into maybe working remotely with folks? The pandemic impact really brought to light vast differences in education. And when I put this presentation together, I immediately pulled up these photos. And my first thought was, holy cow, look how close those kids' faces are to each other. And a year ago, we wouldn't have thought anything of it. The girls on the left are closely inspecting the red wigglers that we have in our vermicomposter. The kids on the right were making bird feeders. We didn't think anything of it. Taking these lessons that we do day to day, week to week, had to be altered. How do we do that? How do we continue our outreach and education when I can't necessarily be in the same room with them? Field trips also changed. I have, through the past couple months, in talking with different school districts, discovered that field trips aren't an option this year. And that's one of the things when you do outdoor and environmental education, you rely on the experience aspect of it. You rely on being able to put something tactile in their hands or having a certain scent that they can experience or the temperature of the water. All of those things are experiences that create memories and knowledge, and that aspect has been taken out of the education. So by nature of both informal and formal programming, how do we make this work? And we have gone forward. So one of the things that we've been working on, as I had mentioned, I also write grants, and we had been working on a large grant regarding Headwaters Park. So in March, April, and May, all of my field trips, those are my big times, all of my field trips were unable to happen. And so I'm thinking to myself, people can still come outside. They can still go in the park. How can I reach out to them? How can I give them experiences or create opportunities for experiences out in our park. Fortunately, one way was to create our nature play area in our community garden. And we've been working on that all summer. You can see in the center photo, we have a stump jump and we have our boulder crawl, and we also have the tunnel with the mound over top of it. By next summer, that will have all grown in and we'll be able to plant our community garden and it will be up and running. And from the feedback we've gotten from the public, they've been watching all year to see how this is growing and what's going to be happening. And that in and of itself, just engaging with the public via emails and social media messages, what's going to happen, what does this going to mean? That in itself was a form of outreach, educating them on why we're doing things the way we are and what it means for them in the future, in the weeks and the months to come. We were also able to expand our trail system. And this was very exciting in the sense that while I might not be able to take the kids to the end of the overlook in our wetlands right now, I will in the future, but I'm able to create some opportunities and outreach for people informally. So I might be able to put up something on social media, invite folks to come out and experience it that way. So they're still on their own timeline, but they're still able to gain a little bit of knowledge and some new experiences in a new habitat. So I was able to work in Headwaters Park in that way. The scavenger hunts have actually grown in our park over the past several years. Lauren Sherman, an intern from a few years ago, started the program and it has only grown from her efforts. And it serves a dual purpose of getting people outside, which is also one of our main focuses, but also learning a little bit about their surroundings and finding that balance between making the experience fun and challenging but not too difficult, also over an entire age group, because we have folks that bring infants all the way up through senior citizens. That's actually been really fun. And we focus them month to month. Each month has a theme. We have done clouds, we have done bats, we have done migration animals. In the beginning of the summer and in previous years, we used little red boxes and we would put them out in the park and have a map. And you could pick up a map at the beginning of the trailhead and find the boxes. Inside the box was a little stamp that you could stamp your map, and then a laminated card inside where you could read and get a fun fact or two about the theme. It was great for kids, especially the younger kids. They loved that visual of finding the red box. It wasn't great for vandals, which we found this summer. So we've tried a few alternatives. And currently we have up in our park a QR code scavenger hunt. The only problem we're finding with that is that you need to have a smartphone. So again, we're back to the balance, and I talk with kids quite a bit about this. We absolutely want to engage people, but we want to have the balance of maybe less technology when we're in the park. So the QR codes seem to be working. They're staying on the trees. They don't seem to be walking away the same way the red boxes did. However, we may be giving up a little bit of the interaction as far as adding the QR codes. On the flip side, using the QR codes, you can immediately go right to the website. You can listen to the bird calls, you can see the visuals, and it can lead you to further links. So we may be trading one thing for another by switching up our scavenger hunt. And I've also noticed that there have been other scavenger hunts in other parks, or you could even create one around your home. When it was quite cold before we really got into the summer season, I created a weekly bingo game and I simply put it up on social media. There were no rewards or prizes or an incentive aside from an internal incentive to just go outside and try something new. And since things change so quickly week to week here in the spring, I simply went with things that you would see every week in the spring. We had a lot of great feedback from that. So I'm hoping by just adding those things to poison ivy, multiflora rose, a wasp, if it interested someone that was doing the bingo game, they might go and maybe spend 5 minutes Googling it and find a little bit more information. If you do happen to have your smartphone on you, you could actually scan the QR code on the right and that will bring up, I believe, the dark-eyed junco, which is out in one of the QR codes in our park right now. Very easy to create a QR code scavenger hunt. There are numerous free websites that you can use. It'll pull up and link to pictures. It'll pull up and link to a PDF if you create one. And it'll also link to different websites where we can actually pull up an audio of the junco on this. So that actually has been a really nice interactive way of bringing it to the folks as they come out on their own time. A larger project that was county-wide that we tried this summer that we had actually a surprising amount of success with, we weren't sure what was going to happen when we tried it in July, was Erie County Park Bingo. And this was a coordinated effort between 9 parks across the county to actually have people explore new parks. There was a lot of free time and people were looking for something to do that was outside. So we challenged folks to take a friend, take a pet with them, and visit these 9 parks, take a selfie of themselves, and then submit the 9 photos, and their name would be put in a raffle for over $200 worth of local gift cards. Truth be told, I think a lot of people would have done it even without the incentive of the raffle, but we were happy to offer that also because then it ended up supporting a lot of local businesses. So this was the information that we put out on social media, also on our website. Not only did we put it out at the Conservation District and at Headwaters Park, but the other 8 parks were able to put it out on their social media, and it spread quite a bit that way, and with people sharing with each other. Ruth Spice was the winner of the raffle this year. She and her dog Charlie, you can see, is on the left side. They were able to visit all parks over the 3-month period, and some other friends were able to bring their entire families out. That probably was a really fun part of this project, was to see how many people engaged, but also the fun that folks were having as they were looking at new parks. And I received a lot of feedback expressing that they had never considered going to this part of the county before, and they didn't know this park existed before. So one of our goals, getting people to go out and try something new in an outdoor environment, actually was very successful. We were very pleased to see that. Just a couple weeks ago, we did have our Halloween rock walk, and that was with a park supporter, Melissa Hall, had suggested, what if we took various rocks, painted them like Rock Out Erie, and just put them out there like you would with trick-or-treating, since we didn't know exactly how Halloween would be this year. We ended up having over 300 rocks decorated and donated, placed them out in the park, and it was such a success, both at Headwaters Park and at Scott Park, that we're considering doing an Easter rock walk and perhaps some other educational rock walks. The nice thing about doing rock walks are that the rocks, if they go somewhere, that's okay. And if you keep the rocks just with Sharpies or acrylic paint and a top coat, there really isn't too much of a concern about injuring the wildlife or harming the environment in any way. So it really was a really nice, easy, quick way to reach out to the community, get them engaged in an activity, and bring them outside to hopefully further their curiosity to more activities. And there's a couple of pictures of some of the rocks that were decorated. And we did have a great amount of donations from both Scouts and other groups. We have been able to engage in some small formal programming with masks and social distancing. A bonus of it being outdoor education is that it's outdoors. So with some extra thought involved, we have been able to bring a little bit of formal programming back. We don't have indoor access at our facility right now, but fortunately the weather has been helpful. So we've been able to continue with small groups outside, doing a little bit of water quality, some exploration there. So one of the things that we've been experimenting with are videos, and I'm sure all of you have experience with either creating the videos or being on the receiving end of watching videos from professors and teachers and other agencies. For some people, it's great. For others like me, it's a big learning curve, used to being face-to-face. So because I am unable to go into classrooms right now, and classrooms are unable to take field trips out to my facility, I'm putting together some classroom kits with our 8 biggest, most well-received topics and sending those out to the classroom, either pick up by the teacher or dropping off in the lobby. And then we'll do a Zoom meeting. The kits will be disposable, or I'll be able to disinfect them, and hopefully we'll be able to rotate them through the schools and give them at least some sort of tactile visual experience and be able to do some questions and answers with the students. And you can see. So when we talk about animals,
This is
there's
my
a lot of
experiment
different
with the
things about
video.
animals that we can discuss. We can talk about their differences in ears, their differences in their... Putting this all together, what does it mean? So far, what I've discovered is that we're able to help keep safety in the public. We're able to work hard at making sure that everybody is able to engage on some level, but engage safely. It is a really new way and a nice way to engage with the younger generations because of their comfort level with technology and being able to move fluidly between people and technology. We're able to reach out without physically being there. And as you just saw with that quick video, I can show visuals on demand that I can't necessarily do in person, and that's nice. Some downsides of remote is that we're still missing that personal connection. It does limit some of our group abilities and capabilities. We often play group games and we do group challenges, and when we can't be face-to-face, that's tricky. The other part of the technological aspect is sometimes folks don't have the connection. They don't have the connectivity or the access that's necessary to work remotely. So putting those two together, I'm actually looking forward to the future of outdoor and environmental education, because once we're able to incorporate both, I think we'll actually be able to expand our capabilities with outdoor and environmental education, especially in areas like Erie, where the weather can sometimes really inhibit what we can do outside safely. But then again, if we have those connections with the technology and internet access, and I'm not able to physically be there, I can still reach out remotely and connect with some people. So I do look forward to the future when we're able to put both of these together. Does anybody have any questions?
Kristen, that was a really interesting presentation. We currently do not have any questions for you. Except I did want to ask, is that your rat?
Yeah, I have two of them. That was one of them.
Okay.
I thought about bringing him out, but I know he wouldn't hold still.
We will be, oh, go ahead. Okay, so we will be moving on to our next presentation. That is being presented by Ann DeSaro and Ray Beer. They are both with the Pennsylvania DCNR, and they are presenting Presque Isle State Park COVID-19 teaching strategies.
Okay, hello everyone. My name is Ann DeSaro, and I'm the Environmental Education Specialist Supervisor here at Presque Isle State Park. And I am co-presenting with Ray Bierbauer, who will be on the presentation in a little bit. I would like to tell you a little bit about Presque Isle State Park. It is actually a complex state park. We encompass Presque Isle State Park, or the peninsula, as many people know it, the Tom Ridge Environmental Center at the top of the hill before the entrance of Presque Isle, and Erie Bluff State Park, which is 13 miles west of Presque Isle on Route 5. And so those are three very different places. We host programming at all of those sites. We have a staff of 6 educators on our team. There's 3 full-time educators that work year-round and 3 seasonal educators. 2 of those are environmental educators, which work during the school year, helping with school programming. And one of them is an interpreter that works in the summer season, doing mostly interpretive programming, but they do help us with school programming as well if needed in a very limited way. We also have interns that usually work with us during the summer season, and a large group of volunteers that help us in all aspects of the programming work that we do. In normal times, we serve about 4,000 to 6,000 students coming to Presque Isle or the Tom Ridge Environmental Center, or very, very rarely Erie Bluffs, almost never there due to lack of facilities. It's a very primitive park. And working with our educators one-on-one, either doing a program or seeing a movie, visiting the trek, doing a scavenger hunt. So a lot of students visit us, and most of those students come from the tri-state area, between Cleveland, Buffalo, and Pittsburgh area. Some schools have a significant drive to travel here, but so many of the schools in the local area as well. Little bit about Pennsylvania State Parks. We work for the Department of Conservation and Natural Resources, the Bureau of State Parks. And I wanted to share this because front and center, the very first sentence of our mission indicates that the parks are to serve as outdoor classrooms for environmental education. So across the state, you will see environmental education specialists working in the state parks, teaching people of all ages, really, school children, the general public. Also important concepts in that mission is the preservation. The first consideration should be the preservation and conservation of our natural, scenic, aesthetic, and historical resources, and stewardship for the enjoyment of current and future generations. So the education team takes that mission very seriously, and we try to cue our programs that we do to reflect that mission. We have received guidance from our organization when we returned to work. We had about 2 months of working from home, and we returned to the office in approximately mid-May, and there was a lot of guidance that we were operating under and that we are still operating under. We must be masked at all times when working with any kind of educational programming. Everybody must keep a social distance of 6 feet or more unless they live in the same household. There is hand washing, sanitizing, increased cleaning of all of our facilities. Of course, people must be symptom-free to participate. There is no outreach or no guests currently. So in normal times, we visit schools and do lessons in the classroom. That is stopped for now, and we also cannot bring guests in that are maybe specialists in their area to present. So that is at a halt right now as well. We are doing increasing sanitizing of the equipment that we use for programming, and we're really trying not to use a lot of equipment. There's certainly no shared equipment permitted when we're working with students. Each student, if they are here live, or each person coming to an interpretive program, is going to get their own piece of equipment to use during the program that is sanitized pre and post the lesson. And then there are pretty strong limits to our indoor spaces and outdoor spaces. Our room in the Tom Ridge Center that holds 100 is now limited to 20% or 20 people. Similar limits are to the outdoor spaces that we can work with. And also the challenge of if people are keeping a 6-foot distance, you really have to project your voice over a broad area to speak to them if you are doing outdoor programs. So most of our programming has been virtual. And there are certainly pros and cons, as the previous presenter mentioned. Some of the positives, we can reach anyone. And there have even been some people from out of town and across the state that have been able to participate in our virtual programs. It's free. In the past, we charged $3 a person for our programming. And so because we also were learning how to do virtual programming, we decided not to charge for our programs. The students can get a little closer look with virtual programs. They can see things easier than in the field, and Ray is going to show you that in a minute or two. We can work with larger groups. In normal times, we only work with 10 to 18 students per educator. Now we can have 2 educators work with 100 students or more without any loss in quality. Smaller carbon footprint, and this is because of the busing, of course. Many students traveled anywhere from a half an hour to an hour and a half to get to the park, driving to the locations of their programs, so a lot less bus traffic. Maybe more appealing to the younger generations, as was stated previously. They're already used to doing everything online, easier to attend. You don't have to leave your house or your classroom. Maybe there are challenges that not everybody has the same kind of connectivity. Weather doesn't matter is a big factor too. A lot of times field trips can be ruined if it's a bad stormy day or radically changed. So we can go forward regardless of the weather. It does require us to look closely and decide if the program is effective, meaningful, and worth the time and effort. And that was a directive that was given to us. And so it really allowed us to step back and choose what our most meaningful programs are, to be able to really develop those for the virtual experience and offer those. So that's what we did. Some of the negatives, technology, of course. We have faced just about every challenge that you can with every piece of equipment, from cameras to microphones to computers to internet. And we have muddled through. I must say, the teachers and the students, tremendously patient and willing to work with us. At one point, when my team was trying to get a microphone to work, the teacher had the students get up and kind of do some exercises around the classroom to keep them from getting bored. So I think the teachers are on board with being flexible as we all learn. System limits, we had some limits in our structure. We just recently were permitted to use some of the Google apps. Our agency only permits us to use Skype and Microsoft Teams at this time. So there's many different types of ways to do presentations, and we're still learning all the different systems. The biggest thing is the loss of the outdoor hands-on experience. When you're working with a naturalist or an environmental educator, that's very powerful to students. And we try to replicate that virtually, but still you are not outside with an educator. And so that is something that's being lost. Also, it's very hard for students to get the sense of place. Presque Isle is a really special place, a unique place. And it's kind of very hard to translate that in the virtual world, especially since we don't currently have the technology capability to be broadcasting from the field, the beach or the woods or places like that. But we are overcoming a lot of the challenges, and we're making it work. And now I'm going to turn it over to Ray Bierbauer, who is going to show you some of the strategies we're using live.
Okay, thank you, Ann. So yeah, I'm Ray Bierbauer, one of the environmental educators here, and I'm gonna share with you guys our classroom that we set up to do this. So like Ann was saying, when we came back about mid-May, we were doing other things, but then as we got into the school season about ready to start, we needed to get some lessons and stuff to still connect with the students and everything, because obviously they were not gonna come out here. So we picked Three of our most popular lessons, a pond's life, lake dynamics, and then shifting sands. And we've kind of really focused on those lessons and got components of those ready so we can work with anywhere from K through college age students and try to bring that park experience to them. Ideally here in our classroom where we're set up, we kind of have three people is kind of the best way I think for us to work. One's running the controls and doing all the behind the scenes stuff. And then the other ones are the ones doing the presentation itself throughout. And as Ann mentioned, with the different formats that you use to present, it's kind of challenging what you can do on some versus others. So using Zoom, we are able to bounce from our webcam on the computer to a PowerPoint. And then we also set up a second camera here that I will show you our classroom in a moment and also highlight things up close as well as far away. So with that, I'm going to hook up our camera here. Get that squared away, and I'm going to share with you what our classroom setup looks like. Okay, let me see. Okay, you're going to switch cameras. Alrighty, so what you're seeing now is our classroom here. As Ann mentioned, our building, we're not using any of our classrooms, so we're able to make this more of a permanent setup for us. So we brought in all of our stuff that we use for most of these lessons. And again, we kind of, depending on what we're teaching that day, we can kind of clean out. All the other stuff's hidden behind our gray wall, and then we can bring it forward to set up for our next lesson and work in here. So ideally, we have about 3 different stations in here. We got our up front there. We got our table there. You can see with our beaver on it. And with our camera and stuff, we can really zoom in nicely and do it nice and smooth. And again, somebody else would usually be here helping out with this. So as we're talking about something, one of the other educators to me are talking about the beaver skull, holding that skull up close and talking about the teeth and everything, and really bringing that experience right to them with some pretty good detail as well. And so we would focus on that. Depending on our lessons, we got our aquarium in here for live fish. The bucket below there has macros, which I will get to in a moment. So we have our aquaponics lab, and I am able to go back there and bring in some of our fish, native and non-native, and have them in here for up close viewing as well. We can take them out of our aquarium, put them in a little tub close by here, and kind of the same way I'm going to show you the macros in a moment is how we've shown our fish. And then it's also given us an opportunity to use some more of our collections materials here at the Tom Ridge Center. So down in our basement, we have all of our specimens, plants and animals, the jarred specimens. So again, most of our lessons are evolving around the water. So we got a lot of our water critters here, some fish, we got some frogs, there's a sea lamprey there, turtle, things like that. And we also will use the live turtles that we have here at the Tom Ridge Center for part of these lessons too, to discuss that. So it really helps to engage the students. When we're doing our presentations, typically we're bouncing back from like seeing me on the web camera, PowerPoint, and then we cut to the USB, the other field camera here. So things are constantly moving. You're not just staring at one thing the whole time. So the engagement's been great with that sense. As Ann was saying, with some of our groups, we've had as few as like 4 students in a group or as many as 40 at one time. The small groups I really like because we can do some Q&A during the presentation. With the larger groups, we kind of limit that towards the end. So we can just keep things flowing smoothly as well, especially it's challenging when the students are at home and some at school and everything. So you've got a lot of different feedback. If teachers are a little more able to control the students' questioning and stuff, that makes it a lot nicer for us. So I'm going to move around here and set up and show you how we do our macros now. So give me a moment. Okay. So when we're collecting our stuff, we'll have our little macro tray here. So we show the students what it looks like, what they're going to be looking at. So this would be our pond sample, plants and stuff. We would dig through there, hopefully kind of stage some of the bigger bugs that we found in the sample. And then as we find them, we can take them, put them in our little guy moving around in there. And then try to make it a little bit easier for them to see. I can then show them our card with what the actual critter looks like, drawn up a little bit bigger and everything. This is where we would share some of our more information. This guy's a decomposer, shredding up the leaves and everything related to the pill bug that you would find under a rock or a log as well. So, and then here's the second one, one of the more common guys that we find. We're going to cooperate. He doesn't want to. Oh, there we go. So, and then we got our dragonfly nymph climbing around in there. And same thing, once we show them that, we can go back to our card. And this has been something that the teachers have really enjoyed us doing, is having these specimens, having some of the live stuff, and then going to the actual drawing and stuff like that. It is a little difficult to see them, and oftentimes they don't cooperate. And we're still learning with the proper lighting and all that stuff as well. Okay. So we're still playing around with some of the different techniques that we have here. We try to get it as interactive as we can. Like I said, we're showing and demoing many of the things. And some of our future plans are to try to get some little kits together, especially with our shifting sands lesson, to send to the schools, to kind of have some of the things that they can actually try there themselves within the district. That's all I have at this end. So if there's any questions.
Hi, Ray. We do have a question, and it's of a technical nature.
Okay.
The question is, it says impressive camera use. I don't suppose you're able to share the list of equipment you use.
I can get that to one of you guys, I guess, to share it out. Would that work?
We could also, I can share back our emails and teachers could contact us directly. We'd be happy to share.
We still have some stuff that it will be coming here to enhance our program. So like a lot of this stuff we've pieced together through our personal collections right now, but we do have an order of materials, cameras, microphones, lights, and stuff like that. But as with everything, a lot of that's on back order right now, given the nature of the pandemic. So.
Okay, thank you. Thank you for posting your contact information. That's wonderful. Am I muted? Okay. Hi, everyone. We have our next speaker coming up. It's Brian Bookhammer from the Fairview School District. And his topic is STEM and science during hybrid learning.
All right, you can see that PowerPoint pretty good?
Yep, you're good.
All right, great. Well, hello everyone. My name is Ryan Bookhammer. I'm the STEM and Gifted Administrator at the Fairview School District. And so my job is really to work on developing STEM and gifted education throughout the district. That's K to 12. And the majority of my work in the STEM areas is focusing on different science curriculums, engineering, new technologies with computer programming, and trying to integrate that into regular education curriculums where we can really work with our students to develop their problem solving skills in a number of different areas within skill-based development. Well, it didn't show up. How about, no. Let me see if this will go forward. No. I guess I'll present it on here because that does not seem like it's allowing me to present. Does that seem right? All right. Well, I'll present it in this fashion then. So within STEM, what we talk about is, STEM is the acronym for science, technology, engineering, and math. And really our main concepts is that we want to focus on interdisciplinary learning, integrated learning, and project-based learning. So within those three areas, we're talking about cross-curriculum learning where the application of STEM is directly integrated into curriculums. And there's an exploration and an investigative learning process that takes place with our students. And that's really critical as far as we're talking about, wanting to have hands-on projects, but also projects that cross curriculum and go across from ELA to math to sciences, where we're pulling in different functions of those curriculums. And so we're allowing instructional models that really reach students and engage students at different levels. So we want to talk about that as, within our hybrid learning and what our models are right now currently amongst the school districts is that, some of our students are learning at home, some are learning online, some are learning, half in school and half online on opposite days. So how can we create, projects and activities that allow our, our students to have that experience at home as well as when we're in the classroom? And so one of the first things we think about when we're talking about STEM science is just what's the importance of STEM. And sometimes people might think that it's an overused term or it's a term that's maybe vague. And so one of the things that we talk about with our STEM education is developing career-based skills that we look at for long-term development. We're talking about research skills, analysis skills, even self-learning skills where students can understand how to explore and investigate problems and become what I call innovative problem solvers. And so we want to engage our students in science-based curriculum and science-based learning and give students the opportunities to design, test, analyze projects and activities where they can actually understand not only just the basic concepts, but how those concepts are applied within curriculums and within, exploration of different ideas in different school curriculum areas. So developing those cross-curriculum skills and aligning the in-school instruction also with the out-of-school exploration. And so we do that in a number of ways. And so what I want to share today is just a quick framework to how we do that for teachers and administrators, just to kind of see how we do that amongst our STEM programming. The first thing is it should be curriculum, should be aligned to standards. So that's a very basic thing, and all curriculums should be aligned to those state standards that we have, whether they're the science-based standards, the ELA or the mathematics standards. And then we want to know the map of our curriculum. So really to develop projects that are influential within the curriculums, we need to know what are in those actual curriculum areas. And a lot of times, the sciences, as you get down into maybe the younger grade levels, the K to 2 areas, they're maybe not as clear cut as when you get to the 3rd, 4th, and upwards, where the standards really drive all those curriculum areas. So we want to know, how do you map out and what is the mapping out of your curriculum so that you can identify areas that you really want to work within. And so that third thing there is just identifying the content areas or the content areas that you're going to develop the projects and the interdisciplinary learning within. The fourth one there is just collaborating as teams. So really one of the highlights of probably developing any STEM and science-based projects that reach cross-curriculum levels is that you're collaborating with your grade level teams. You're collaborating with, you know, your ELA, your math teachers, your science teachers at those grade levels to really look at what are the overarching concepts that we could use throughout the grade level to integrate something that would be impactful for the students, that would utilize the skills from each of those different content areas, and then that we can integrate that STEM or science-based learning into the ELA, the math, and also driven by the science curriculums. As far as the planning for success, this is one of the things that we focus on, is determining how the instruction is delivered. And with the hybrid learning, that has certainly changed. You know, we have part of our instruction on online. We have part of our instruction in the classroom with the teachers. And so really, you know, how is that delivered effectively? And so some of our programs that we use, like Schoology and Seesaw for the younger grade levels, those are really the tools that we use through our development of our instruction to deliver that content out effectively. So the resources, the materials the students need, and also the outline of how that instruction is going to be delivered to them. We want to identify and gather the resources. So we know that, you know, everybody's at a different level of provision of resources within different districts, but identifying and understanding what resources you have. Also in the areas of STEM, you know, we don't always need a 3D printer or some huge technology to do basic research, and also just utilize the things that we can find outdoors as part of those science curriculums. And then what needs to be built or what needs to be bought is really just how we gather those resources. The next thing is just understanding how the students will learn, submit, and display their knowledge. One of the things that we're working on with the younger students is they might not always be able to relate, you know, in sentence form or in paragraph form, how they can, you know, demonstrate their knowledge. So we're doing a lot of work in Seesaw where students will submit pictures, they'll submit drawings, they'll submit, you know, words and sentences, but also video clips of them expressing their information about how they're analyzing and how they're viewing these different concepts of science. And so we're trying to utilize various forms of submissions to be able to really allow our students to engage and talk about and discover their information that they're analyzing with these projects, but without just submitting it in only one form, right? So we're trying to really display that knowledge, and then allowing our students to engage in their own experience. So how can we make these projects more personalized? How can we make them, you know, learning experiences that they engage with at their own level, and provide students with a pathway for investigating their learning? The way that we do that is really an engineering design process, and there are 1,000 of them out there. NASA has their own. A lot of the big universities have their own engineering design process. This is the one that we've developed at the Fairview School District. And it really comes down to identify, explore, evaluate, and communicate. And so within STEM-based projects, whether they're based in ELA or science or mathematics, we want to do these different things. We want to identify the problems. We want to know the criteria, the research, the constraints. We want to explore, which means brainstorming, researching, identifying other content areas that you correlate to that. We want to come up with ideas, obviously, in that exploration stage, and then evaluate those. So build some sort of physical model or some sort of abstract that we can then, you know, analyze and test. And so that student goes through that process of designing and redesigning and reiterating those designs, which really allows them to kind of grow as a learner through that design and engineering process. And then the last thing, which is really one of the important aspects of it all, is communicating that idea. So how do you articulate your ideas as a student? How do you articulate your ideas in formulating your solutions? And so that becomes a really intricate part of the design processes, you know, sharing out your ideas. And so even as our youngest learners learn in the sciences, one of the science aspects of being a scientist is that they communicate their solutions. They report on what they're doing in their projects. And so that's something that we really emphasize in our science and STEM development. So one of the things that we really focus on, and I'm going to talk more to the K-4 projects that we do throughout the district, is just providing all students access with the materials, opportunities, and exploration and learning. Usually when we build some of these projects out, I build, you know, hundreds of kits. I'm thankful that other people in the district help me, and we build those kits out to make sure that every student has the opportunity to work on those projects and that those projects are available, that we provide the resources, whether they're simple things from paints to color pencils, the whole way up to, last year we did a project in the spring where students were studying biomes around the world in first grade, and we gave every single student an inflatable globe, which I thought was just amazing because, a globe is probably not something that we see in every household nowadays. But it was just really interesting and neat that, it was something that every student could go and have a globe to look around the world and then identifying where different biomes were located throughout the world. And then they took those and they built a biome shell, and then they turned that into this biome shell turns into a biome nightlight. And then they do research on the different biomes, and just a really interesting way to engage students, also to provide them with some resources that they might not have, but really giving them resources that expand beyond just maybe the science concept, but go into geography and other aspects of their community. On the right-hand side, there's a picture down on the bottom, and that's just a bird adaptation project that we work on in 4th grade. We've developed molds and injection molded different bird shapes and designs, and our students do different study and research on the birds within our region. Then they paint and develop their own birds. We talk about some of the aspects of how those birds are developed in injection molding to get some of the technology and engineering aspects into those. And so really providing those students the opportunity to learn about multiple subject areas and about how these parts are actually produced and built. And so with the K1-2 grade levels, we talk about, being a science explorer. Each of the grade levels do a wonderful job of having different implementation of their STEM science projects. And one of the things that we want to continue to develop is, how do we integrate that into, some of the other areas of the ELA and the mathematics, and bringing those into those other subject areas. This STEM kit on the one side here is just the 5 senses. The students really investigate those different 5 senses and talk about what it is to be a scientist. And so they have some different little tools to be able to do that, and each kid gets their own little science kit. On the right-hand side, this is one that I worked and talked with the Regional Science Consortium about, was a shipwreck project where we 3D printed a number of different ships, and I'll just show you here. We 3D printed ship parts that snapped together as a shipwreck, and then we used the resources from the Regional Science Consortium website. We talked about a community map and where those shipwrecks were in the community, and then what components of the community there are. So really used a shipwreck as a starting point to talk about, our community and things throughout our community, as, different components of, our students investigating, aspects of our community. We also turned it into landforms. So you can see on that community map, we talked about a peninsula, and there's a volcano up there, and then islands. And so we talked about the different landforms that you can find throughout, the world. And so we turned that into our community map landform project, which really then integrated a lot of different subject areas with our students. We also let them personalize it by building their own sails for the ship. So then each student got to develop their own sails that they put on their ship. And so really allowing our students to get involved, to personalize their project, and really learn a number of different areas around the different concepts that we're trying to focus on and learn within the projects. Then there was this anchor that we 3D printed and molded, and that was a key that actually would be used, that students would identify different words, and they'd identify the letters in the words, and then it made a code word at the end. And so they had to identify the different landforms and the shipwreck and some of the parts of the community, and then it led to a decoder word. And so that was one of the parts of the lesson, was something that integrated, those aspects of the different concepts we were talking about, but then engaged them in some movable activity. The next thing here is, just how we're really trying to make sure that we develop these projects, is that we're aligning them with standards. We're aligning them to the curriculum. So even if it's aligning to something in ELA, we're identifying content and areas to develop the projects and develop this interdisciplinary learning. We're collaborating as grade level teams, and I think that's one of the most important things as you get to the middle school and high school grade levels, is if you're going to develop STEM science-based projects that want to cross curriculums, we need to do that as grade level teams. And then one of the things that I always say is, just that we're starting small and we're integrating within curriculums. And so as we look at our curriculum mapping as K to 12, that we can, identify where STEM and science can be integrated into, reading content, into writing content, that it doesn't have to be an overarching, everything has to be, just huge projects, but also just that it could be smaller components of things that we find inside of our current curriculums where we can build engaging activities for our students. Things where our students can really be engaged in building some of the content, in personalizing the content, and then really using themselves as, the scientists, where they're exploring, they're researching, they're developing hypothesis, they're understanding how those concepts interact and apply to real world environments. And so that's kind of the concept that we do with building STEM and science projects throughout our curriculums. And so that's just one of the things I wanted to share. And if people are ever needing just assistance with, looking at, where you can integrate things throughout the younger grade levels or the middle school or high school grade levels, please feel free to let me know. And I'm always available to help or assist people with that. So if there's any questions, feel free to please shout them out to me. All right, I'm gonna stop sharing. All right.
Ryan, we do have a question for
Yep.
you.
Sure.
And the question is, are you selecting specific students to participate in this program, or is it based on grade level? So how are you determining who is, kind of going into this this curriculum or this type of learning?
Yeah, so we develop this as whole grade level. We don't in the younger grade level. So we're talking when we're doing projects K to 4, and even if we're doing projects in 6th grade, we have a number of projects that we work in. Those are whole grade level projects. So when we're building a set of kits for the shipwreck or science exploration or some other component, I mean, we do ones with bird adaptations, we do magnetic levitation vehicles. When we do a lot of these projects, they're whole grade level, especially in the K to 4, 6th grade projects. As we get to the high school, we have a STEM academy. And so that's a very different type of curriculum aspect. But when we're talking the younger grade levels, it's a whole grade level to make sure that all the students are being exposed to, those concepts and also getting the engagement interaction within the curriculums.
Okay. Wonderful. Thank you very much, Ryan. We appreciate your presentation.
Yeah.
And we are going to move on to our next speaker. His name is Drew Mortensen, and he is the assistant principal at Harbor Creek Senior High. He will be presenting on the impact of COVID-19 to education.
All right, thank you so much. I think that we're all set here and ready to go. Just do a quick mic test, make sure that everybody out there can hear me. I know that there's quite a few people out on YouTube watching, and I appreciate that. You guys have been great sticking around. I've been watching the numbers fluctuating throughout the day here, and it's great to see so many people that are sticking around and participating in each and every session. So thank you for doing that. All right, so I will just jump right in then. I'm going to assume that everything is working properly and that you're seeing my screen here. So the impacts of COVID-19 to education, right? It's a really small topic in the grand scheme of things. All right, thank you, Jennifer. Yeah, really small topic, right? Like we didn't want to bite off too much. We just wanted to tackle all of education and how COVID has impacted it. So nothing too grandiose coming to you in the next few slides. So here's what we've got. Really, the biggest impact to education over the past 6-7 months or so has been that it has forced education to essentially rethink itself, right? You know, education has a propensity to move, to adjust, to change at a glacial pace. And so a lot of different things that can happen at different times in education can often be significantly lagging behind the rest of the world. And there's a lot of really good reasons for this. And then there's a lot of reasons why the rapidity should and hopefully will increase over time. But one of the major things that's happening now is that we're seeing not just administrators and teachers and the random person that is inclined towards a particular new idea embrace, but what we're seeing is that everyone, wholly and systemically, we are reevaluating and rethinking what education looks like and how it works, and what it is. And so we're having these giant philosophical conversations in the midst of a really difficult situation. And I don't want to ever undercut the difficulty that is being experienced by so many people around the United States and around the world. But at the same time, I'm also trying to balance that with the fact that we are making some really good and interesting changes and progress in education, and trying to find that silver lining in the midst of this really difficult situation. So one of the things that we've had to do is, and again, we're going to talk about a lot of different things here, but one of the things we've had to do is really rethink how all sorts of different activities that we typically look at as being hands-on function and work in an alternate environment, right? In a remote environment, in a hybrid schedule, in an online format. Things like science labs and 3D printing, and our CAD work that we've spent years, and our computer science work that we've spent years, progressively building up. As Ryan had just shared all about the STEM, activities and things that they've done over the past 8-10 years or so at Fairview. Likewise, here at Harbor Creek, we've been spending a lot of time doing that as well, and just trying to figure out now how this functions in a completely different modality. And a lot of it does have cross applications, and a lot has required some very significant adaptation. But it's more than just the STEM and the science. Simple things like the physical fitness classes. We have a course called team sports, right? And team sports in a classroom environment where you're trying to mitigate distance and you're trying to mitigate, touching of common resources, like, okay, how do we manage working with this football or this soccer ball? And how often does this need to be cleaned? And how often does, all of those questions that come around, some very simple, like using of the weight room, things that seem like they might be simple, but are not at all simple in terms of having students rotating through, especially at the junior and senior high, where, students might move through 7 or 8 different classes in the course of a day. So there's a lot of nuances that are being affected that we've had to really rethink. As I start to bring in the school that I work at, and we've heard from several others now, it's easy to start to think that there is a one size fits all approach. But when in fact, our schools are similar in many instances, but they're also very different, right? A school that's operating on an intensive block schedule has a lot less changes in their rotation in their schedule than a school that has an 8-period day. And the consequences of that are significant, and the ramifications downstream are very significant in terms of things like, how do you do physical education classes? How do you do lunch? How do you do, science and labs and the handling of materials? There's just a lot of nuanced differences that do come to bear as well, that we can't look at this as being any kind of a one size fits all approach, which makes analyzing the system really difficult and very challenging. So in our process of rethinking education, one of the things, again, that we've had to do is we've had to rethink things, but we've had to rethink essentially every single facet of education. We've had to rethink everything from, how we handle food and how we deal with attendance, athletics, cultural events, the high school experience and what that means, and the expectations that communities have for that experience, beyond just the academic. But then the academic is obviously the crux of why we are here and the essence of what we are to be doing. But then there's also the mental health component. There's also a lot of ancillary services that all get factored in. And so really, we truly have had to rethink every single facet, every single type of experience, every single service that we provide, and how that works. So when we're talking about impacts, I mean, there is not an aspect of K-12 education that has not been impacted by this. And I truly mean that. I don't believe that that's an overstatement in any way, shape, or form. And of course, every district is adapting, and every district is adapting differently. There is, different numbers of students that have gone remote. For instance, some schools have last year had, anywhere from 6 to 30 students that were maybe in the remote or online learning experience. And now they might be upwards of 200 to 300 students. A lot of schools, especially in the Erie County area, have percentages of students that went full remote, somewhere in the neighborhood of 10 to 20%. And those numbers, while 10 or 20% might not seem like a drastic number compared to what it could have been, 10% of students going to an online sphere means gigantic changes in terms of staffing and how we handle and how we work with, and the companies or providers that we contract with to provide those the courses and content, or if internally staffs are developing that content themselves. So it's just a really big set of changes. And of course, in through all of this, there's always going to be, driving and restraining forces. There's going to be competing philosophies that are going to be at play, folks that really want to embrace different things and others that do not. And all of those have valid points and valid, ideas behind them. It's just a matter of implementing a system and actually enacting a system when you have so many things that are in flux and just need to be addressed in a very short period of time. So one of the things that we keep coming back to a lot in this COVID world and how we're being impacted is through the use of data, science, and the guidelines. And not that we haven't always used data, science, and guidelines, but even more so now to attempt to take a more objective, a more systematic approach to the way that we're making decisions and the way that we can then communicate those decisions out. And schools, not just in Erie County, but all across the state and across the country and world, are all experiencing this because the constituencies are not homogeneous, right? They are diverse, right? And we need them to. We need everybody as much as possible to have an understanding and have a measure of clear communication both directions so that everybody's on the same page as to what we're doing and why we're doing it. So one of the things that we have to look at in terms of the data is, what metrics are we using? What metrics matter to us? From where do those metrics come? Because not just having data, but understanding where that data is being derived and what that data ultimately, I don't want to say means, because it's data is data, and we oftentimes ascribe the meaning to that, but understanding that data truly and fully. Then of course that philosophical question and application of what should we do with the data once we have it. Back in the summer, there was a Mathematica report that was put out by PDE, by PDE. PDE had commissioned this, this study, this statistical analysis, talking about how schools are likely to see the spread of COVID-19 inside of the schools, and then mitigating factors and what types of things could be done in order to assist schools with remaining open, and how those decisions might affect the propagation and the spread. Now, obviously, everything was new, everything was still very much in flux. There wasn't a lot of data to base things off of at that point, especially because most schools had shut down in March. However, the statistical analysis came out, and schools around the state of Pennsylvania were starting to look at this and saying, hey, wait a second, what do we need to do, and how do we need to adapt and change? And so, as we've heard about hybrid models, we've also heard about online learning models. We've heard about, asynchronous and synchronous, and there's just so many different modalities that can occur, and then how those affect the spread and how those affect the continuity of education that our students are experiencing, right? Because ultimately, that's what we're trying to maximize. We're trying to maximize the continuity of education and the safety of our students and the safety of our communities, so that way we can continue to do what we have been tasked with doing, which is providing a good and robust education to our students. And looking at the models and understanding the modeling of the statistical modeling of the spread of the virus is important. It is something that's completely new and very foreign to many of us, but we've had to learn that. We've had to adapt to it. And then we also start to look at things like the county data, right? And so here's the most recent county data, and we find ourselves looking at the data that's being released by the state on a regular basis. This is released every single Friday, and looking at this past week compared to the week prior, and saying, like, what are the trends, right? And starting to get a real understanding of what are those trends, and what are those trends relative to what we're seeing inside of our buildings. Because what's happening in the county may or may not reflect what's happening in our school. And, and I'll talk about this in just a few minutes, but like there's a need to be very, very analytical, and a need to be very critical in the way that we dissect and process this information, because it's very easy to jump to many different extremes with any piece of information, but finding a clear, concise way and a clear understanding of it is sometimes elusive, especially not with a ton of experience trying to do it with this type of data sets. So, when we're looking at this information here, we're also trying to balance that out with PDE recommendations as far as when do we close, right? And so just thinking about that, right? Just that one aspect of the impact of COVID on education, we're talking about trying to figure out how and when we close. And we're trying to make those decisions ourselves. That's not necessarily coming from the Department of Health. It's not necessarily coming from the county Department of Health, right? It's coming from us making those decisions in conjunction with, but we're having to work alongside and process that information and make informed decisions based off of it. Relative to the recommendations that are put out by PDE. Again, even the word their recommendation is not a fixed, right? It's not telling you have to do this, but it's giving you a suggestion. And, of course, then there's all sorts of things that, lawyers become involved in all of it. And that's always an interesting thing. Anytime there's a lot of voices in any decision-making process. And one small example of that is the CDC guidance on opening windows. I love this, because this was a conversation that we just had recently, which was, how much should the windows be open, right? Like, we should have windows open, right? We know we should have windows open for airflow, but how much should they be open, especially as the temperatures drop? Like, what's the statistical modeling? What's the airflow aerodynamic modeling that shows how many times the air will be refreshed in a room to mitigate and to minimize the amount of spread of the virus in the classroom, should it be present, relative to if the windows are open completely for the whole day, or if they're open for a stretch of 1 hour or 10 minutes every hour. And having those questions be asked is just, it's in many ways mind-boggling, because we're trying to figure out some incredibly deep, incredibly complex problems, but at the same time not having a great support structure for being able to answer those, right? Like, we go to Google And it would be great if Google just had an answer for that, but really like, finding like the Q dot of like all of this, modeling in a classroom in the flow, like it's some really crazy stuff that we're trying to answer as educators. Of course, we've had school closures. There's been plenty already that have happened. There will be more. But again, that's a lot of those are our decisions in relation and in conjunction with the Department of Health, not mandates. And so again, the onus being placed back on the schools to make these decisions is really difficult. This is far and wide above, do we close for a snow day? Right? Like that's been the greatest question that we've had to ask relative to closures in years past. Like, do we close for a snow day? Or it's like, are the roads good enough or bad enough? Like weighing risk and reward and but this is completely different. The scales are radically different and the community expectations are radically different. And so it is not an easy thing on anyone. Not on teachers, not on administrators, not on the community members, parents, students. It's not easy anywhere. And then we get into student quarantines and saying like, okay, we're going to keep the building open, but we're going to individually quarantine, this student or these groups of students because, well, they might have had an exposure or they participated in an event and that one student may have been exposed at home relative to a parent or relative that had that was considered a close contact. So we could be 2 or 3 times removed, but still needing to quarantine out of an abundance of caution. And even making those decisions is not black and white always. And it's not always easy to understand exactly when and where we come down on, on the different sides of having to make those calls. And it's not simple. Of course, the comparative data, a lot of times people want to say, they want to jump right to the gun. Like, is the education that the kids are getting better now, or is that as good now, or is it worse now than what they were receiving in years past, right? And the reality of that is that it's really difficult to answer that question, right? Because we've never seen this before. We've never been through this before. We've never had whole-scale mass tens of thousands, hundreds and hundreds of thousands of students going into these various models of hybrid or synchronous remote or asynchronous. And students that would not ever traditionally have done those things are now doing them. And teachers are now being thrust into this, need to create content for being distributed and consumed online or through video. And it's just a completely different experience. And so there really isn't much data to answer some of the most fundamental questions that we're saying. Is this good? Is this working? Right? Like sometimes we look at failure reports and say, is it working? But then we say, oh, well, but is the standard that we're holding the students to the same that we would have last year? And there's just so much that's just completely up in the air. And ultimately what we keep coming back to, and I keep coming back to, is like, we are going to make it through this and we are going to learn a lot in this process. And our teachers are doing an amazing job and so are the students. And we are so incredibly thankful for our community that has been incredibly supportive of the difficult decisions that we've had to make and the sacrifices that so many have had to make in order to get what's working well. And it's definitely not a simple thing. So the last thing that I really would want to say was that this has been the shift to online and digital and one-to-one. This was inevitable. It's just that we accelerated this process exponentially, right? We were going down this road and teachers were learning these things in terms of being able to transition to being able to have a hybrid modality, but it was going to take 15 years more. And we're doing it now in 6 months, what would have naturally taken the next 15 to 20 years to do. And that's not a bad thing. It's just really difficult. And it's a really stressful experience for everybody involved. So we've learned a lot in the past 36 years from the time that there was the first online learning offerings. And that's really how long online learning has been around, 36 years now. There's all sorts of standards that are out there. It's a question of, when do we get to introduce those to our teachers and how do we do that and work with them to be able to be really supporting them in getting ourselves up to that higher level? And right now, a lot of it's just survival. there are a lot of good things that are happening. I know that I'm running out of time here, but there are a lot of really good things that are happening. A lot of good relationships are being developed between teachers and students. Discipline is at a historic low, which is amazing, especially for an assistant principal. Teachers learning technology, years of advancement happening at a rapid pace. Schools that have gone one-to-one that never would have otherwise. That's a really wonderful thing. Quieter classes. If you walk around, classes tend to be really quiet when the masks are on. There's going to be all sorts of different things with high stakes testing, and stress, of course, is through the roof. So lots of different things at play. So, you know, just the impacts of COVID, right? In a nutshell. Any questions? Not that there would be any on this topic.
Hi, Drew. We do have a question that came in, if you could answer it briefly.
Sure.
And it actually dealt with whether you felt that the students, have they been adaptable to these changes? And have you noticed any differences based on grade level?
So I have the good fortune of being able to work with teachers K-12, doing a lot of professional development. And I've heard across the board that it's probably in the neighborhood of somewhere between like 80 and maybe 85, 90%, somewhere in that range, that have been very adaptable. There does seem to be a 10% chunk of students that are really struggling in the hybrid modality, or if anything, online. And there's a lot of different reasons for that, but that 10%, we're using different intervention strategies to be able to help reclaim them and keeping, work with them while still maintaining social distancing and other things. So we're actually seeing a lot of success. It's just that it is true. There is struggle, but it's across the board. K-12, somewhere in the neighborhood of anywhere around 10% is genuinely struggling with that.
Thank you, Drew. That was really, really interesting. So we are going to move on to our next presenter, and his name is Connor McKelvey. He is from the Iroquois Elementary School, and he is going to be presenting Engaging Elementary School Students During a Pandemic.
There, can you hear me now?
I can hear you, Connor.
All right, perfect. Thank you, Drew. Thank you for having a great introduction, and thank you, Jennifer, for inviting me and introducing me. Drew did a phenomenal job of introducing what it is to be, in a classroom setting right now during a pandemic. And with being right down the road from him, we're seeing a lot of the same struggles, but we're also finding a lot of positives out of all of this. And I want to share with all of you what I've found through being able to engage my students through virtual learning as well as in-person brick and mortar learning in the classroom. So let me go ahead and share my screen. Okay. Hopefully that will load. All right, so what I want to discuss with you today is talking about how we can better engage our students in the elementary classroom during a pandemic. And obviously, going online in March, it was difficult for teachers to scramble and figure out, okay, what's going to be a great method or a tool that's going to be effective for us, or what can we find that's going to be effective to keep our students engaged while they're online. And I've learned a lot incredibly from March all the way up until now, being in school 5 days a week. We've been in school 5 days a week since the beginning of the school year at the end of August. And there's been a lot of effective tools that I have found that I want to share. And we as educators, we're always looking at ways that we can help our other fellow educators out. So I'm hopeful that, someone during this presentation will find a lot of this information useful that they can translate and then continue to use in their classroom as well. So just to give you let me go back to my beginning here. Just to give you a quick overview, my name is Connor McKelvey, and I am in my 4th year of teaching 4th grade mathematics and science at Iroquois Elementary School. I also did my student teaching in Dublin, Ireland, so I found a lot of effective strategies that worked there in terms of using technology. And I also spent a lot of time teaching in the Virgin Islands. That's where I spent my very first year of teaching. And there's my fellow class. But I also learned a lot there in terms of using technology effectively and also finding ways to engage my fellow students. I currently hold a certification. I was a Golden Apple recipient back in April of 2019. I'm also currently working on my master's degree right now at Slippery Rock University, working towards my pre-K to 8 mathematics and science master's. And coincidentally, even before pre-COVID, I was already getting a lot of instruction with using technology a lot in the classroom to help engage students, which is why I'm now the recipient or a Google certified as well as an Apple certified educator. And how that's been effective is there's been so many resources I've been able to implement and use and share with my colleagues that have helped them become successful and help their students become successful. But as Drew stated, there still is about a 10% population that is having a difficult time with online instruction or with being in front of a computer screen. So I've been looking at other ways that I can think outside the box to help them become engaging learners by finding other meaningful ways that I'm going to go through and give you an overview of next. So the first inner thing that I want to talk about was different online platforms. And when we first started online instruction online at Iroquois, we were not given a specific platform to use. And after a parent survey had been sent out, that was one of the main issues that a lot of parents and family members and students were having difficulty with, is it was like, okay, log on to this app here. Okay, then you need to click on this and log on to here, and then type in your ID to enter into this or use your QR code. And it was very dysfunctional and it was very stressful for parents and very stressful for students. And we had to find a way where we could make sure that all students were being engaged on one continual platform. And I know Drew had discussed, Schoology had come out in the early 2000s, and obviously they've made a lot of changes. They're still making changes day by day, where sometimes the platform does not work effectively. But for the most part, it has been a great resource to have students log on to one continual page and find all the information they need there. So how we conduct all of our information that we put online is we put it by week, which makes it very effective for a student that might be homesick or a student that's home in quarantine, waiting to come back to school. They know that they've been out for week 9. They can click on the folder, they can access those files, and then I have it down by day. So they can click on Monday's information. So whatever they missed on Monday, they can find all that information there. And then it's very helpful for parents too to know that, okay, I've completed everything in the folder. I know that my student has completed all the information that they need for that specific day. Another online platform that my students have been using this past spring during quarantine, and that my students this coming school year will be using this as well, is a platform called Weebly. It is an online platform where students can make their own websites. And this has been a great way to engage students because it's a very user-friendly application where they can add their text boxes, videos, images. And Dr. Jeanette Schnars was a phenomenal field trip coordinator where our students had the privilege to be able to go to Presque Isle, and we were taking a look at erosion. So during the spring, we were talking about how does erosion effectively, how is it changing the landforms around Lake Erie? And my students' challenge was to find ways they could leap to the rescue to save Lake Erie from erosion. So they conducted what was called a Lake Erie Advancement Project. And I just want to give you a quick little example of what some of my students have conducted. So I actually use the Weebly platform for my own classroom website on top of Schoology. But my students were able to create their very own website using this platform. And one thing that I really enjoyed about this is that students were really excited. And even though they were at home and they were learning virtually in the spring, this was something they could easily do online where they were conducting their own research. So this one individual, he talked about what strategies should we use to reduce erosion. He says that he lives less than a mile away from Lake Erie shorelines. Please help stop erosion on Lake Erie. So he was able to find some resources and insert some widgets with YouTube. He was also able to find some images on Google. And then they also had to give an introductory paragraph that described what was taking place. So this was something that really engaged students because I had so many parents that were texting me and emailing me saying, I can't believe you found something that my student's interested in. They love going on at home at night and they're coding their own website. And they were able to create different tabs that discussed different components of our science topic. So using the Weebly platform has been very effective, and I'm very excited and ecstatic to use it again this school year because it's something that can be done in school or it can be done, online if your students are virtual. So that's a great resource that a lot of students have been finding effective, and it's been keeping them engaged. I finally found something that clicked because there's been so many trial and errors this year where I've found different programs that have not been effective. And thankfully, this one has been something that has been effective for our students, and I hope it's something that would be effective for your students as well. So that is the Weebly platform that we've been using. The next topic that I wanted to discuss was some other ways that my students are engaged. And another meaningful way that's been engaging for my students is finding ways to assess my students to make sure they're understanding the material, but to make sure they're also enjoying what they're doing during that time. So when you're engaging students through assessments, you want to find tools that can be effective not only in a brick and mortar setting, but also virtually. And all these components that I'm going to be discussing with you have been very effective for me. These are the top programs that I've been using in my classroom that my students also enjoy. The first one that some of you might be familiar with is Flipgrid. How Flipgrid works is it is a 2-minute, well, you can actually set, it is a 2-minute video where students can record themselves talking about a specific topic. So for example, at the beginning of the school year, I had my students create a video where they were discussing at least one of their favorite activities that they did during the school week, just to kind of introduce themselves to what Flipgrid is. So we use it on a weekly basis, and every Friday we have what's called Flipgrid Friday. And students are familiar with every Friday we log onto our Chromebooks. They're able to access Flipgrid, and they're creating a video that's at least 2 minutes long that describes or discusses something in the mathematics or science related field. And the first time that we did this, we actually had students pretend that they were a reporter. So I want to give you a quick example, and hopefully the screencast doesn't lag. But this is one of my students who did a breaking news. Her video is only 33 seconds long, but it is a great way for me to assess to see if my students are understanding the concept. This was when they did a science lab based off of the properties of matter. We were looking at solids, liquids, and gases. So I'll try and do this, and hopefully it doesn't lag for you. You can get an idea of what Flipgrid is.
I'm going to tell you how I feel about the Ice Ice Baby. It was very good. It was very experiencing, and I love how you measure by percentage. It was so weird. That is all the way down. Right here, we want to on our earbud.
So what that particular student was discussing was they were describing how we were talking about the Ice Ice Baby Lab, where they were finding things that surprised them or was interesting to them. And it's a great way for those students that aren't very good with physically typing on a computer screen or with having to conduct their research, with having to write it out. They can actually verbally describe what they observed and what they noticed during their survey. And Flipgrid was a very meaningful application that students could use to find different ways to describe what's been effective for them during our pandemic. Another area that I've been using is something that I call Movie Math. This is through Google Slides. With being a Google certified teacher, I found a lot of strategies over the summer that have been effective for my students. And basically how Movie Math works is students log on to the Google Slides. It's already in present mode, and they click on whatever specific day of the week it is. I kind of call it my bell ringer on steroids. And I use students' names from the class, and they find during their Google slide, they find what the problem is. So we've been doing a lot with rounding lately. So for this particular problem, they had to round 83 feet to the nearest 100. So they were given 4 particular options. And it's assessing them because when they click on the correct option, it actually takes them to a short clip of the video. So we were rounding, so we were rounding up or rounding down. So it was appropriate to use the movie up for this particular week. And every single day, it's themed based off of that movie. And a lot of students are engaged because they're very into cinematography and into watching particular films that are interest of them. So being able to apply real world applications and using that to realize, okay, here's how you can effectively use mathematics, in the real world rather than just in the classroom. And they're able to apply that to a particular movie. And if they answer that particular movie correct, they get to watch a short clip from that scene. And if they get it incorrect, it's taking them to a screen where it discusses how they need to go back and review the material to find an effective way to understand that particular problem. So creating a Google slide and having it accessible with hyperlinks has also been an effective way to engage students in the classroom. A third resource that's been effective is called Edpuzzle. And Edpuzzle was new to me this summer, but Edpuzzle has saved me a ton of time because Edpuzzle is able to take questions that you create and embed them inside any YouTube video that's already been created. So for example, for this particular YouTube video, it was just all about me meeting Mr. McKelvey at the beginning of the school year. But wherever there's a dot is a question, and the YouTube video automatically pauses and a multiple choice question comes up onto the side of your YouTube video. and you're assessing the students based off of, are you really paying attention and watching the video, or did you just scroll all the way to the end and say that you watched the video? So the students aren't able to fast forward. They actually have to watch the video, and when it pauses, they have to answer a multiple choice question before it will let the video continue. And the program is actually already synced with Schoology. So as long as you have a roster, it tells you the exact score of what each student had earned after they are done watching the video, which is very effective. So when students click on the YouTube video, they can watch this, and that can be applicable to reading. It can be applicable to science, can be applicable to social studies. There's a lot of great resources that you can find a YouTube video that's already been created. And then you as the teacher can then go in and create an effective way to assess your students and keep them engaged while they're watching that video. So that was a new resource to me this summer when I had been working on my master's, and my students have been enjoying it. And I'm glad that I've found that resource that hopefully others such as you can also implement in your classroom. The fourth area that I've been focusing on is an area called boom cards. And boom cards are another effective way to engage students because it comes up as a flashcard on their screen, and it will not let them move on to the next flashcard until they've answered this correctly. So it's kind of similar to Quizlet, but with boom cards, they can be more engaging where they can actually interact with the flashcard that's in front of their screen rather than just typing it there. So this has also been an effective way for repetition, checking to make sure students are understanding the content. And that's been another effective means to keeping students engaged. Another area, I just tried this out last week. If you have not looked into Gimkit, definitely check out Gimkit because this is my students' new favorite activity. I used to use Kahoot, but there have been so many other applications that have been way more engaging than Kahoot that I don't even use Kahoot anymore because Gimkit is like Kahoot on steroids. Basically how it works is students answer questions on a team and their team can earn money. And then what they can do is they can save that money and they can sabotage other teams, like freezing someone else's screen. They can have someone's screen become blurry where they can't see it. They can change the background music. So there's a lot of different ways that Gimkit has been effective for students, especially with rounding, with looking at place value, and also examining other means such as science-related vocabulary. So it's been a very engaging way that students can not be looking just at one screen. They can have, a separate screen that they're using Gimkit for the question and then answering on a different device. But if you haven't looked into Gimkit, I highly recommend checking out that application because out of all the programs that I've shown you thus far, that is definitely the most engaging one that my students have found to be effective with assessing them to see are they understanding the material or whatnot. Another area is also quizzes. I also think quizzes is like Kahoot on steroids. Funny memes pop up in between each question if they answer it correctly, which they get a good chuckle out. They also can earn power-ups. They can also freeze other players' teams. So this is another way that you can assess students, and then you can go back and review to see have they answered the question correctly, or is there something we need to go back and review together as a class because so many students have been missing that particular category or that particular topic. So quizzes has also been another effective, engaging way to assess my students. And finally, if you are a subscriber with Smartboard with Smart Suite, this is another application. It's called Monster Quiz. And Monster Quiz is a team collaboration. So although, students are separated 6 feet apart at their desks, they can still collaborate with their team and be engaged, whether they're online virtually or whether they're in the classroom in a brick and mortar setting. And how this works is when they're doing their Monster Quiz is their objective is they want to try and hatch an egg. And you can see that there's a couple eggs there and they want their monster to hatch out of their egg. Well, every time a student gets an answer correct, their egg starts to crack and starts to shake. But every time a student does not get an answer correct and the egg will start to shake around and your monster gets stuck inside. And the objective for this is you want your student to be able to hatch their monster out of their egg, and then your team would end up winning. But you're collaborating, you're working together as a group, even though you're socially distant and you're separated. But this is also another effective means to engage students and also help them review content that we've already previously taught. And the great thing about Monster Quiz is you can gear the questions towards your specific students. So I personalize the questions to say, Mr. McKelvey was walking, through Lawrence Park and he noticed that there was 35 squirrels, over in Napier Park. Round how many squirrels there were to the nearest 10. So you're able to create your own questions throughout Monster Quiz and make it more personable and applicable to those particular students as well. And then the last category that I want to talk to you about was also different themes. Something that's been really effective for me, not only through in-person learning, but also through virtual learning, is coming up with a theme for your instruction throughout the week. So in April, when we were online, everything was geared towards the Titanic. So everything math related was about the Titanic. I created a virtual background like we were on the Titanic. So even though we couldn't physically be in person, we were still able to discuss the Titanic and discuss meaningful ways of how we can apply real world applications in math with the Titanic. During May for the 5th of May for Cinco de Mayo, we discussed tacos. We talked about ingredients and using fractions and how those fractions can be applied in a classroom setting with looking at fractions and finding out, okay, how much taco meat are you going to need? How many peppers are you gonna have to cut up? And then another area was I became a chef. So my students then also enjoyed changing their usernames on Zoom to, Chef Sam or Chef Colston or Chef Aaliyah. And they were able to pretend that they were working in a kitchen, and they had to effectively find different ways to add up your ingredients or subtract your ingredients. So there was a lot of math applications that we could apply. So I invite you and challenge you to find an engaging way to find students to be, engaged in your classroom and make sure they're excited to be online if you're, on a computer, in a hybrid setting, or also, if you're in a brick and mortar setting, you can still dress up, and enter your classroom as a specific character. And it certainly has been very effective in keeping them engaged. I have a lot of DJs that, come and visit our classroom that have also been a great way to keep my students engaged through music and through the arts. So I've been trying to find, a lot of arts infusion to keep my students engaged in the classroom. And it's a great way to assess them as well. Are they understanding the content? So, there's a light at the end of the tunnel. Although we've been finding that there aren't a lot of, ways that have been effective, there also are a lot of ways that are effective to keeping students engaged. And I'm hoping that, you found something during this presentation that's effective, that will help you in your classroom or help you, in a science lab to keep students engaged and excited to be in school, or to be online, even though we're trying to get through a pandemic. But, we're all in this together. We're all working together as educators and as professors. So I'm hoping that, you found something useful throughout this presentation that you can, take away and implement, into your classroom. So if anybody have any questions for me?
Connor, thank you so much. Those are really fantastic resources that definitely will be implemented, I'm sure, by many of the viewers. I know I can't wait to look up some of those websites, but right now we do not have any questions currently, so we are going to move on to our last speaker of this session. His name is Ben Tost, and he's with the Fort LaBeouf School District. He will be presenting classroom instruction in a corona climate.
All right, thank you very much.
Real quick, I left my main computer at home today, so I'm using a Chromebook, and I've been told that my volume isn't the best. So if you could just confirm for me if you can or cannot hear me.
We can hear you. You're good.
All right, thank you. All right, are you able to see my screen?
Not yet, no.
All right, we should be moving now.
That's good.
All right, thank you. So my name is Ben Tost. I am a high school biology teacher at Fort LaBeouf, and my opening slide has an experiment happening in a classroom, and unfortunately, due to the restrictions of the pandemic, there are some changes that are being made in the classroom. So my opening title, a new kind of classroom experiment, because literally we are in an experiment with the restrictions of the pandemic and restrictions of being able to work with one another. So at Fort LaBeouf, we have been with one another the whole time since we started in September. So some of the mitigation procedures that we have in place are plexiglass screening for teachers at their desks. Every student, as they enter the building, they are wearing a mask, and students have been given masks if they haven't had them. We have temperature screenings being taken to gain access, I guess, into the building. Many hand sanitizer stations, and obviously the 6 feet distance between one another. Some additional mitigation procedures is a home health screening survey that has been provided to every family. So they are encouraged to be aware of the signs and the symptoms and not to send their children to school if they may be having any of those symptoms. Teachers, staff, we have a daily survey that we have to fill out kind of with the same idea to identify whether or not we have any of those signs or symptoms and to help us decide whether or not we should or should not go to school. Within the halls, we have a lot of material to kind of encourage social distancing and also encourage proper hand sanitation and hygiene. When we send students to the nurse, we have some guidelines in place to make sure that we are not crowding that place. And the students that are going down there for concerns with regard to the coronavirus, they're going to a separate location or they are kept separate from those other students that just need to go there for everyday visits like medication. Contact tracing is a big part of the school day. Every teacher has been encouraged to spread their students out as much as possible within the classroom. And we have a Google form that is used to let us know when a student leaves. They fill out this form, where they're going, when they left, and when they return. So that makes contact tracing a little bit more manageable. And a big mitigation piece is just transparency. Whenever there is a new case within the community, within the district, we receive an email and the families receive emails. So that makes it really important to know that we're doing what we can to be safe and trying to make everybody as safe as possible. The last piece there on the mitigation procedures is that 100% buy-in. This is probably the most important. Our administration, our staff, and our students are all committed to being in school because we definitely believe that the classroom environment is the best place to learn. So we try to make sure that we are doing those things that we can to stay here as long as possible. So with regard to changes to teaching, there are some classroom procedures and as well as instruction procedures that have kind of been modified. The biggest one is with mask breaks. We are asked to provide our students mask breaks, or they may ask for those mask breaks. And so it's usually, I have a wristwatch and I set it for 5 minutes, and I have certain students remove their masks. And then after that 5 minutes is up, they put their mask up above their nose, and then another group is able to do that. So that kind of helps to remove the, I guess, the claustrophobia that a masking creates. But as time has gone on, there are fewer and fewer mask breaks as I think people are just getting more used to this lifestyle. The biggest thing is frequent mask reminders. The masks have a tendency to drop below the nose. So we do our best not to call out our students and really just use it as a teachable moment and a community building moment. And that's really important that we have to kind of continue to do that. I'm fortunate that my classroom has 3 sinks in it. So I have equipped all my sinks with soap, and we have towels. So I'm encouraging my students to wash their hands frequent times throughout the day. And I myself am doing it as well to model it and just for my own personal safety. A big thing is flexibility. Even before the coronavirus, flexibility with student work was always important. But now, with absences as a result of that survey, we really have to be able to work with families and work with the students to make sure that they're able to learn even if they aren't in class. And then finally, the biggest one is during one of our in-services, we received some mental health awareness training. And right now, we're so used to working with people, and now we're trying to isolate ourselves, and that's having a huge negative impact on mental health. So we are trained to kind of look for that within our student population and provide a staff referral should we see something. With regard to my instruction, fortunately, because I have students, I've had students in my classroom Monday through Friday for the full day, there hasn't been a whole lot of changes to my actual instruction. One change is there's less movement, so for the students and the teachers, which that is so difficult for me, and it's difficult for the students as well. But being planted at a desk with a plexiglass barrier is definitely not my style, and having students not interacting is not my style either. But it's something we do for to maintain that we can still be in the building together. That with the fact that I'm a science teacher, I try to have as many labs as possible. And some of the labs I've had to cut out or swap out. I do this one lab typically every year called the Bubbleology lab, where students are blowing bubbles. Couldn't do that one anymore, so I swapped it with another lab where they were reacting a substance in a solution and measuring the amount of bubbles that were created by it. So being able to be flexible and modify my labs is important. E-labs, I'm not a huge fan of them at all, but I can use those, particularly if I have a student that is at home temporary on a quarantine. I can use an e-lab as kind of an option. The biggest thing is, ever since I was in teacher training mode, they're always pushing cooperative learning. And there's a reason. Cooperative learning is fantastic. It's great learning with others and learning from others. So unfortunately, we've had to reduce that or rethink it. So one thing that I've been doing as a science teacher, I never really had a whole lot of writing, but now I decided I'm having my students write out, not just writing their notes, but I call it a WIB, a what is biology? It's always the same question, but it's answered in the context of the content we're covering. And I always tell them, it's worth X amount of points, but I always reward exceptional work. So I try to give them an incentive to go above and beyond the expectations of just writing out a vocab word in this definition. I think Connor mentioned Flipgrid. That's something I also use, and oftentimes I use that with processes. So, and I was doing this prior to the coronavirus, having students talk about cellular respiration, photosynthesis, explaining all the parts and pieces. As they move through the motion, I feel like they start to buy in and believe the words they're actually speaking. So the Flipgrid is an awesome tool in the classroom or from a remote learning perspective. Lastly, this is my means of rethinking cooperative learning, using Google Slides and creating a cooperative slide project that students can work on together. I'm actually doing that today and tomorrow, where students are going to be using these chalk pencils to write on their desks, take pictures with the Chromebook of the models of the cell going through the cell cycle. So a lot of things I haven't changed. The biggest thing is just this idea of making sure the students are wearing those masks and keeping distance, which is not easy whatsoever. These kids, they have a tendency to be magnetic and be attracted to one another. I did speak with another teacher as I was preparing this, just to give a perspective of what it looks like from an in-school class to a virtual class. Because at Fort Buff, we do have about 20% of our high school population is online. So as you can see there, we had to hire 4 additional temporary staff members to facilitate some of those courses. The big difference is comparing spring 2020 to fall 2020. We had this idea of a golden ticket. Essentially, if you did 70% of the work, you got 100%. That is no longer in play, thank goodness. But there is still some struggle, as some of the other presenters were alluding to. So comparing them side by side, looking at participation and performance. It's an honors bio class and a regular bio class, which is kind of like apples and oranges. But it's no surprise that in an honors bio in the classroom, we have 100% participation and 100% passing. And that's above 70%. In a regular bio in the virtual, this instructor informed that it was very low. It took a while to get going, but it is at 80%, 86% and growing. And there is about 60% passing. So these individuals are definitely having a hard time making those connections. And I really think finding the motivation, because as a classroom instructor, we are really big in driving the motivation, engaging our learners. So that's where I wrapped up. I'm no fortune teller, but I do anticipate having to go to a virtual format in the future. So I really appreciate, Connor, all those awesome tools. I'm really looking forward to looking into GimKit, but I know that I'm going to have to be flexible as this world is changing and moving forward. But if there's any questions, I'll certainly entertain them.
Thank you, Ben. And we do not have any questions at this time. So I did want to say that we appreciate you presenting, and it's been wonderful to listen to our presenters talk about the challenges that they have been facing. So we technically are finished with today's session. We will have a break until 4:30. Our poster session resumes then, and you can find that on www.discord.com and follow the instructions to log on. There is a link available on the Regional Science Consortium website at regsciconsort.com as well. And thank you very much. We'll see you back here tomorrow.